Chapter 7, Section 6, The Respiration of Cells and Tissues. This section was completed during the tenure of a fellowship of the John Simon Guggenheim Memorial Foundation. Most of our knowledge of the mechanism of cellular respiration has been obtained during the last 20 years. One of the most striking features of the modern picture of cellular respiration is the similarity of its mechanism. For in spite of minor variations which occur among different organisms, the major theme is common to all. A priori, one has no reason for expecting essentially the same mechanism of respiration in bacteria, yeasts, the higher plants, and man. Increasing knowledge may at first seem to result only in increasing complexity, but as the picture nears completion, its universality results in a profound simplification. During an earlier era, biochemists searched for an enzyme which would directly catalyze the oxidation of glucose by molecular oxygen. No such enzyme has been discovered, nor is it likely that such an enzyme exists. The studies of the last 2 decades have shown with great clarity that the oxidation of organic foods occurs within living cells by a series of graded steps involving many distinct enzymes, each reacting in a cyclical manner, and the whole constitutional mg, an integrated reaction system smoothly oxidizing sugars to carbon dioxide and water. These studies have slyly that many intermediate compounds are formed along the pathway of glucose oxidation, and that these intermediate molecules are rapidly formed and rapidly degraded, with specific enzymes controlling the rate of formation and degradation of each substance. Each partial reaction of formation and of degradation must have a velocity at least equal to the velocity of the overall reaction of respiration. Not only is the life of an intermediate molecule short, but its concentration may be very low. The series of graded chemical reactions results in a stepwise release of the potential energy of the organic foods, rather than its release in a single explosive reaction. The stepwise liberation of energy results in efficient utilization, for the transfer or coupling of the energy will be most efficient when the reactions are operating at or near their equilibrium points. Though this ideal situation will be rarely attained, it is much closer of realization in a series of graded steps than in one overall reaction. Living cells are unique among the engines known to man, for the living engines are constructed of the same kinds of materials that are used for fuel. If the fires of life burned too brightly, not only the fuel, but the engines themselves would be consumed. If the potential energy of a sugar molecule were to be released in a single reaction, the released energy might frequently activate the structural molecules of the cell, resulting in their destruction by oxidation. The series of graded reactions never liberates enough energy in any one molecular reaction to surpass the activation energy of the structural molecules, and their stability is not endangered. Physical chemistry furnishes a theoretical basis for understanding the apparent complexity of cellular respiration, its kinetics, and its mechanism of energy transfer. Perhaps physical chemistry has served even more in furnishing powerful tools to probe within the living cell and to separate and study the individual enzymes. Such tools as the spectroscope, spectrophotometer, ultracentrifuge, and an electrophoretic apparatus have been used in conjunction with the more usual biochemical methods. And as a result, the physicochemical data and interpretations are so intertwined with the biochemical that any attempt to unravel them would destroy the fabric, leaving isolated threads with little meaning. In the pages which follow, an attempt will be made to present the general pattern of cellular respiration, with an effort to emphasize the physico-chemical approach, whether its contributions be theoretical or as tools in the hands of biochemists. 1. The rate of respiration. The rate of respiration is normally measured by following the uptake of oxygen per unit time. Some typical examples are shown in Table 29. Considerable disagreement exists as to the best method of comparison between organisms. Frequently, the comparison is made on the basis of the O2 consumed mg, or GM dry weight hour. Reference to dry weight is fairly satisfactory, but the amount of non-protoplasmic material, such as cellulose walls, hair shells, etc., is very variable between organisms. In many cases, wet weight is equally satisfactory and is sometimes preferable. For some purposes, the amount of oxygen consumed is referred to unit total enteroprotein N. This is usually a matter of convenience, though at times it is more reliable than dry or wet weight. It is observed that in mammals, the respiration per unit weight of small species is much higher than that of large species. Brody has proposed that the basal metabolism of adult mammals is related to the weight as follows. This equation appears to be valid to about plus or minus 11% for all mammals, from 0 to kilograms. Mice to 600 kilograms, cattle and horses. This is illustrated in the metabolism of birds likewise decreases per unit weight with increase in size. Their metabolism is not represented by the same curve as for the avian. Benedict. The Physiology of the Elephant, Carnegie Inst. of Washington, 1936, tab.e for different organisms. Soleus muscoe at rest. Solus musco, active, A. Krogh, Comparative Physiology of Respiratory Mechanisms, Univere, Pennsylvania mammals. Brody and Proctor give the relation between metabolism for 24H or HRS in cal and weight in kilo. For birds, as Rubner and Voigt propose that for mammals the basal metabolism is constant for unit surface area. The historical background has been summarized by Dubois and Lust-Dubois height. Formula gives excellent results in predicting the metabolism for men. And the surface area formula is of greatest value as a basis of comparison of the respiration Dubois basal metabolism in health and disease. Philadelphia, 1924, of large and small animals within a species. For comparisons between adult individuals of different species, surface area seems to be no better than Brody's weight formula. The surface area comparison is only valid for related organisms. The metabolism of cold-blooded vertebrates may not be predicted by an equation used for mammals. The surface area concept has been used uncritically in an attempt to explain the high respiration of some microorganisms, without the realization that different microorganisms with similar surface areas per cell may differ in their metabolism by 1,000 times. For lower organisms which do not store food, there is a real question of what constitutes the basal metabolism, for the metabolism may be 100 times as great in the presence of an exogenous substrate as is the endogenous rate, B. Activity. Even more striking changes in respiratory rate occur in the same tissue upon changed activity. Dormant cells, tissues, or organisms usually have a low rate of respiration, and when growth occurs, there's a sudden burst of respiration. This is illustrated by the increased respiration upon fertilization of sea urchin eggs, or the breaking of diapause in grasshopper embryos. A striking illustration occurs when dormant seeds germinate. Mary and Godardi found a rate of 0.06 millimeters VGTChat-AIHR for dry barley seeds. 1 hour after adding water, the rate had increased 386 times, and in 24 hours to 6,500 times the dormant rate. Perhaps the best-known example of explosive changes in respiratory rate are those associated with muscular activity. The rate in frog muscle increases tenfold in tetanus, while the overall O2 consumption of man may insert 240 to 250 milliliters per minute at rest to over 4,000 milliliters and 2. Oxygen supply and diffusion. Rate of respiration depends not only on internal factors, such as the concentration of respiratory substrates and enzymes, but also upon the oxygen supply. It is probable that for the majority of land organisms, oxygen supply is rarely limiting. This is not always the case for organisms in deep ponds, at great depths of the ocean, in marshes, or where decay is rapid, or perhaps in some soils. In these environments, the oxygen tension may determine the respiratory rate. The oxygen content of water in equilibrium with the air depends upon the partial pressure of oxygen in the atmosphere, the temperature, and salt content of the water. The relation is shown as when the respiratory rate falls off with decreasing pressure of oxygen It is difficult to determine whether the result is due to failure of diffusion to furnish an adequate supply of O2, or to the effect of the O2 tension on the rate of oxidation of the cellular oxidases. However, it is generally assumed that the cellular oxidations are independent of oxygen tension, except at the very lowest values, less than a few ml. Though this assumption is probably not fully justified, it may be made as a simplifying case. The problems of O2 supply and diffusion have been reviewed by Crowe, Gerard, Hill, Tang, and particularly by Jacobs. The reader is referred to these references for details. A few of the applications are presented here. A general discussion of diffusion is found in Sec. 1, Chap 1 of this book, calculated from Peters and Van Slyke. Quantitative Dinical Lineastry, Volume 1, p. 555. We will first consider oxygen diffusion into an aquatic organism, which is a plain sheet of tissue like the marine alga, ulva. Ulva is 2 cells thick, with an average thickness of 34 N. We will assume that the oxygen consumption of the tissue is uniform throughout its mass. We may ignore any gas diffusion from the edges and realize that O2 may enter from either surface. If the oxygen in the water is in equilibrium with that of the air and the water is replaced sufficiently rapidly at the surface, the external pressure of O2 will remain constant. We may use Warburg's equation for determining the O2 pressure at the center. In this equation, we have used the atmospheric pressure of O2 and not the molar concentration of O2 dissolved in the liquid. Crowe has shown that this is justified, and the solubility constant is then included in the diffusion coefficient. When we apply this equation to UHA using 1 LMB's value for the respiratory rate, 2.6 millimeters, dry WTHR, and assuming dry weight is 5 of wet weight, we obtain a P2 at the center of 0.99 atmospheres, or very nearly the outside pressure. The equation may be given in a form to determine at what depth a given pressure, C, may obtain. The equation is then, for the particular case of the center of the sheet, x3 of 3 of the thickness, or 2, and the equation simplifies. If we then take C, 0.2 atmos, the respiratory rate the same as Ulm, 8.710 mmn tissue, and at the center equals 0.01 atmos. The maximum thickness equals 0.35 millimeters. This would be the maximum thickness of a plain sheet of tissue with the respiratory rate of ulva and without any circulating mechanism if the O2 tension at the center is to remain above 0. If the tension just at the center is to fall to 0, the limiting thickness is increased to only 0.36 millimeters. If the respiratory rate were that of L-Gilvay, the thickness would be increased to 1.1 millimeters. Cytoplasmic streaming might allow a somewhat greater limiting thickness. We find, in fact, that aquatic organisms which have no blood transport have greatly irregular surfaces and are branched or hollow or circulate water through tissue openings. With the higher respiratory rates of mammalian tissues, the limiting thickness, when the circulatory system is non-functional, is even less, about 0.2 millimeters. Similar equations can be derived for cylindrical respiring tissues and for spheres. The equation derived for cylinders by Fenn and by Gerard is for a frog nerve of 1 millimeters. Radius Fenn calculated that when in air with a respiratory rate of 1.2310 ml 2 ml of tissue, at the center is just about 0. When the pressure of O2 at the center is just 0, U equals 0. R equals 0, the external pressure necessary to supply the oxygen is, or the limiting radius for a given external pressure with 0 pressure reached just at the center, is for a spherical organism without internal circulation. And in water in which Pu equals 0.2 atmos, with a respiratory rate of 100 millimeters VGTHR, the radius must be just under 1 millimeters, or diffusion cannot supply the necessary oxygen. Land organisms usually allow the entrance of air into tissue or intracellular, large spaces of leaves, trachea of insects, etc., since the diffusion of O2 in air is nearly 300 times as rapid as in water. Considerably greater thickness is possible where lungs with forced ventilation and blood transport are involved, the diffusion path may be reduced to a few microns, and great size is possible. The problems of tracheal respiration have been discussed of higher plants have been discussed by Brown et al. Since the rate of CO2 absorption in photosynthesis is usually at least 10 times the rate of O2 absorption in respiration of the same tissue, and since the P2 equals 155 millimeters and the P2O is 0.3 millimeters, it is apparent that photosynthetic tissues should not normally have respiration limited by O2 diffusion. It has been calculated that for a barley leaf, the water diffusion path is only E5, the leaf thickness. In the chloroplast, the calculated steady state for diffusion would be set up in barley leaves in 0.9 sec. In the trunks of large trees, where tissues are present which are potentially able to respire, either some mechanism of longitudinal O2 transport may exist, or these tissues may obtain their energy by fermentation with an export of the end products to the well-aerated leaf. Further investigation of this problem is indicated. 22A Crow, Comparative Physiology of Respiratory Functions, Philadelphia, 1941, D. T. McDougall, and E.B. Working the pneumatic system of plants, especially oxygen tension and the respiratory rate. There are few data in the literature on the effect of 7.2 on the respiratory rate, where the factor of diffusion can be eliminated. Some of the problems involved have been discussed by Gerard, Warburg, and Kubovy, having investigated the problem in the bacterium Micrococcus. They set up conditions in which the rate of invasion of the liquid by O2 from the atmosphere was not limited. And they found that the respiratory rate at 1 degree Celsius was independent of 7.2 above atmos. 7.6 to 1010 Niemer. Winsler used the dropping mercury electrode to investigate the relation between 7.2 and respiratory rate for Baker's yeast. In the absence of substrate, the rate was independent of 7.2 at all tension above 0.2 millimeters. At 20 degrees Celsius, in the presence of glucose, the rate was independent of 7.2 above 2.5 millimeters. At 20 degrees Celsius, temperature has a marked effect on the 7.2 respiratory relation. Winsler found that for Aston glucose, the respiratory rate is independent of at 0.25 millimeters. At 5 degrees Celsius, while at 34.3 degrees Celsius, the rate becomes independent only above 4 millimeters. Winsler, by a clever use of the known inhibitory effect of Keogh on respiration of yeast, has shown that the respiratory rate is being limited at low O2 tensions, not by the diffusion of O2 into the yeast cells, but by the rate of reaction of O2 with an enzyme. In mammalian muscle, the O2 is brought to the muscle as oxyhemoglobin. The 7.2 of arterial blood is approximately 100 millimeters, while in venous blood approximately 40 millimeters. We may assume that the average pressure in the capillaries is 70 millimeters. In mammalian muscle, the distance from the muscle cells to the nearest open capillary probably varies from 50 gene in rest to between the capillary. He assumes venous 7.2 and the muscle cell to maintain the observed O2 consumption. Y arrives at a maximum difference in tension of 46 millimeters and a minimum of 1.8 millimeters if the oxygen tension in the capillary is 70 millimeters and in the muscle cell is 3 millimeters. An adequate supply of O2 is available by diffusion. Many muscles contain myoglobin. The dissociation curve of myoglobin has been shown to differ from hemoglobin. Since myoglobin is approximately 50% saturated with oxygen at 3.26 millimeters, at 37 degrees Celsius, and apply of 7.4, and 0% saturated at 20 millimeters. O2 pressure. At a lower TIAC, a saturation occurs at lower O2 tensions, and since the muscle 7H is probably nearer 6.8 than 7.4, the above values are probably too high. Thus, a marked gradient may exist between the capillaries and the muscle cell, and so long as the myoglobin is more than 50% saturated, the intracellular PO2 must be at least 3 millimeters. 2. Warburg and F. Kubovitz, Biokern, Zichier 214, 5, 1929. 33A. Krav, The Anatomy and Physiology of Capillaries, New Haven, 1941. Living cells receive their energy supply from the potential energy stored in the organic foods absorbed, or in the case of green plant cells, from the foods produced in photosynthesis. The diet, or photosynthesis, furnishes only a few of the organic compounds required by a cell, and for a cell to survive and grow, many different kinds of substances must be synthesized from the few foods of the diet. For these syntheses, a continuous energy supply is essential, and in addition, energy is necessary for movement, osmotic work, transport, and the maintenance of man-wise steady states. Cellular inspiration is the fundamental mechanism by which the potential energy of the organic compounds is released as kinetic energy and made available for the work of the cell. The organic foods are oxidized in a stepwise manner by a series of catalysts, the respiratory enzymes, and the potential energy is likewise released in a series of graded steps. Much of the kinetic energy is in the form of heat, and this energy is useless for the cellular work and is wasted except as it serves to maintain body temperature in the mammals. However, the cell has various contrivances for utilizing some of the liberated energy. In Sec 1, Chap 3, is found a general discussion of thermodynamics. Here we wish to consider a few aspects of the energetics of respiration. The quantity of heat formed upon the oxidation of a compound may be determined by burning it in a bomb calorimeter. Some typical results are shown in H calculated for Kikios for biological oxidation in the mammal. If the N was oxidized to nitrate, the value would. One of the great achievements of physiology was in showing that the amount of heat liberated in the oxidation of food in the body is the same as in the external burning of the same foods. From a knowledge of the amount of oxygen absorbed and the ratio of Ko2 liberated to G8 absorbed, the respiratory quotient, or LLQ, it is possible to predict the heat formed. These results are ably summarized by Lusk and Dubois. However, it is the free energy change, ppr mole, rather than the change in heat content. A, which is the more useful term for two reasons. 1. It is the variable of AF, which determines whether a particular reaction will occur spontaneously. 2. The maximum amount of work which may be obtained from a reaction is related to ATA, not AT. When there is a decrease in free energy, alpha is negative, a spontaneous reaction is possible. When there is a large increase in free energy, a large and positive, a spontaneous reaction is impossible. If AAF is positive to the extent of 3 to 4 cal per mole, a spontaneous reaction may be possible at physiological temperatures by absorbing heat from the environment. For a reaction of larger positive AF to occur at physiological TC temperatures, the reaction must be energetically coupled to a reaction of AF so that it will be recalled from Chap 3 and 6. 1. That the relation between AF and A is where AF A, and as are in terms of cals per mole, and T is the absolute temperature, as is the change in entropy. For a discussion of the physical meaning of these terms, reference is made to Chap 3 of Sec I, however. It may be recalled that the entropy is a measure of the randomness of a system. An increase in entropy means decrease in organization. A decrease in entropy, an increase in orientation. The entropy term also determines the maximum work which may be derived from a chemical system. When as equals 0, as it does at equilibrium of all reactions, the maximum work equals F for a thermodynamically reversible system, as C0. Such chemical reactions probably rarely occur in biological systems, but the reversible oxidation reduction systems approach theoretical reversibility and as approaches 0. Such reactions will be the most efficient for the transfer of cellular energy, and the nearer EF Dubois, Basal Metabolism in Health and Disease, Philadelphia, 1924. They operate to the equilibrium point, the more efficient they will be. Thus, we find another reason for the stepwise oxidation in cells and the stepwise liberation of energy. Though the oxidation of organic foods occurs in many separate steps, the total free energy change is the same as if the oxidation occurred in one step, for the value of AF is determined only by the initial and final states. and not by the pathway. A knowledge of the value of each proposed partial reaction in cell metabolism is useful in predicting which are possible and impossible steps, but it does not tell us which of the possible steps actually occur. For a knowledge of the actual pathways of cellular metabolism, it is essential that we determine the actual intermediate substances which are formed, the enzymes which catalyze their formation and disappearance, and the kinetics of their appearance and disappearance. The general aspects of chemical kinetics are presented in Chapter 2 of Sec 1. Here we will discuss a few aspects in relation to enzymes. When glucose is oxidized to CO2 and H2O, there is a large decrease in free energy. F is about L704 cals. Thus, glucose in the presence of oxygen is thermodynamically unstable. However, in the absence of the appropriate enzymes, glucose is not oxidized measurably by molecular O2 at physiological temperatures. The rate of a chemical reaction is determined by the frequency with which the reacting molecules may surmount an energy barrier. This is illustrated in Figure 33. The energy barrier may be thought of as the energy essential for the rearrangement of the atoms in the molecule to form an unstable configuration, the reactive molecule or complex, or the energy necessary to form a reactive compound with a second molecule. This may be illustrated as follows. Free energy of reaction then is the reactive molecule and acts the reactive compound. The value of AF for the conversion of U2 is the same in both reactions, but the value of the activation energy is not necessarily the same. If A equals ZOO and the energy of the activation for equation 2 is less than for equation 1, A is a catalyst. A molecule may gain the energy for climbing the energy barrier by molecular collisions, by chemical reaction with a catalyst, or by a combination of the two methods in Sec 1, Chap 2. The mechanism of evaluating the energy of activation is given. It is determined from the Arrhenius equation, which is given below, where K velocity constant, the base of natural logarithms. An inspection of the equation shows that the rate of the reaction depends upon exponential functions of the activation energy and the reciprocal of the absolute temperature. At any given temperature, the kinetic energy of translation of the molecules is given simply by and at 25 EAC. This equals 0.89 cals per mole. However, at any moment, the energies of the individual molecules will vary considerably. The fractional number of molecules with energies equal to or greater than any particular value of EA is given by the relation. The values of NAGN are shown in Table 33 for two temperatures for various values of EA. It will be found by calculation that increasing the temperature from 25 degrees to 37 cm causes an increase of but 3.97% in the average energy of the molecules, 0.89 to 0.925 cals, but that the number of molecules with high energies has increased considerably. At 25 cals by 25 cal, another relation which follows from the Arrhenius equation is that a small decrease in activation energy means a large increase in rate. For example, decreasing the activation energy from 20 to 15 cals results in an increase of the rate by 1,300 times, while having the activation energy results in an increase of the rate by 500 times. Energy of activation, thus catalysts, which may only decrease the activation energies by a few calories, may result in tremendous increases in rate. Cytochrome oxidized cytochrome succinate dehydrate, data from IWW. Scissor advances in enzymology, 1943. The activation energies of some catalyzed reactions are collected in from the previous discussion. It would appear that reactions in which the energy of activation was high 50 cal or over would not occur at a measurable rate. In fact, some such reactions are known as the denaturation of proteins and the inactivation of enzymes. Here the energies of activation are very high. Arrhine and others have pointed out that the energy of activation of the Arrhenius equation is really a heat of activation and not the free energy of activation. The free energy of activation is related to the heat of activation as follows, where alpha I and N are the free energy. Heat and C-enteroicleonge of activation. Now, Ig may be calculated from EA of the Arrhenius expiation by the Fol-Lowing relation. The rate of the reaction depends upon F rather than upon heat of activation. In many reactions, the on term is small, and no GRAR at error is made in using the Arrhenius equation. However, if N is positive and large, F may be smaller than a piche, and a rapid reaction may occur even when Ia equals 50 to 100 cals per mole. This is the case in protein denaturation work at 5 degrees Celsius, and at pH and a rapid rate of denaturation. Ehring, note 37, has pointed out that the correct formulation of the Arrhenius equation is other constants as above. For a discussion of the derivation of this equation, the reader is referred to mechanism of enzymatic catalysis. An enzyme increases the rate of a reaction by dekineizing tlie an energy barrier of a straight molecule must pass. This may be due either to a decrease in the heat of activation or an increase in the entropy of activation. The net result is the same, a decrease in activity. The detailed mechanism is unknown, but certainly the critical step is the formation of an enzyme substrate molecule. In most cases, the evidence for the intermediate molecules comes from kinetic data, and this has been discussed in Sec 1 Chapter 2. However, in a few cases, spectroscopic evidence exists for the presence of Ya, such as the compound between catalase and ethyl hydrogen peroxide, or between it should be noted above that the enzyme actually enters into the reaction. The conventional definition of catalysts says that the enzyme remains unchanged, and the equilibrium point is unchanged by the presence of the enzyme, and for a complete enzymatic cycle, this is true. However, if the partial reactions are studied, we find that in all oxidation or reduction reactions, the enzyme itself undergoes oxidation or reduction. For each partial reaction, the enzyme does undergo chemical change. However, since one enzyme molecule may react several hundred to several thousand times a minute, an extremely low concentration of enzyme may catalyze a large amount of substrate. And for practical purposes, the equilibrium point will not be shifted by the concentration of enzyme present. Classically, WC think of oxidation as the addition of oxygen to a molecule, and this may be illustrated by the oxidation of hydrogen to water. This type of oxidation is one of the least common methods of oxidation. Two other types of reaction are also considered as oxidations. One of these is the removal of hydrogen from a molecule, and may be illustrated. We say that succinic acid has been oxidized to fumaric acid, even though no oxygen is involved. The other is the removal of electrons from an atom or molecule as follows. We say that ferrous ion has been oxidized to ferric ion. In oxidations of these two latter types, free hydrogen or free electrons are not formed. Hence, there must be present some substance which will combine with either the hydrogen or the electrons. These are designated as either hydrogen or electron acceptors. The reverse of oxidation is reduction, and the removal of oxygen or the addition of hydrogen or electrons is reduction. Michaelis and Schuberin have developed the idea that, basically, all oxidations are the removal of electrons from the molecule oxidized. In the case where hydrogen is removed from a molecule, we can think of the process as occurring in two steps: the liberation of protons or acid dissociation and the liberation of electrons or oxidation. This concept of Michaelis, that all oxidation is the removal of electrons, may seem strange at first sight, particularly because many of the hydrogen atoms which are removed are not those which we are in the habit of thinking of as dissociating, such as, however, Michaelis's interpretation has the advantage of unifying the concepts of oxidation and of electron removal, and of pointing out that the removal of electrons and protons from an organic molecule need not occur simultaneously. Organic compounds usually have an even number of electrons, so that in their oxidation or reduction, we are accustomed to think of two electrons added or removed simultaneously. In many, perhaps the majority of the instances of oxidation, This is not the case, as only one electron is added or removed at a time. A has an odd number of electrons and is known as a free radical or a semiquinone. As an example of a semiquinone, TVA may take the case of quinhydrone. This particular substance is insoluble in water and precipitates as a double or dimeric molecule of quinhydrone. However, many organic dyes, such as pyocyanine, may exist in solution in the half-oxidized state as semiquinones. The evidence for the existence of half-oxidized molecules or cerniquinones is based on 3 lines of inquiry. 1. Results of potentiometric oxidation studies. 2. Absorption spectra. 3. Magnetic properties. Michaelis and Schuberta have reviewed the evidence for the existence of cerniquinones and their wide occurrence. The semiquinone may react in one of several ways. The concentration of semiquinones in biological oxidation systems is often 10 to 50% of the total concentration of a particular substance, or it may be very much less. Their significance probably lies in the fact that they may speed IQ, the rate of cellular oxidations, because an oxidizable substance, which exists as a semiquinone, may accept or donate 1 or 2 electrons at a time, and thus they may function between a compulsory univalent system like ferroferricytochrome and a bivalent system like succinate fumarate, where the concentration of semiquinone is extremely low. In the absence of an intermediary semiquinone, a trimolecular reaction would be necessary. We know that trimolecular reactions are too slow to account for the observed rate of biological oxidations. In the absence of a catalytic semiquinone, the rate of the reaction would depend upon the extremely low concentration of the free radical of the substrate, rather than upon the concentration of the total substrate. If an intermediary substance, R, is present in which R represents its semiquinone, we may diagram the reaction as follows. We may expect that some of the enzymes of cellular respiration may exist temporarily as cerniquinones. And this is true for those intermediary enzymes, like the flavin and di- and triphosphopiridine nucleotides. Biochemists had long searched for a respiratory enzyme which would catalyze the oxidation of glucose to QO2 and H2O2. The indophenol oxidase and catechol oxidase were discovered in the last century. However, they did not act on glucose, and their significance was unknown. Definite progress in this field came in the 1920s from two different sources. Warburg's studies on the effect of KOO and HCN on the respiration of intact cells, and Keelan's spectroscopic observations on cytochrome and its correlation with cellular respiration. At the present time, no worker in the field holds the view that there is a unique respiratory enzyme bringing about the reaction between cell metabolites and molecular oxygen. Rather, respiration has been demonstrated to be a complicated process, catalyzed by a series of enzymes functioning together in a cyclical manner to form a connected series of reactions, a reaction system. Several such reaction systems have been identified, and a few of the component enzymes well studied. In a few cases, the quantitative data indicate that most of the respiration may pass over one or another of these systems. In other organisms, several systems may occur even in the same cell. It is almost certain that other respiration systems, as yet little studied, exist. Nature of respiratory enzymes. The respiratory enzymes which have been isolated are all conjugated proteins. That is, proteins which on hydrolysis yield amino acids, and in addition, non-amino acid residues, such as heme riboflavin phosphate, thiamine diphosphate, etc. Proteins do not readily undergo reversible oxidation and reduction, and, in all cases which have been studied, it is the ion protein part of the enzyme which is readily oxidized and reduced, such as the iron of the heme or the pyridine ring of triphosphopiridine nucleotide. Some oxidation enzymes may be separated by dialysis into two parts, a specific protein and a coenzyme. The specific protein, which because of its high molecular weight, remains within the dialysis sac, and the coenzyme which dialyzes out. It is found that neither the specific protein nor the coenzyme has catalytic activity by itself, but if the coenzyme is added to the specific protein, the catalytic activity is regenerated. The protein is usually heat labile and the coenzyme heat stable. In some cases, as the flavin enzymes, dialysis alone will not separate the enzyme into two parts, but dialysis against 0.013. HCl does, while the separation of copper from catechol oxidase requires dialysis against HCl. Whether a particular enzyme is classified as one with or without a coenzyme is only a question of the value of the dissociation constant. For example, certain dehydrogenases readily separate into two parts. Some enzymes, as cytochrome, have not been reversibly separated into a specific protein and coenzyme. That is, the equilibrium must be far to the left. That is, the dissociation constant must be very small. In this case, the heme is referred to as a prosthetic group rather than a coenzyme. The same coenzyme or prosthetic group may occur in several different enzymes. For example, diphosphopiridine nucleotide, cosimase, occurs in several different dehydrogenases, and protohone is a constituent of the enzymes catalase, peroxidase, and cytochrome peroxidase, as well as the oxygen carrier, hemoglobin. The coenzymes and prosthetic groups are found to be reversibly oxidized and reduced apart from the protein, but usually the oxidation and reduction are sluggish. Combination of the coenzyme with the protein not only determines specifically with which substrate the coenzyme may react, but tremendously speeds up the rate of the reaction. In some cases, the protein stabilizes the semiquinone form of the coenzyme, for example, in the flavin enzymes. We will retain the name enzyme for the catalytically active molecule and refer to the dissociated molecules as specific protein and coenzyme. Euler refers to the enzyme as holoenzyme, and Ply dissociated products a respiratory system usually consists of the following components. An oxidase. Oxidases are enzymes which are directly oxidized by molecular oxygen, autooxidizable, and in turn reduced by some component of the respiratory mechanism. The best known oxidases are cytochrome oxidase, polyphenol oxidase, D-arnino acid oxidase, and xanthine or aldehyde oxidase. The oxidase is usually reduced by a carrier to a carrier. A carrier is a substance or enzyme oxidized by one component of the respiratory system and reduced by a second component, which is not the final substrate, usually another carrier or a dehydrogenase. Occasionally, an autooxidable substance may function as a carrier if the rate of autooxidation is slow compared to the rate of catalyzed oxidation. Thus, some flavoproteins may function either as oxidase or as carriers. Cytochrome, cytochrome reductase, and the catechol compounds are the best-known respiratory carriers. Two or more carriers may function between an oxidase and a dehydrogenase, for example, cytochrome and cytochrome reductase. The dehydrogenase systems may be connected to carrier, coenzyme I, and probably coenzyme the second may function as carriers. 3 dehydrogenases. Most respiratory metabolites will not react directly with either a carrier or an oxidase. The metabolites are oxidized by substrate-specific enzymes, the dehydrogenases. The metabolite donates 2 hydrogen atoms to the dehydrogenase, which is reduced. In exceptional cases, a single enzyme may react with both a cell metabolite and molecular oxygen, for example, xanthine or aldehyde oxidase. For substrates, metabolites, the substrates respired in cells usually are either carbohydrates or products derived from carbohydrates. Often the substrates may be phosphorylated compounds, such as glyceraldehyde phosphate. The substrate, which has become oxidized by the dehydrogenase carrier oxidase system, may react with a second dehydrogenase and be further oxidized, or it may react in a cyclic system, as outlined below. Amino acids and fatty acids may constitute respiratory substrates. 5. Decarboxylation system. In the initial oxidation of a substrate, no CO2 is produced, and only 2 hydrogen atoms of the substrate are oxidized at a time. Most of the CO2 of respiration has its origin in the decarboxylation of keto acids, either directly by the decarboxylation of such keto acids, as pyruvic acid by the carboxylase system through oxidative decarboxylation, or through a system such as the Krebs cycle, where a cyclical condensation, oxidation, decarboxylation system functions and results in the complete oxidation of 3 carbon compounds to CO2O and H2O6 inorganic oxide. The activity of many respiratory enzymes depends upon high presence of inorganic ions. Among the ions known to be essential for cellular respiration showed that the respiration of many cells and tissues is partially or nearly completely inhibited by low concentrations of cyanide or sulfide, and Quillen has shown that azide acts similarly, since these compounds form firm complexes with iron and its compounds. Warburg suspected that iron was the catalytically active material, and since it was known that CO forms compounds with ferroiron, and particularly with heme compounds like hemoglobin, Warburg investigated the effects of CO on cellular respiration. He found that the respiration of yeast was inhibited by CO, and the inhibition depended upon the ratio of CO, and not upon the partial pressure of CO, while the cyanide inhibition was independent of O2 tension. He suggested that HCN combined with the ferry enzyme, while CO and O2 competed for the ferroenzyme. Since the inhibition respiration was the same, whether the yeast was respiring glucose, alcohol, or acetate, Warburg assumed that a single enzyme was undergoing inhibition. Warburg named the enzyme the Sauerstoffliebhertragen fermenter Atmung, or the oxygen-transporting enzyme of respiration. In England and America, the enzyme is commonly known today as cytochrome oxidase. Warburg made the simplifying assumption that when respiration is independent of O2 tension and substrate concentration, the rate of respiration is directly proportional to the concentration of the enzyme combined with O2 today, we would say, upon the concentration of ferry enzyme. Lai then formulated his results as follows, where K is the relative affinity constant for CO and O2. Now, if the maximal rate of respiration without CO is 1, and the residual respiration in the presence of a particular ratio of O2, the inhibited respiration is 1, on the basis of the above assumption, N should be dependent upon N since N and E, and it may be directly determined by measuring the respiration with and without CO and O2 are known. K may be evaluated. Warburg found in yeast at 20 degrees Celsius that K lay between 9 to 10 when the substrates were glucose, alcohol, or acetate. When 7I equals 1N, or at 50% inhibition, K is the ratio of KO2. For yeast, this means that the concentration of CO must be 9 times that of O2 for 50% inhibition. The dimensions of K should be expressed in terms of the molar concentrations of CO and O2. It is in fact given in terms of the partial pressures of the gases in the gas space, and since the solubilities of CO and O2 are not identical, K includes the ratio of their solubilities. Warburg's formulation is only correct if the rate of respiration depends only upon the concentration of in feed. This is probably never completely true. It may be approximately true in some cases, where the substrate concentration, oxygen tension, or other factors limit the rate. The above formulation is not valid. Warburg attempted to correct the equation for substrate concentration, and as Fischer has pointed out, his formulation is mathematically incorrect. Though one may derive a mathematically correct formulation, so many simplifying assumptions must be made. It is doubtful whether any simple equation presents the kinetic picture with any validity. Cyanide, azide, and sulfide all accordingly with the ferry form of trienzyme, Slannard and Armstrong and Feslier have studied the relation between the concentration of these poisons and the respiratory realized that. It must be clearly realized that in many cells and tissues, only part of the respiration is inhibited late, the inhibition as follows. If the maximal respiration equals 1, and the respiration at any particular azide concentration is N, the inhibited respiration is 1 and N. Making the assumptions used above, if the log 1 and W be plotted against log NJ, a linear curve should result, the slope of the line giving C, or the number of molecules of a Z inactivating 1 molecule of enzyme. For the caffeine-induced respiration of frog muscle, the azide inhibition depends upon one molecule of azide, combining with each molecule of enzyme, see figure 34. And lies between Sirot and a similar formulation may be given for cyanide. Armstrong and Fislier and Fisher and Olnu have given a summary of the results. For cyanide, C lies in different organisms between 0.52 and 1.6 in K, between 10 and 10 John. C should theoretically be a whole number. Deviations may be caused by inhibition of more than one metabolic step, or due to the fact that the kinetics of respiration flow not depend only upon the concentration of the enzyme. Warburg and Negeline investigated the effect of light of various known wavelengths and of measured intensity on the reversal of the CO inhibition of respiration in wild yeast. Torula, Udelich measuring the respiratory rate manometrically. From the results obtained, they could plot the relative photochemical absorption spectrum for the coenzyme. The absolute absorption spectrum was also determined by Warburg and Negeline, and to be similar to a coheme compound, but it was not identical with that of any heme pigment known at that time. Warburg and Negeline investigated the absorption spectra of the CO compounds of spirophos heme coupled with the globin of hemoglobin and with fesohamin B prepared from chlorophyll H by reduction with high and replacement of the Mg with fat. The spectra were similar to that shown in figure 35. Warburg, therefore, felt that his enzyme was a fesohemin, and this view is generally accepted. Recently, Melnicke has determined the photochemical spectrum of co-cytochloroid oxidase in extracts of rat heart, and he has obtained a marked band at 450 NJX and lower bands at 510 and 589. MX Phillips has determined the similar bands of cytochrome oxidase in a membrane from the chick embryo, and obtained bands at 435 to 445, 553, and 589 MX. Though neither of these spectra is identical with that of Kubowitz and Haas, they are very similar and are typical of the spectra of heme compounds. It is now certain that cytochrome oxidase is a component of Warburg's oxygen-transferring enzyme. However, it is to be realized that cytochrome oxidase is a generic term, and one must not expect identical spectra for the enzymes in different organisms. It has been generally believed that cytochrome oxidase is firmly anchored to the structure of the cell, or to cell particles, or to macromolecular constituents. The heart oxidase preparations of Keelan and electric lose most of their activity on repeated filtration through Seitz filters. Stern has found that oxidase activity is partially in the sediment when centrifuged at 1000 G gravity and completely so at 10,0 G. He believes that the oxidase is linked to a macromolecular particle of a diameter in the range of 50 to 200 mx. Haas has obtained, for the first time, water-clear solutions of cytochrome oxidase, in which 80% of the activity remains in solution after centrifuging at 10 G for 100 minutes. If the initial material is centrifuged at 10 G, the solution contains 20 to 40% of the activity. By treating with ultrasonic waves at 30 degrees Celsius, he obtains active water-clear solutions. 5. Cytochrome. Kylinum rediscovered in 1925 a class of heme pigments in living cells, which had first been seen by MacMahon in 1886. The significance of MacMahon's discoveries had been lost until Keelan extended MacMahon's observations and correlated these heme pigments with cellular respiration. Kylan named these pigments the cytochromes, and in his original publication recognized three separate pigments, cytochromes A. Kylan observed that in a contracting muscle of bees' wings, a sharp four-banded spectrum appeared, that at rest the bands became weak and diffuse. Under anaerobic conditions, in activity, or in the presence of cyanide, the dark bands reappeared. Similar observations were made on yeast under aerobic and anaerobic conditions. Kylan demonstrated that organisms from many phylogenetic groups contain cytochrome, though the relative concentration of the three pigments varied. and in some cases one or another of the pigments was absent. Kylan proposed, and in later studies has demonstrated, that the cytochromes represent a new class of respiratory enzymes, the carriers intermediate between an oxidase and other respiratory components. Cytochromes I and C are not oxidizable, but are catalytically oxidized from a ferro state with distinct bands to a ferri state in which the absorption is more diffuse. Cytochrome B is sluggishly autooxidizable. Kylan correlated his findings with those of Warburg and proposed that the cytochromes were oxidized by a cyanide-sensitive co-inhibited oxidase, which is probably identical with Warburg's enzyme. Recently, several additional cytochromes have been identified. Cytochrome AI, a 2 and bi, occur in some bacteria lacking cytochrome A, such as Bacillus coli cytochrome, as occurs in heart muscle and in yeast, and will be discussed more fully below. Cytochrome 62 has been found in preparations from yeast. There is possibly a cytochrome Ci in heart. The preparation of concentrated cytochrome C, and Rudolf has purified such preparations. Cytochrome E is a water-soluble red heme protein of relatively low molecular weight, 18.0, fairly stable to heat, and non-autooxidizable, nor does it form compounds with NaFHCN in the pH range of 4 to 9. This is probably due to the fact that over this pH range, the iron is attached by 6 coordination bonds, while at higher or lower pHs, it is attached at only 4 positions and can thus form compounds. At physiological pH, cytochrome C is readily oxidized by molecular oxygen, only in the presence of a specific catalyst, cytochrome oxidase. It is reduced by cytochrome I catalytic activity of cytochrome C. May be easily demonstrated, may be made from heart muscle, which are practically free of cytochrome C. These preparations have scarcely any activity in the oxidation of cysteine p-phenylenediamine, or hydroquinone, cytochrome C, alone will not catalyze the oxidation of these substrates by molecular oxygen. A complete system gives a rapid O2 uptake, as is shown in figure 37. This catalyzed oxidation is heat-labile and is inhibited by HCN NANs and photoreversibly in figure 37. Cytochrome oxidase from the cytochrome concentration determined by Haas, and from the rate of O2 uptake of yeast in glucose at 20 degrees Celsius. Warburger calculates that the cytochrome in living yeast must be oxidized and reduced about 4,000 times a minute. It has been known for a long time that many tissues could oxidize a naphthol and dimethylphenylediamine to indophenol blue, and this property was ascribed to an indophenol oxidase. Recent work has made it clear that the indophenol reaction is not due to a single enzyme, but to cytochrome oxidase and cytochrome C functioning in a manner similar to that diagrammed above for the oxidation of cysteine. Haas has determined the rate of respiration in Baker's yeast by following the rate of cytochrome reduction. This was done by measuring with a photoelectric spectrophotometer the difference in absorption of aerobic and anaerobic yeast at 550 mAd. His conditions, 0 degrees Celsius, well-washed yeast, no substrate, were such that in air all the cytochrome was in the ferri form and in a nitrogen atmosphere in the ferro form. He found the concentration of the cytochrome equals ISX RNMML cell suspension. Then, preventing the reoxidation of cytochrome by adding cyanide at equals 0, he determined the rate of cytochrome reduction. The results may be expressed by the relation. He found that C equals 4. To convert this into terms of oxygen, we may recall that 1 mole of cytochrome is equivalent to HEI mole of oxygen, and that REI. Direct determination of the oxygen consumed gave, for the same yeast, that OAGCl 34 millimeters. The exact seriation of the cytochrome actions in respiration is still unclear. It is possible that cytochrome AZ can oxidochrome C, and the latter may have a higher potential than cytochrome BC. Cytochrome and cytochrome oxidase. There is some reason to believe that cytochrome as maybe cytochrome oxidase. The avidity for this view has been ably summarized by Keelan and Alartre, and may be briefly given below. Cytochrome is thermolabile and soluble. It is rapidly reduced by biological systems, and it forms compounds with HCN, NaNA, and H2S. In the ferro state, it forms a compound Whitley CO, absorbing at 590 MeGa and 432 MPa, which is similar to results obtained by Warburg for the oxygen-transporting enzyme. Cyanide forms compounds with both ferro and ferri, but the cyanide compound is not readily reduced. The CO ferro as appears not to be light-sensitive, and this is contrary to what would be expected of cytochrome oxidase. Straub and Yakushiji and Okunko have prepared from Liart muscle colloidal solutions containing cytochrome A, and in Straub's preparation was free of HCl. Straub finds that his preparation forms a compound with CO with a shift of the gamma band from 443 mx to 430 mZ. He observed that p-phenylenediamine is oxidized only after the addition of cytochrome C, and Yakushiji reports cytochrome A is partially reduced by C. No data are available as to light reversal of TO inhibition, and inadequate evidence as to the catalytic activity of these preparations is available to decide whether cytochrome A2 is really the elusive cytochrome oxidase. Hydrogen mobilization and transport. A. Dehydrogeases. The cytochrome oxidase cytochrome system is primarily concerned with the transfer of electrons from the intermediate enzymes to oxygen. Wieland has emphasized the fact that the respiratory substrates normally lose 2 hydrogen atoms upon oxidation, or 2 protons and 2 electrons. Thunberg discovered a class of enzymes, the dehydrogenases, which catalyze the oxidation of SG-gesific substrates by the transfer of 2 hydrogen atoms to a hydrogen acceptor. Thunberg used methylene blue as an artificial hydrogen acceptor and followed the course of the reaction by observing the decolorization of the methylene blue to its reduced or leuco form. Such a reaction may be illustrated as follows. Succinic acid, fumaric acid. Today, a large number of dehydrogenases are known, which catalyze the oxidation of a wide variety of specific substrates. Some of these are shown in Table 36. As dehydrogenase preparations, washed bacteria or yeast, toluene-treated bacteria, F-phosphate extracts of seeds or animal tissues may be used. Several dehydrogenases have been concentrated, and a few are known in essentially pure form. Most dehydrogenases consist of 2 parts, a specific protein and a coenzyme. At the present time, 2 coenzymes for dehydrogenases are known. Each coenzyme may act with several different specific proteins. See Table 36. Phosphopyridine nucleotides. Hardin and Young discovered chyzimase, a coenzyme essential for alcoholic fermentation by yeast juice. Its chemical nature remained unknown until the 1930s. In 193, Warburg and Christian were studying O2 consumption of an artificial system consisting of glucose 6-phosphate, a yellow protein from yeast, and a protein fraction, zwischen ferment, from yeast. And they found that this system required a coenzyme which differed from the chyzimase of Hardin and Young, and therefore they called it coenzyme 2. The Warburg groups isolated this coenzyme from horseblood and showed that it was a dinucleotide containing 1 molecule of nicotinic acid amide, 1 of adenine, 2 of root preparations from animal tissues, Ristolin formed from heart, Ristolin formed from yeast. Root preparations from animal tissue, from seeds and boreas, culframeus in purified state, from horse red cells, root preparations from horse. Seeds, animal tissues, and yeast for yeast, specific protein for animal-specific protein for plant-specific protein data from F. Schleich in a symposium on respiratory enzymes, Univi, Wisconsin Press. Warburg showed that when glucose 6-phosphate was oxidized, the coenzyme was reduced by the acceptance of 2 hydrogen atoms, and that it was the pyridine ring which underwent reduction. The reduced coenzyme could be reoxidized by the flavin enzyme. Coenzyme chyzimase was soon obtained in pure form and it was shown to be identical with coenzyme 2, except that it had but 2 phosphorus atoms. Warburg renamed coenzyme 1 diphosphopyridine nucleotide and coenzyme 11 triphosphopyridine nucleotide. Both coenzymes are widely distributed, for they are known from yeast, bacteria, and higher plants and animals. The evidence is very strong that many of the dehydrogenases dissociate into two whole parts, the protein and coenzyme. It is equally evident that the catalytic activity is a property of the complete dehydrogenase molecule. We may formulate this as follows, where Ka is the dissociation constant of the enzyme. In practice, K may be determined by using a small amount of specific protein in the presence of an excess of substrate, in the example used here, glucose 6-phosphate, and with increasing amounts of coenzyme, follow the velocity of the reduction of the coenzyme. It is essential that conditions are such that the velocity of the reaction depends upon the concentration of the enzyme, and that the concentration of the specific protein is sufficiently small and its dissociation constant sufficiently large, so that the concentration of total coenzyme is essentially that of free coenzyme. Then the J-Michaelis-Menten equation, see Saccharide chap 2, may be used. The rate of the reaction may be followed spectrophotometrically at 340 milligrams. For both coenzymes, I and 2 absorb strongly at this wavelength in the reduced form and but slightly in the oxidized form, see FAG 39. The value of K for glucose 6-phosphate dehydrogenase has been determined by Negeline and Haast and found to be 1.1 x 10 moles lighter at 38 degrees Celsius. Similarly, the alcohol dehydrogenase has been studied by Negeline and Wolf. The protein has been isolated and shown to be coenzyme I-specific. The dissociation constant has been determined, and in this case is different for the reduced and oxidized enzyme. We may formulate the equation for the dissociation of enzyme into specific protein plus coenzyme. 1 in enzyme. 112 into specific protein plus coenzyme TH2, just as was done above. The K of oxidized enzyme was found to the activity of the SPC-specific proteins may be followed by adding them to coenzyme I or 11 and lowering the oxidation of the coenzyme. This is illustrated in figure 40, where phosphoglyceraldehyde-specific protein is added at 0 minutes to coenzyme I, phosphoglyceraldehyde, and acetaldehyde. The coenzyme is reduced. Then acetaldehyde-specific protein is added and the coenzyme is oxidized. The ODI steady state to a new value. C. Succinic dehydrogenase, one of the most active and widely distributed enzymes of animal tissues, is succinic dehydrogenase. This enzyme occurs in most cytochrome oxidase preparations and is oxidized by cytochrome E oxidase, cytochrome C system. So far, the enzyme has not been obtained in solution, nor has it been separated into a coenzyme and a specific protein. No intermediate between the dehydrogenase and cytochrome C has been established. Dialyzed preparations lose much of their activity, which can be restored by the addition of calcium ions and aluminum or chromium ions. This enzyme seems of particular importance because it may serve to link other dehydrogenases to cytochrome C. It is not certain whether succinic dehydrogenase is widely distributed in plants. The observations of Hill and Bagbying would indicate that it occurs in bean seeds. D. The flavin enzymes, it is apparent that a substrate may become oxidized by its dehydrogenase, and the dehydrogenase become reduced. But if the dehydrogenase is to act catalytically, it in turn must become oxidized. The great majority of dehydrogenases cannot be oxidized by molecular oxygen, nor directly by ferricytochrome. Some intermediate carriers must be present, or some oxidase which reacts not only with the reduced dehydrogenase, but also with molecular O2. Warburg and Christian in 198 isolated a yellow protein from bottom yeast, which could be separated into two GURs, a colorless protein and a mononucleotide. The latter is now known as riboflavin phosphate, or the phosphate ester vitamin G. This protein could oxidize the reduced form of coenzyme second. And in the presence of O2, the flavoprotein was reoxidized, and H2O2 was formed. The structure of the flavin is shown in Figure 41. Warburg and Christian reconstructed a respiratory system using glucose 6-phosphate coenzyme the second, a specific protein, Swishen enzyme, and flavoprotein, which is diagrammed below. The reaction with O2 is slow, particularly low O2 tensions, and there is hot that aerobically a broad band appeared to thrive the blue, with its peak 460 mg. Anaerobically, the band disappeared. This behavior and the structure of the band were typical of a flavoprotein. He determined the total concentration calculated as flavin, and the rate of oxidation of the flavin by adding anaerobic ally, methylene blue, to the bacteria. The flavin concentration was X10 nm per velocity constant 0.99. This gives an oxygen equivalent of 1.2 millimeters VNDAN suspicion. He observed in direct measurements an O2 consumption of 1 millimeters LC suspension by thus all the O2 consumption could be accounted for by the flavin enzyme. Warburg and Christian have calculated the turnover number of the flavin that would be essential for the observed oxygen uptake in this bacteria, and they obtain a value of 80. And since they found a turnover number for flavin enzyme in cetolized horse red cells of 50, all the respiration of lactobacillus at 38 degrees Celsius may be catalyzed by a flavin acting as an oxidase. The turnover number equals 2 moles of C enzyme moles of O2 consumed per minute. The results of Haas and Warburg and Christian do not allow us to determine what flavin enzyme is involved, since several have similar absorption spectra. It has frequently been suggested that a flavin enzyme is responsible for the cyanide-resistant respiration of animals and higher plants. These organisms do not accumulate H2O2, as is typical of flavin oxidation by molecular O2, but the nearly universal presence of catalase would prevent that. The low turnover number of cytochrome reductase and the original flavin enzyme of Warburg and Christian, which are respectively 2.2 and 27 at 25 degrees Celsius, in air, and their strong dependence upon O2 pressure make them appear unlikely as oxidases. The O2 consumption of cyanide-resistant leaves is frequently 400 millimeters, wet WT, and this requires a flavin concentration of approximately 1x10 molar if the flavin enzyme is the oxidase. However, some as yet undiscovered flavin enzyme may well be active in this catalysis for D-amino acid oxidase, which is a flavoadenine protein, has a turnover num of 1400 at 100% E cytochrome reductase. Haas, Eilericher, and Hognes discovered in 1940 a new flavin protein which has the same prosthetic group as the flavoprotein of Warburg and Christian, but which is rapidly oxidized reductase. Rapidly oxidized dehydrogenase systems which are coenzyme to second DAS specific. It thus furnishes a connecting link for these systems with cytochrome C, cytochrome oxidase, and O2. The new enzyme has but slight reactivity with molecular O2 or with coenzyme artificial systems have been reconstructed, and their kinetics followed reconstructed system is shown in the diagram below. Coan 2 plus cytochrome reductase O-coan plus leukocytoclarome reductase leukoferrione cytochrome ferros of the velocity constants of the partial reactions have been determined. With cytochrome C has been established. The velocity condissociation constant K flavin, enzyme of Warburg, and Christian flavin, enzyme of Haas-aloxazine adenine, deoreolide cytochrome reductase. Coenzyme front. The cytochrome reductase of Haas at Ali is not reduced by coenzyme I, and therefore cannot link coenzyme I dehydrogenases to cytochrome C. There is uracil IgG in liver dispersions, an insoluble enzyme which reduces cytochrome C and oxidizes reduced coenzyme I. The nature of this reductase is still unknown. Other flavin enzymes, Warburg and crystallizated a protein which had flavin adenine dinucleotide as the prosthetic group. This protein is very active in the oxidation of many D-amino acids to NACE3 and the corresponding alkylo acid. Since the naturally occurring amino acids are one amino acids, the physiological function of this enzyme is unknown. It has long been known that an enzyme existed in tissues and milk which would oxidize xanthine and lipoxygen uric acid and acetaldehyde to acetic acid. This enzyme is known as xanthine oxidase, or Schardinger enzyme. Has purified xanthine oxidase, and his preparations contain a flavin adenine dinucleotide. Several additional flavoproteins have been isolated, but their physiological function has not been established. If catechol present lactobacillus delbrueckii type flavin enzyme dioxymalic acid oxidase peroxidase 1 peroxidase, I inhibited peroxidase, it's not inhibited. Respiration catalyzed by the cytochrome oxidase cytochrome C-sum is widespread throughout living organisms, but is not universal. There are organisms known whose respiration is not inhibited by the poisons cyanide, azide, sulfide, or photoirreversibly by carbon monoxide, and which do not have cytochrome. We know less of the mechanism of the respiration in such organisms than in the cytochrome oxidase organisms, but several features are known. It must also be realized that cytochrome oxidase organisms may contain additional oxidative mechanisms, so that several respiratory mechanisms may exist side by side in the same organism and even in the same cell. Cyanide-resistant respiration, commoner, has reviewed the effects of cyanide on cellular respiration. In many cells and tissues, a residual respiration exists in the presence of cyanide, and in some plants or parts of plants such as chiwe and in certain bacteria, no inhibition occurs. The nature of the oxidases responsible for this respiration is unknown. It has frequently been proposed that the flavin enzymes are responsible, and though this suggestion may be probable, it is not established. HAZ-resistant respiration. Stanardon has shown that the respiration of stimulated or contracting frog muscle is inhibited by azide, cyanide, and photoirreversibly by CoA, but that the respiration of resting frog muscle is not sensitive to azide or CoA, but is inhibited by cyanide. The nature of the oxidized active in resting muscle has not been established. See catechol oxidase. The polyphenol oxidases have been known since the work of Bertrand in 1894, and these have been recently reviewed by Nelson and Dawson. However, it is only recently that they have been purified and their role established. In 1929, Keelan showed that the catechol oxidase of potatoes was inhibited by HCN, H2S, and CoA, and that, unlike cytochrome oxidase, the CO inhibition was not reversed by light. Kubelvitsens has isolated a copper protein from potatoes which has high catechol oxidase activity and has shown that the activity depends upon the copper content. He was able to remove the Cu by dialysis against HCN and to show that neither the Cu nor the protein was active alone, though he could regenerate the enzymatic activity of the protein upon addition of Cu. The catechol oxidase of potatoes oxidizes orthodiphenols such as catechol to orthoquinones. Meta and para compounds such as resorcinol and hydroquinone are not oxidized, and monophenols such as phenol and pyrazole are only oxidized after a lag period. Catechol may itself act catalytically as a carrier for a small amount, say 0.01 milligrams, may be used to oxidize several mgs of p-phenylene. In the latter case, coenzyme the second and the siEAFIC protein for glucose 6-phosphate are essential. The whole reaction may be diagrammed below. Thus, any system which functions through nucleotide could be oxidized over X or catechol oxidase. The naturally occurring diphenols are probably not catechol, but glucoside derivatives of catechol. Kubowitz's preparations were very active, 1 milligrams, of enzyme catalyzing the uptake of 34,500 millimeters O2 per hour. At 20 degrees, it means that each molecule of enzyme went through over 30 complete cycles of oxidation and reduction per minute. Catechol oxidase is widely distributed in higher plants and in fungi. That catechol oxidase functions catalytically in cellular respiration appears to be very probable from the results of Baker and Nelson, who have shown that the respiration of potato tissue is largely 2-divided by icin and 4-nitrocatechol, and the latter is a specific catechol oxidase poison. Further, they showed that the addition of small amounts of protocatechuic acid produced a marked stimulation in respiratory rate, RQ, of 1, which continued for several hours, and the total extra 2 consumed was several times greater than would account for the oxidation of protocatechuic acid. Protocatechuic acid or other substituted catechols are probably the natural carriers which function in cell respiration. D. Other copper enzymes. Several additional polyplanolases are known for which no respiratory role has been established. For example, Lacoste and tyrosinase have been isolated and shown to be copper proteins. Polyphenol oxidase has also been reported to occur in animal tissues. Ascorbic acid is widely distributed in plant tissues and is in some cases rapidly oxidized in injured tissues. Protein, which specifically oxidizes ascorbic acid, has other heme enzymes. Ytterochrome and presumably cytochrome oxidase are heme enzymes in which the hemes are not identical with the heme of hemoglobin. However, a series of heme enzymes are known which have the same heme for prosthetic group as occurs in hemoglobin. Some of the mammalian blood acetic bacteria, Hick embryo extract, rat heart heme enzymes, and their spectroscopic properties are referred to in Table 39. Each enzyme has, however, its specific properties, none of which is identical with that of hemoglobin. That is, the different proteins impart different catalytic activities to the separate enzymes. Typical of these enzymes are catalase, crystallized by Sumner and Dounce. Catalase forms a compound with the substrate ethyl hydrogen peroxide of sufficiently long life to be demonstrated spectroscopically, and this is the first case of the physical demonstration of an intermediate molecule between substrate and enzyme. The enzyme is extremely active, for one molecule may decompose 10 molecules of H2O2 per second at 0 degrees Celsius. No clearly established function of catalase in cellular metabolism has ever been shown except to get rid of H2O2 produced in some oxidations. Cytochrome C peroxidase is a heme protein isolated from yeast that oxidizes cytochrome C at the expense of H2O2. Thus, the H2O2 may be used to bring about respiration by way of the cytochrome system. Peroxidase has long been recognized as an enzyme present in higher plants, which will catalyze the oxidation of some organic compounds, phenols, phenylenediamine, etc., by H2O2. It has been isolated by Theorelpcore from horseradish and by electrophoresis resolved into 2 crystalline fractions, both heme proteins, which he has named peroxidase and periperoxidase. Further, he has separated peroxidase into a white protein in heme and then has regenerated 93% of the original catalytic activity by combining the protein with heme prepared from H-hemoglobin. Peroxidase photons, a compound with H2O2, which is green in color in the presence of an oxidizable substrate. This intermediate is ephemeral. The physiological role of peroxidase may really be due to its oxidase activity, for Sweden and Theorelp have shown that peroxidase has catalytic activity in the oxidation of dioximalic acid. It had been shown that many plants contain an enzyme oxidizing dioximalic acid to dioxytartaric acid, dioxide. Dioximalic acid occurs in plants, and if dioxysuccinic acid is rapidly and reversibly reduced by dehydrogenases, the peroxidase dioximalic acid system may constitute an oxidase carrier system. Periperoxidase oxidizes dioximalic acid only in the presence of phenols, but unlike peroxidase, this oxidation is not inhibited by HCN. Thus, either cyanide-sensitive or cyanide-resistant respiration could be catalyzed verdoperoxidase is a green heme protein isolated HET from leukocytes of man, which has peroxidase activity, but is distinct from the horseradish enzyme. 124H-theorel, Art Chemi Minor. GL 14b, number 2019-40, 16b, number 24/1942. 16. One of the characteristic features of the respiration of living cells is the production of carbon dioxide. However, the oxidation of many substrates mentioned. Less is known concerning the mechanism of the formation of CO2 than is known concerning the oxidation of hydrogen to water. Both processes are, however, equally important to an understanding of the mechanism of cellular respiration. Carboxylase is an enzyme discovered by Newbergen yeast and is widely distributed in higher plants. It catalyzes the decarboxylation of keto acids to the corresponding aldehydes in CO2. The enzyme prepared from yeast cheese is a diphosphothiomin magnesium protein, which in alkaline solutions dissociates into a specific protein and diphosphothiomin, pyridosphate, ester of vitamin BI. The coenzyme was discovered by Hagen and Lomami, and Schuster demonstrated its vitamin character. Divalent manganese or cobalt can replace magnesium without loss of activity. The yeast enzyme is active with pyruvic and oxaloacetic acids, but is inactive with a ketoglutaric acid. The plant type of carboxylase has been considered absent from animal tissues, but Evans and his colleagues have shown that pigeon liver contains an enzyme which decarboxylates oxaloacetic acid to pyruvic acid and CO2. The AF for this reaction for ionic reactants is 5.25 cal, and the equilibrium constant is 4.9x10. This means that the equilibrium is far to the right, but the reaction is reversible, as they have shown with the use of CO2, which contain radioactive carbon of atomic weight 11. One may calculate that at 0.01 m pyruvate and 0.02 m NACOs. The equilibrium concentration that other types of decarboxylating enzymes are present in animal tissues is clear from Green's preparations with pig heart. He found a diphosphothiomin protein which is inactive with oxaloacetic acid, but acts on a ketoglutaric acid, forming succinic semialdehyde. With pyruvic acid, CO2 and acetylmethylcarbonyl were formed. Pyruvic acid is formed in nearly all cells and tissues, and may be formed in many cases either aerobically or iatobically. TL and DREA are probably several mechanisms for the formation of pyruvic acid, but the scheme shown in the diagram below is probably a fairly well-established mechanism. This scheme is the result of the work of many PO, Ley, notably Kelmbeden, Parnas, Meyerhof, Korai, Lomaim, and Warburg. Several recent reviews of this scheme have been published, and here the scheme will be presented diagrammatically. 1. Oxidative decarboxylation. Baron, working with Gonocoxy, had discovered that pyruvic acid could undergo aerobic decarboxylation. Lipmont has shown that Lactohasius delrucci carried out the same reaction, and that active extracts could be prepared from vacuum-dried bacteria. Littman showed that the complete system required at least the mechanism of the oxidation of acetate has long been a mystery. Recently, Slade and Workman have shown by the use of the heavy carbon isotope that the bacterium Aerobacter indogenesi can condense two acetic acid molecules to succinic acid. 138, an excellent review, C.S. Ochoa in E.A. Evans. The biological action of the detailed mechanism is unknown and may involve intermediates. The acetate may then be oxidized as succinate by one of several mechanisms. Petersch had shown that in vitamin B deficient pigeons, pyruvic acid accumulated in the bloodstream, and that upon administration of vitamin B, the pyruvate disappeared. He showed that vitamin B thiamine would maintain or increase the respiratory rate of preparation centrations of added cocarboxylase on M fumarate, 0.5 m's, and adenylate the rate of oxidation of pyruvic acid in acid, 0.0014 m, on the oxidation of dispersions of brain from thiamine pyruvic acid by dialyzed dispersions of deficient pigeons at 3 degrees Celsius, from I Normal pigeon brain with optimal Conbanga, S. Ochoa, and R. A. Peters, centrations of and diphosplanothia of pigeon brain from vitamin B-deficient birds, when pyruvate or lactate was the respiratory substrate. Ochoa and Petersing showed that the pigeon brain would synthesize diphosphathiamine from H3PO4 and thiamine, and that it was the diphosphate derivative which was active in the enzyme systems, see figure 4. In pigeon brain preparations, the oxidation of pyruvate is not complete, but proceeds beyond acetate. The activity of this system requires not only specific proteins and organic phosphate, diphosphathiamine, MGE, but also a decarboxylic 4-carbon acid and adenylic acid, 2 4-carbon decarboxylic acids. St. Georgii proposed that decarboxylic 4-carbon acids played a catalytic role in cellular respiration. The evidence upon which this was based consisted of 2 parts. One part. Malonic acid is known to inhibit succinic dehydrogenase, the enzyme which oxidizes succinic acid to fumaric acid, and the respiration of many tissues, particularly pigeon breast muscle, is inhibited by malonic acid. Fumaric, but not succinic, acid can overcome the inhibition of malonic acid. 2 low concentrations of fumaric acid will cause a marked increase or maintenance of respiration of pigeon breast muscle. And the extra oxygen consumed is much greater than would account for complete oxidation of the fumarate to CO2. And H2O in the St. George scheme of 4-carbon acid, oxalacetate, at one end of the cycle was presumed to be reduced to malate. By receiving hydrogen from a substrate, and the hydrogen was then passed through the cycle of 4-carbon acids by way of succinate to cytochrome C, and the cycle was regenerated. There is considerable question about the validity of the St. George cycle, and it has been reviewed by Sternbaumann, Elliott, and adversely by Balk and Potsierdor al. The significance of St. George's work has not been in the particular cycle he proposed, but in his pointing out the catalytic activity of the 4-carbon decarboxylic acids. The recent experiments from Corey's laboratories him to show clearly that succinic acid acts catalytically in the oxidation of glucose and pyruvic acid. A particular mechanism in which these acids may act catalytically is the Krebs side. 3. Krebs tricarboxylic acid cycle. We have seen that neither the carboxylase system of yeast nor the aerobic decarboxylation mechanism of bacteria will account for the complete oxidation of pyruvics and lactic acids, or of trioses to CO2 and H2O, yet complete oxidation of these compounds does occur in virtually all cells. The most original and fruitful approach to this problem has been that of Krebs in what he calls the tricarboxylic acid cycle. While some of the details of the original Krebs cycle may not be generally valid, the fundamental idea that pyruvic acid is oxidized by synthesis to a larger molecule, and then by stepwise oxidation and decarboxylation to CO2 and H2O, is probably valid for reviews of the Krebs A. St. George on oxidation, fermentation, vitamins, health, and disease. Williams, Krebs, and Johnson, in a study of pigeon breast muscle, discovered a reaction between fumarac and pyruvate, which occurred in the presence of malonic acid, inhibits the succinic dehydrogenase. The succinate could not have been produced through the reduction of fumaric acid. Further, the oxidation of pyruvate was inhibited when malonic acid was added in the absence of added fumarate. But upon the addition of fumarate, each molecule brought about the oxidation of one molecule of pyruvate. If renolinate is absent, succinate does not accumulate, but is oxidized to fumarate, and the reaction continues. This is the equation for the complete oxidation of pyruvate. In muscle poisoned with arsenite, Krebs found that pyruvate and fumarate gave rise quantitatively to aketoglutarate. Since at USL is known that the oxidation of citrate stops with aketoglutarate in the presence of arsenite, Krebs proposed that citrate is an intermediate in the oxidation of pyruvate. The evidence is as follows. 1. Fumarate may be readily converted to oxalacetate by the enzymes fumarase and malica dehydrogenase, which are known to be present in muscle. 2. Oxalacetate and pyruvate react anaerobically in pigeon muscle to form citrate and CO2. 3. Low concentrations of citrate act catalytically in the respiration of carbohydrates and pyruvate. 4. The rate of synthesis of citric acid is conversion to a ketoglutarate, and the rate of the degradation of the latter to succinate are all high enough to account for the observed respiration. Krebs then summarized his cycle as follows. Adding 4a through 4 gives equation 3 above. Deoxylacetate pro at Krebs and W.A. Johnson, Enzymologia 4 through 148, 1937. Diced in 4F can react with another molecule of pyruvate and go through the cycle again. Each turnover of the cycle will then completely oxidize one molecule of pyruvate to an H2. Krebs has emphasized that he has omitted coenzymes and intermediates. Inspection of the formulae of the compounds would suggest intermediates in reactions, 4A and 4B particularly. It is very likely that some of the compounds are phosphorylated during the course of the reactions. Krebs has shown that all the above reactions do occur in pigeon muscle and at sufficiently high rates to account for the rate of respiration. The chief criticisms of the Krebs cycle revolve around 2 points. 1, its general validity. 2, the role of citric acid. Smythe has shown that the above scheme will account for the respiration of pyruvate in pig heart muscle. For other tissues, direct evidence is not available. Evans has shown that in pigeon liver, pyruvic acid can be oxidized in the presence of malonate without the addition of 4-carbon decarboxylic acids. The probable explanation of this reaction is the origin of oxalactic acid from pyruvic acid and O2 by the Wood and Workman reaction. Krebs and Eggleston have shown that pyruvate is oxidized more rapidly in a bicarbonate, T2 medium, than in a salink phosphate medium. They proposed that oxalacetate was produced according to equation 5. Evans and Slotin, using the radioactive carbon isotope ECU, have shown that pigeon liver may synthesize a ketoglutarate from pyruvic acid and CO2. For the isolated, a ketoglutarate contained radioactive carbon. When the latter was initially present only as CO2, since a similar reaction did not occur with pigeon muscle, the radioactivity of the aketoglutarate could not be attributed to exchange reactions. Evans and Slotin consider it probable that CO2 is fixed by carboxylation of pyruvate dye. If, however, the aketoglutarate is formed via oxalacetate and pyruvate by way of symmetrical citrate, G of the radioactivity, should be in the carboxyl adjacent to the carbonyl group, and 2 in the distal carboxyl group. When aketoglutarate is degraded to succinic acid, it is the carboxyl group adjacent to the carbonyl which is lost as EO2, and Evans found that all the radioactivity came off in the CO2. The succinate had none. Thus, in pigeon liver, aketoglutarate is not formed by way of citrate. Wood and his colleagues have studied the same problem using the heavy carbon isotope ECs. In every detail, they have confirmed Evans and Slotin. They have proposed a mechanism of the decarboxylation cycle for pigeon liver, which is reproduced here as a diagram. This mechanism is provisional and may not be correct in all its details. It is interesting to note that Krebs and John Soik have shown that isocitric and cisaconitic acids are as rapidly oxidized by muscle as is citric acid. It is possible that citric acid is not on the main path of the cycle, but is produced by side reactions. Krebs and Johnson have pointed out that bacillus colli and yeast do not oxidize citric acid, 1, the common path. It has long been believed that the first steps of carbohydrate degradation were common to respiration and fermentation. In part, this is certainly true. The phosphorylation of starch or glycogen is a process common to both types of metabolism. In addition, such substances as glyceraldehyde phosphate, pyruvic acid, and in plants acetaldehyde are rapidly oxidized. The first 2 of these are intermediates in lactic acid fermentation, and all 3 in alcoholic fermentation. Glucose may be respired without previous degradation in fermentative reactions. For Warburg and Christian have shown that glucose 6-phosphate may be oxidized to phosphogluconic acid, and that the phosphogluconic acid can be oxidized with the absorption of 212 inols of O2 and the liberation of 2.7 moles of CiO presumably to a 3-carbon compound. Thus, a mechanism must exist for the degradation of glucose to 3-carbon compounds by stepwise oxidation. The view that glucose may be oxidized by a non-fermentative pathway was strengthened by the results of Lunsgaard with ionoacetic acid poisoned muscle and yeast, where the glycolysis or fermentation was poisoned, but respiration continued. Schure and Standard have shown that with low concentrations, 10 to 10 m of ionoacetic acid, glycolysis may be inhibited without a decreased rate of carbohydrate oxidation. Since glyceraldehyde dehydrogenase is poisoned by low concentrations of ionoacetic acid, the respiration in the presence of the poison may not proceed by the Meyerhof scheme shown on P423. The Pasteur effect. The cells of many organisms contain complete enzymes for the fermentative and oxidative degradation of sugars. However, Pasteur discovered that in yeasts the rate of fermentation was much lower in air than in nitrogen. Pfeffer and Pfluger believed the lower rate of fermentation in air was due to the oxidative removal of the end products. This explanation is inadequate because in many organisms the enzymes of fermentation may produce the end products far more rapidly than the E. Schure, Old Spring Harbor, Cyan P7, 323, 1939. Maximum rate of oxidation, in other words, the presence of air or a normal respiration, serves to suppress or completely inhibit the fermentative mechanism, and this is known as the Pasteur effect. The effect may be seen clearly when animal tissues are poisoned with ethyl carbylamine, for this poison does not inhibit the oxygen consumption or the fermentation, but does poison the Pasteur mechanism, so that in poisoned cells, both fermentation and respiration occur simultaneously at their maximum rates. That the Pasteur effect can be considered a regulatory mechanism may be seen when it is realized that the fermentation of 1 mole of glucose to lactic acid liberates 54 cal, while the oxidation of 1 mole of glucose to CO2 and H2O yields about 700 cals. Thus, far more energy is available from a given amount of sugar by respiration than from fermentation, and the suppression of fermentation when oxygen is available is an economy for the organism. The Pasteur effect is widespread, for it occurs in yeast, some bacteria, some higher plants, and invertebrates. Oxidative resynthesis. There have been several explanations of the mechanism of the Pasteur effect. The one proposed by E. Meyerhof is known as the oxidative resynthesis theory. Meyerhof initially proposed that lactic acid produced in nitrogen is removed aerobically partially by resynthesis to carbohydrate and partially by oxidation to CO2OO2O2. It is clear that sufficient energy is available by the oxidation of 1 molecule of lactic acid to resynthesize about 12 molecules of lactic acid to carbohydrate, provided some mechanism exists for coupling the energy liberated in oxidation to the resynthesis. Meyerhof found that between 4 and 5 molecules of lactic acid are synthesized for each molecule oxidized. Meyerhof has pointed out that it need not necessarily be lactic acid which is oxidized, since the oxidation of an equivalent amount of carbohydrate would be equally as consistent with the data, and the material resynthesized may be a precursor of lactic acid. There is a considerable amount of experimental data which supports the resynthesis theory, but perhaps none of it is absolutely critical. In brewers' and bakers' yeast, the rate of CO2 production is greater in nitrogen than in air, and resynthesis of the alcohol to carbohydrate could not explain the decreased CO2 production in air. Meyerhof proposed that in yeast, some 3-carbon intermediate is resynthesized, Blacko-Meyerhof, in a symposium on respiratory enzymes. Ng, of Wisconsin Press, 1942, proposed essentially the same scheme to explain the relation between fermentation and respiration. In apples, mots of lactic acid in and 2 mots of lactic acid in air. The quotient for many tissues lies between 1 and 2. The quotient is often expressed in terms of glucose, and then each molecule of glucose oxidized prevents the fermentation of 3 to 6 molecules of glucose. If respiration is inhibited by HCN, NANS or X fermentation occurs in air and often at very nearly the maximum rate. In peas, 1-M-equine does not inhibit respiration, but do SES inhibit the Pasteur effect so that aerobic fermentation occurs. Similarly, ethyl carbolemonate poisons the Pasteur effect in mammalian cancer. The Pasteur effect in yeast and in mammalian tissues is more sensitive to CO than is respiration. Lazarisecphoria is also found that lowered O2 tensions, which do not inhibit respiration, induce fermentation. The situation outlined above need not be universal, as Marsh and Goddard were unable to induce aerobic fermentation in carrot root tissue, which has a good Pasteur mechanism, with IECE2 tensions, or lowered A2 tensions. IDES, a respiratory inhibition of over 45%, occurred. Kempner has shown that in goose erythral das, the respiration may fall to I3 aerobic rate in 3.8 volume per C. Now, too, without increase in the rate of glycolysis. Similar results have been obtained with bone marrow by Warren. H. Pasteur enzyme, Lippmann, does not accept Meyerhoff's resynthesis theory. Lippmann believes that fermentation is inhibited by oxygen, but not through the respiratory mechanism. Stern, realizing that the relative affinity for CoA in competition with O2 of the Pasteur mechanism was greater than that of cytochrome oxidase, decided to demonstrate the existence of a Pasteur agent or enzyme. Working with rat retina, he has poisoned the Pasteur effect with Ko, and then determined the relative photochemical absorption spectrum by relieving the inhibition with various known wavelengths of light. Fly respiration of rat retina is not inhibited by Co, so he compared the spectrum he obtained with that which Mednick obtained on extracts of rat F. Lippmann biocurrent. 265, 133, 1933, and in a symposium on respiratory enzymes, Univ of Wisconsin Press, 1942. Heart cytochrome oxidase. These spectra are given in Table 39. It will be seen that the Pasteur enzyme differs from cytochrome oxidase in the position of two bands. Then evidence for TLI existence of the Pasteur enzyme is 1. It combines with ethyl carbolenone, ECN, and Co. 2. Its spectrum differs slightly from that of cytochrome oxidase. 3. Its relative affinity for CO in relation to O2 is greater than that of cytochrome oxidase. Stern believes the Pasteur enzyme is a phytohemon. Melnick, in Stern's laboratory, has similarly determined the photochemical absorption spectra of botulicoscytochrome oxidase and the Pasteur enzyme in baker's yeast. The spectra are identical at 3 of the bands, but there is an absorption at 560 MI in the oxidase, which is a little greater than in Pasteur enzyme. The evidence for the existence of a Pasteur enzyme is not too strong. If such an enzyme exists, the mechanism oxygen for the effect. These results of Stern and Melnick need not necessarily be inconsistent with Meyerhoff's resynthesis theory. 1. Assimilation. It is clear that the synthetic powers of living cells are very great, and this becomes strikingly illustrated when one realizes that many bacteria and fungi may grow rapidly on inorganic salt solutions to which a single organic carbon compound has been added, such as acetate, propionate, pyruvate, glucose, etc. In many of the synthetic reactions of growth, an increase in free energy occurs, and such reactions may only occur if they are coupled with an oxidative reaction furnishing the energy deficit. Thus, one carbon compound must then furnish the energy source, as well as the carbon for the synthesis. Oxidative assimilation may seem much more striking in plants than in animal cells, but animal cells also possess such synthetic ability. This may be illustrated by the conversion of carbohydrates to fats, for the potential energy of the fat is much higher than that of the starting material. The difference must be furnished by energy liberated in cellular oxidation of part of the carbohydrate. The efficiency of the assimilation may at times be high. Goddard found that the fungus Trichopaton degraded 0.625 milligrams of protein and 1 milligrams of glucose for each mg increase in dry weight over a period of 6 days. The Japanese workers have conducted many experiments on the ratio of the carbon source oxidized to amount assimilated for the fungus Aspergillus. These results have been summarized by Tamiya Barnelpi has shown that during the germination of barley seedlings, 3 of the stored glucose, as it had long been known that oxidative assimilation occurred in growing cells. But it had been generally assumed that such assimilation did not occur in non-proliferating cells. Marker first clearly showed that oxidative assimilation may occur in non-growing cells with a fixed ratio between the substrate oxidized to CO2 and H2O and that assimilated. Working with the colorless alga proteca, he showed that in short-period manometric experiments, the O2 consumption fell to the low endogenous rate when the total O2 uptake and CO2 liberation were only of the calculated value for the complete oxidation of acetate. And he recovered the rest of the carbon in the cells. He has been able to show that a large variety of substrates are assimilated in fixed ratios with their oxidation. In all cases, the assimilated material corresponded to HUO and is probably glycogen. The acetate experiment may be summarized as follows. Clifton and Logan have also found that Escherichia coli likewise assimilates fixed ratios of the various carbon compounds furnished it. In Uiapoisoneral cells, the coupling between oxidation and synthesis seems to be obligatory, and the ratio of material oxidized to that synthesized is constant over a wide range of concentrations. Clifton and Logan have been able to separate this coupling by poisoning the cells with 0.0025 m NANS or 0.005 nm dinitrophenol. The poisoned cells oxidize the substrate completely to C2 and H2O. Scheid and Thiemann have shown that the fungus Lephometis assimilated JGO of its acetate and oxidized Benoit and Elliott have shown that slices of rat liver synthesize carbohydrates from adenine pyruvate and lactates, while rat kidney cortex forms carbohydrates in addition from alanine, fumarate, succinate, and malate. Winsler and Bonneberger have followed the heat production by yeast in a microcalorimeter. They have found that aerobically, Wadian, the rate of heat production falls to the endogenous rate. The glucose has all disappeared from the medium, but only the expected heat has aerated of the glucose has been assimilated. Similar results were obtained with acetate, but here percent of the acetate has been assimilated. They have also shown that glucose may be assimilated under anaerobic conditions. They have calculated the efficiency of the synthesis on the assumption that the synthesized product is glycogen using the relation. Total free energy made available in breakdown for the aerobic assimilation of glucose, they find an efficiency of 2.88%, for anaerobic assimilation 4.62%, and for the aerobic assimilation of sodium acetate 12.2%. Van Niel has measured the anaerobic assimilation in yeast by the manometric technique and obtained results in excellent agreement with Winsler and Baumberger. This widespread oxidative assimilation must be at the very basis of the chemistry of growth, and the fundamental problem of how the energy coupling occurs has not been solved. However, 2 suggestive mechanisms may now be examined. Energetic coupling. The statement is sometimes made that the outlines of the problem of cellular respiration are clearly understood. This statement has a limited validity if one refers only to the mechanism of the oxidation of carbohydrates. The central problem of cellular respiration, the mechanism of using the energy liberated in oxidation for the work of the cell, is a virtual mystery. Cells use the energy in many ways in contraction, cyclosis, synthesis, secretion, ion accumulation, transmission of nerve impulses, etc. In no single case has an adequate explanation of the mechanism of the energetic coupling between the energy liberating reaction and the cellular machinery been given. A. Oxidation reduction coupling. It will be recalled that the equation for the potential of a reversible oxidation reduction system is, where A is the observed potential, R equals gas constant, T equals absolute temperature, F equals Faraday constant, and N equals number of electrons involved in the oxidation, Sek-Sik-Warnchap 4, reduced flavin enzyme, at a fixed pH, say 7, the equation simplifies to. For biological purposes, the value of at PLL of 7 should be used in comparing different systems. Since if the oxidation reduction system is an acid with a pKa between 1 and 8, the L may not be directly calculated by equation 1. The more involved calculation is given by Michael Isedudand. Unfortunately, the sign of the potentials used by the biochemists is opposite to the sign used by many physical chemists. In the biochemical terminology, a system positive to 1 below, it will oxidize the lower system. The free energy change, AF, may be readily calculated from a knowledge of the potential. A equals the change in potential in volts, where a decrease in potential is considered negative, and increase in potential positive. The free energy under standard conditions is given by we may illustrate the use of these equations as follows. This means that the oxidation of 1 mole of lactation to 1 mole of pyruvate ion in dilute aqueous solution at pH 7 and under such conditions that the potential does not change yields 8.304 cals. In practice, the potential would change unless the volume were infinitely large or the lactate were constantly replaced and the formed pyruvate removed, as would occur under steady-state conditions in living organisms. The reverse reaction, the conversion of pyruvate to lactate, will not occur spontaneously since AFE is positive. However, Borsuk and Scott were able to reverse this reaction with toluene-treated Bacillus coli using the energy furnished by the oxidation of formate. The bacteria contain enzymes which catalyze reactions 5 and 6. Without the bacteria, no reaction between formate and lactate would occur. In addition, a dye had to be added which would be oxidized by pyruvate and reduced by formate, and such dyes as Janus green or methylene violet, whose pounds equals 0.26 were satisfactory. The free energy changes are, thus, the energy liberated in the oxidation of the formate was coupled to the synthesis of lactate. How frequent this type of energy transfer is in the living cell is not known, but Green, Dewan, and Lawar have been able to link the reduction of several substrates to the oxidation of JS-hydroxybutyrate, using diphosphoride nucleotide as the carrier. It is improbable that all steps in the oxidation of glucose by oxygen are espially able to furnish energy for the work of the cell. The energy liberated do not know the potential of cytochrome oxidase. We may suppose it is intermediate between oxygen and cytochrome C. The reaction between oxygen and the oxidase is presumably irreversible, since if it were not, its reversal would be the decomposition of water and the liberation of free oxygen. If we assume that the reaction of the cytochrome oxidase and cytochrome C is either irreversible or not coupled, we can calculate Claude amount of energy degraded as heat as follows. This calculation is valid only at 1 atmos O2 pressure and with the ratio of ferrocito to ferrocito equals 1 under more physiological conditions, say 0.01 atmos of oxygen and a ratio of ferrocito to ferrocito of potentials are shifted as follows. Now the oxidation of a glucose molecule means the repetition of reaction 12 6 times or 138.9 cal are degraded as heat and not available for useful work in the cell. Undoubtedly, other irreversible processes intervene below the cytochrome level, and the complete oxidation of glucose to CO2. H2O6 transphosphorylation. It has long been recognized that the phosphorylation of sugars is an essential step in fermentation and glycolysis. More recently, it has been recognized that idiosphate transfer occurs between various compounds in cells, and that phosphate transfer may play a fundamental role in energetic coupling. The reader is referred to the stimulating articles of Kalkar, Egidi, and Lippmann. They have suggested that there are 2 groups of phosphate compounds. Those in which the phosphate is bound in an ester linkage to an alcohol or aldehyde, and in which the UF of hydrolysis is only 1 to 3 cal, and those in which phosphate is bound to N, or to another p-atom pyridiophate, or to a carboxyl group. We do not know the F of the hydrolysis of these bonds, but Lippmann and Kalkar estimated at about 10 to 11 cal. Lippmann classifies this latter group as compounds with energy-rich phosphate bonds. Idle bond energy is not known and need not equal the AF of hydrolysis. Both authors believe it probable that the energy liberal in oxidoreduations is stored up in phosphorylated compounds and transferred from compound to compound and may well become established. A word of caution here seems not out of place. The extremely stimulating ideas of Kalkar and Lippmann will serve their purpose if they stimulate further work, particularly to evaluate the free energy changes involved in these phosphorylations and dephosphorylations, and in tying these processes into specific loci of cellular metabolism. The recent results obtained on muscle energetics presented below illustrate specifically how transphosphorylation functions in energy transfer. When muscles contract, phosphocreatine is converted to creatine, and the phosphate is bound to adenosine diphosphate as follows. This reaction is reversible, and since the equilibrium is near 1, the free energy change must be small. No enzyme is present in muscle which will hydrolyze creatine phosphate to creatine plus H8PO4. But the adenosine triphosphate may return to diphosphate in either of two ways. Thus, a small amount of adenosine diphosphate may function catalytically in dephosphorylating creatine phosphate, according to Meyerhoff and Schultz. A 14 is about 11 count, and 14b procedural evolution of heat. In recovery of the muscle, under either anaerobic or aerobic conditions, the creatine phosphate is resynthesized. The mechanism of this R-synthesis has long been in doubt, since the reversal of 14A or 14B seems to be impossible from energetic considerations. However, the recent work of Negelai and Brommel, and of Warburg and Christian, has gone far toward answering this question. They have shown that adenosine triphosphate is synthesized from inorganic phosphate in a coupled reaction, in which phosphoglyceraldehyde is oxidized to phosphoglyceric acid. There is still some question as to the detailed steps of reaction 15A, see Meyerhoff, but not of the reaction itself. Thus, the energy furnished in the oxidation is used in the synthesis of adenosine triphosphate, and the latter can transfer its phosphate by the reversal of reaction. 13. The coenzyme may be reoxidized either aerobically or anaerobically. Cytochrome system, unknown carrier. There is reason to believe that phosphorylation is coupled to several other oxidations, particularly pyruvate to acetate and probably succinate to fumarate. That phosphorylation occurs vigorously during respiration of glucose and pyruvate seems clear from the experiments of Kolowich, Welch, Kalkar, and Korean on minced kidney cortex. In the conversion of adenosine diphosphate to triphosphate by reaction with creatine phosphate, there is a transfer of energy. The only way this energy can be liberated is by the loss of the phosphate from adenosine triphosphate. This reaction is catalyzed by the enzyme adenosine triphosphatase. If the hydrolysis were to occur in a random manner in the cell, we should expect the energy to be dissipated as heat. Now, Engelhardt has shown that myosin, the contractile protein of muscle, is itself the enzyme which brings about the hydrolysis of adenosine triphosphate. In this extremely interesting case, it appears from Engelhardt's results that the catalyst accepts the energy liberated by its substrate. Further, he has shown that when myosin threads are placed under tension, the addition of adenosine triphosphate results in a relaxation of the myosin and an increase in length of 1.5 to 2 times. Lai has found no other substance which will replace adenosine triphosphate in this reaction. It now appears that the first biochemical reaction has been discovered which dynamically converts the potential energy of a chemical compound of mechanical work. See further as one surveys our knowledge of cellular respiration. One is impressed are involved. The complexity of the metabolic engine, even in a tiny bacterial cell, is very great. The essential similarity in TLI mechanism from organism to organism is, however, striking and leads to a real unification of our knowledge. The stepwise degradation of the carbohydrate molecule with degradation of energy by means of a series of reversible oxidation reduction catalysts appears to be universal. The liberated energy is probably transferred and made available for cellular work in a series of small packages. The mechanism of energy transfer is far from settled, but two suggestive schemes have been proposed by oxidation reduction coupling and by energy transfer in transphosphorylation. Certain areas are only roughly sketched in. Certain regions are blank, and in places the picture's outline may need repainting, for mistakes may have occurred in the penciled framework, and fine details still have to be added. Even to the most finished parts of the work, there would appear to be plenty of opportunity for future research, along many lines before our knowledge will approach that definiteness of major outline, fine detail, and clear perspective that constitutes the completed picture of a scientific solution. Advances in Enzymiology, ed. B. by F. Nord and C. H. Workman. In Curset's Publishers, New York, Volume 1 appeared in 1941, 1 Voltaire. Published a year, Biological Symposia, Volume 5, Comparative Biochemistry, Intermediate Metabolism of Fats, Carbohydrate Metabolism, Biochemistry of Whole Line, ed. by B. Lewis, Jakes Kattler-Gebnis der Enzymforschung, ed. by F. Nord and R. Weidenhagen, Akademische Verlags, Gesellschaft, Leipzig, Volume 1 to Volume 8, 193-39. An extremely valuable series of articles on many aspects of enzymes and cell respiration. Evans, H.A. Ed. to Biology, All Action of the Vitamins, Univ of Chicago Press, 1940, Crow, August. The Comparative Physiology of Respiratory Functions. Univ of Pennsylvania, Oppenheimer, C. and K.G. Stern Biological Oxidation, Nordeman Publishing, Keogh Inc. Press. Chapter 8, Section 7. Contractility. The manuscript of this section was received for of all types of protoplasm. The skeletal muscles epitomize best the phenomenon of contractility, and it is the study of muscles which has in the past extended, and is most likely in the future to extend, our quantitative knowledge of this protoplasmic phenomenon. Nevertheless, it must not be supposed that muscles operate by any unique mechanism not represented in other contractile tissues. The fundamental mechanism of contraction is presumably the same in all tissues, but in muscles it is less obscured by other functions, such as heligestion, absorption, and excretion, and it is easier to measure the forces developed, easier to observe the physical changes which occur, and easier to determine the chemical nature and quantities of the reactants and end products of the chemical processes involved. From a qualitative point of view, however, the nature of contractility is very much illuminated by a preliminary survey of the phenomenon, as it appears elsewhere in living cells. From a practical point of view, it is ridiculous to propose contractility as one of the fundamental characteristics of all living cells, when no obvious contraction can be demonstrated in many of them, even by the most delicate means. Nevertheless, the type of physicochemical structure which is required for contractility is so similar in principle to the type of structure which is described for protoplasm in general, that the two subjects can hardly be discussed separately. Indeed, the best introduction to the study of contractility is to be found in recent discussions of the structure of protoplasm, and this subject is presented elsewhere in this volume. It is sufficient here to point out that the foundation of these present concepts is a network of protein chains held together by cross-linkages of hydrogen bonds or van der Waals forces, if not by stronger valences. This network may make either a sol or a gel, depending upon the degree of folding of the protein chains and the strength of the cross-linkages. Where the chains are all oriented together in a more orderly array, the structure becomes birefringent and may show visible fibrousy. Any stimulus which causes a folding of the protein chains or an increased aggregation will decrease the birefringence and cause a rearrangement of the material or change of shape, which may be described as a contraction. The only truly essential requirement for contraction, according to this point of view, is the partially unfolded protein chain. The presence of visible fibrillae and double refraction proves only increased degrees of differentiation of the contractile machinery. These appear to represent merely the means whereby more and more contractile units can be packed into a given space. Finally, it may be that the development of cross striations is the last step in the same direction. Berndal has argued that it is a manifestation of an alternately reversing a spiral arrangement of the fibrils resulting from the tightness with which they are packed together in the fiber. From the point of view of this general concept of contractility, the various types of contractile cells may now be examined. 1. Protoplasmic streaming. This survey may well begin with those phenomena which are least definitely related to contractility as we know it in muscles. Protoplasmic movement may be regarded as a common property of all cells, although it is not equally obvious in all. The Zorn show protoplasmic streaming as in the stamen hairs of Tradescant IA or the cycloses in Elodiodata. In the slime molds, Myxovisiphes, the whole protoplasmic mass is naked and unrestrained by a seal wall, and the streaming is accompanied by progression of the whole organism. In other Resicicide phenomena are very similar. The direction of streaming reverses at regular intervals. This becomes most evident when the movement is speeded rhythmical contraction, C-frizz, similar in its time relations to that of smooth muscles. This contraction has been recorded by an ingenious method by Kamiya, who opposed the movement by air pressure in a special apparatus and recorded thus the pressure developed by the contraction. It is not impossible, then, to attribute this phenomenon to the folding of long protein molecules forming the cytoskeleton, Peters, or fibrillar reticulum of the plasmodium. These molecules would also account for the elasticity for the phenomenon of protoplasmic streaming in Tradescantia, Kuhne, 1864, advanced a similar explanation. According to him, the protoplasm is to be regarded as creeping about inside the cell wall like a slime mold. It is apparently not always easy, however, to establish the change of shape. W. Klein, Hunters. Die über das Protoplasma und die Kontraktilität, Leipzig, 1864, which is necessary for this contraction theory. Marsland and Brown, for example, describe protoplasmic currents occurring in amebia when the animals are entirely stationary and are exhibiting no external change of shape. This observation does not perhaps entirely exclude the contractile theory, since the internal boundary between sol and gel layers may show rhythmical variations in position which are easily overlooked. Nevertheless, Marsland and Brown attributed this streaming to sol and gel changes largely because of the effects of high pressure in stopping the movement. Later, Marsland concluded that this theory was untenable. According to Rashevsky and Young, protoplasmic streaming could theoretically occur from energy derived from a continuously maintained diffusion gradient between two Y joints in the cell. If so, then this phenomenon is fundamentally different in nature from contractility. Such a theory, however, would not explain very well the rhythmic reversals which are observed in some cases, and it does not seem to be inherently very probable. This difficulty also applies to the electroendosmotic theory of streaming. In general, it may be said that the problem of protoplasmic streaming is harder to reconcile with contractility than is amoeboid movement. 2. Amoeboid movement. Possibly the movement of the plasmodia of the slime molds is not to be identified with the protoplasmic streaming of stationary cells. If so, it still remains conjurable to the movement of true amoeba 3, where most writers agree that it is the contractility of the superficial plasma gel which causes the movement. Lewis, Schaefer, Dellinger, Mast, Pantin. At the posterior end of the animal, the protein chains in the gel contract in response to some stimulus. This raises the internal pressure in the endoplasm or plasmasol, which bursts through the ectoplasm to lunge forward. At the tip of the pseudopod, the sole becomes a gel again and recovery takes place. Chemical differences between the anterior and posterior ends have been established by differential staining, Okada, Bantit. As in muscle, therefore, chemical changes of contraction may occur in the posterior end, which are reversed during the recovery in the anterior end. The change from gel to sol has been regarded as an exothermic process. Because protoplasm tends to solate or decrease in viscosity, Marsland with increased pressure, and gels, which do this increase in volume and absorb heat during gelation, F.E. Cunliffe, 1937. A corresponding absorption of heat must take place at the anterior end when the gel forms again, and the energy associated with this change of state must be comparable to the thermoelastic effect in muscle, CP 486. Anyhow, the energy for the amoeboid movement must come from some other energy-rich reactions, as it does in muscle. Still another view regarding the solid-to-gel change is possible, however. The solation resulting from an increase of pressure, which was observed by Brown and Marsland, may be due to some secondary effect, and the protoplasmic gel may form nearly isothermally like most thixotropic gels from the proteins in that case. It is simplest to consider that the change from gel to sol during contraction is merely the result of movement, which mechanically breaks up the gel, only to permit it to reform as soon as the movement ceases at the tip of the newly formed pseudopod. In either case, some stimulus is required in the posterior part to initiate contraction. There is no way to decide whether this merely releases some previously stretched spring, the shortening of which is the contraction, or whether the protein chains unfold again passively in relaxation and reestablish their side linkages to form a gel, the active liberation of energy being confined to the contraction phase. One of the most respected arguments against the contractility theory of amoeboid movement has been the observation that in the formation of a new pseudopod, granules are seen to move in the new pseudopod before they start to flow out of the old ones. It was argued, therefore, that old pseudopods collapsed because they were pumped out, and not because of inherent contractility. Movement, in other words, was initiated in the new pseudopod itself, which was not therefore pushed out passively by a force from behind. In reply to this, it may be pointed out that movement may become visible first in the pseudopod, because elsewhere the movement is a fluid only without granules, or because this is the narrowest place on the route with the velocity, if the contractility theory of amoeboid movement is correct. Then it is a primitive form of contractile structure with widely separated and rather disoriented protein chains, because there seems to be no sign of birefringence in either the mycetozoa or the rhizopods, Norris, Schmidt. In the pseudopods of the heliozoa and the radiolaria, however, birefringence is well established. These pseudopods are characterized by an axial skeleton surrounded by contractile protoplasm. It is not certain whether the birefringence is due to the axial skeleton or to the protoplasm surrounding it, or to both. When the pseudopod or axopod is withdrawn on stimulation, the birefringence disappears, there being no sign of it in the body of the animal, Schmidj. The fibrillar protoplasm, or myoids, in these axopods can shorten on stimulation to I5 or less of their relaxed length, and many forms are said to show cross-striations. Contraction can be initiated by induction shocks. They represent plier-4, a more highly developed contractile element than is found in the amoeba. 3 other contractile tissues. According to D.C. Linger, the cilium is a better example of contractile tissue than the amoeba. Both have a fibrillar structure, but it is better developed in the cilium, which shows birefringence. The cilium consists of a contractile fibril working against an elastic structure, or 2 antagonistic fibrillae working against one another with a pliable skeleton in between. The close parallelism between ciliary and muscular activity has been emphasized by J. Gray, to whom we owe some beautiful stroboscopic pictures of the beat of the cilium, and the most extensive study of the subject so far available. Flagelles are in general like cilia, but they function mechanically more like a screw propeller than like an oar. They also are birefringent, and Dellinger has shown that they can be separated into several fibrilla twisted together. The flagello are also birefringent, as are the tails of another striking example of a WTDL-developed non-muscular contractile. Myoid structure is found in the Vorticellida. In the lumen of the lilo stalk is a long contractile fibrillar birefringent strand, Schmidt, Biederman. On stimulation, this can shorten to 40 or 50% of its length and then slowly recover. This structure shows both form and intrinsic birefringence. On contraction, the birefringence diminishes as it does in skeletal muscle. According to Lapica, this myoid, when separated from the body of the animal, can still be stimulated electrically to contract, but in that condition, it no longer relaxes. This observation suggests that relaxation is the active phase of the process, and takes place slowly against the elasticity of the stalk and at the expense of energy mobilized in the body. The extremely rapid contraction is then to be regarded as the sudden release of a previously stretched spring. While this interpretation is reasonable enough, it is not thereby proved correct, and other observations will be mentioned later, which appear to support the theory that relaxation is passive. Movements of plants depend either upon differential growth rates in different parts of the stems or other structures, or upon local changes in turgidity or moisture contact. They must be regarded, therefore, as dependent upon a mechanism which is totally different from the contractility of animal tissues, bones, stern. In the muscles proper, the fibrillar protein reticulum, as found in the undifferentiated cell, reaches its highest degree of specialization for conotratility. Similarly, the nerve fiber represents the highest specialization of that same reticulum for the process of conduction. In smooth muscles, individual cells are still involved. In the still more highly developed striated muscles, many cells have fused together into a large multinucleate fiber, thus bringing the protein chains of many cells under the coordinated control of one end jadeate. Likewise, many fibers are brought under the control of aureaxin, which branches to reach an equal number of end plates. The multinucleate arrangement permits the development of longer fibril without interference from cell boundaries. 1. Macroscopic structure. A muscle consists of individual contractile units arranged in parallel or in series according to the amount of force and shortening required. The principles involved are illustrated by two types of muscles: the parallel fibered muscles, long and short, and the diagonal fibered or peniform muscles, see Pig 44. For greater shortening, more units are arranged in series, or the fibers are long, as in the sartorius muscle. For greater force with minimum shortening, many shorter fibers are arranged in parallel. WLXC, a broad, short muscle, is anatomically possible, as in the gluteus or rectus abdominis muscle. The latter arrangement is suitable, but in other localities, as in the calf of the leg, the contractile units pull somewhat diagonally on a central tendon like men pulling on a rope. This provides large force and small shortening in a muscle of approximately the same convenient shape as the sartorius. The length of the fibers bears a fairly close relation to the amount of shortening demanded of the muscle. It seems likely that this relation is automatically adjusted by the mechanical conditions of the situation, so that muscle can be transformed into tendon or vice versa until the fiber, when stimulated in the body, can shorten to something less than half its resting length alanes. According to the figures of Zechariah, the absolute shortening of any particular muscle in the body is equal to the absolute shortening of any one of its fibers. In the parallel fibered muscle, like the sartorius, the maximum shortening of the whole muscle and of its fibers, when tendinized in situ with insertion cut and a very small load, is 55%, and the fibers are nearly as long as the whole muscle. In the rectus femoris muscle, the fibers are only 14 or 3/5 as long as the whole muscle, and the muscle shortens only 28% of its length. If a sartorius muscle is cut in half, each half will shorten half as much as the whole muscle. In the piemiform muscles, however, the absolute shortening of each half, or of each muscle bundle, is the same as that of the whole muscle, shakayada. In general, a muscle will not shorten beyond the length permitted by the anatomical situation in the body. Even if its tendon is cut, further shortening is evidently prevented by the connective tissue. Further information on structure and the arrangement of muscle fibers in different muscles may be found in papers by Baratov, Janssen, and Linhart 32. The striking appearance of the cross-striated muscles with their alternating light, isotropic, and dark anisotropic A-bands leads to an excessive preoccupation with the details of that particular type of structure. The cross-nations, however, seem to be peculiar only to the rapidly shortening fibrils, and the more rapid the muscle, the shorter the interval between the bands. Likewise, the faster the muscle, the more nearly equal are the anisotropic and the isotropic discs. Brennerdam. From this point of view, it is tempting to regard the smooth muscles as those in which the tempo became so slow and the sarcomeres so long that the cross striations disappeared. Such a classification, however, is purely artificial because their immediate forms are not known, and it offers no explanation of the origin of the cross striations from the presumably more H. Jasper and A. Pezard comprehend Ekadi. St. 198, 499, 1934. Primitive plain muscles. It does emphasize, however, the important correlation between speed of contraction and the presence of the cross striations. According to Bosler, the classification of muscles as smooth or striated is not a particularly happy or significant one. ECT for the muscles of vertebrates, where smooth muscles are vertebrates, where smooth muscles are, in general, specialized for toe, and striated muscles for quick movements. In a survey of all the muscles of the animal kingdom as a whole, the classification breaks down because it fails to maintain its correlation with function. The muscle cell, according to Bosler, is composed of fibrilla embedded in sarcoplasm. The fibrils are of two kinds. One, the large, darkly staining tetanus fibrils arranged around the periphery of the cell and attached to the sarcolemma either continuously or at intervals, Z lines. 2. The small, lightly staining tonus fibrils either distributed throughout the cell or arranged inside the peripheral layer of tetanus fibrils. A cell which contains both types of fibrillae will serve the function of both tetanus and tonus. The more rapid tetanus fibrillae are striated and are attached only at intervals to the sarcolemma. Where the attachment is continuous, the resistance to shortening is greater and movement is slower. The difference in function, however, between tetanus and tonus fibrils is far greater than that between striated and unstriated. In any event, the essential features of a contractile mechanism must be and are present in both smooth and striated muscles. These essentials are the birefringent fibrils and the myosin chains. It has frequently been questioned whether the striated muscle fibers are composed of a series of discs, or whether why it appears to be disc is really a series of layers arranged around a central axis. Like a spiral staircase, one frequently sees boundary lines between the sarcomeres, the Z lines, which appear to branch, or one sees that on one side of a given length line of fiber there are bands, and on the other side, W+1 bands. This is known as a Nanius period or Vernier type of banding and is sometimes interpreted as a section through the central axis of a spiral structure. It has even been supposed that the excitation wave in passing down the muscle has to pass along this spiral pathway. Some of the pictures of these spiral structures are rather convincing, especially in insect muscles. On the other hand, it may be argued that the same appearance might be produced in other ways, as by a true branching of the Z line. A certain measure of skepticism concerning this alleged spiral structure is therefore well justified. On microdissection, the sarcolemma is found to be a tough membrane surrounding the whole fiber. It is composed of interlacing collagenous birefringent fibers embedded in a thick colloidal matrix, which has been likened to a knitted stocking. The fibers of the sarcolemma are continuous with the fibers of the internal paramecium in places. The sarcolemma stretches reversibly over a considerable range, and its stretch is believed to be due to the displacement of the fibrils or to a change in shape of the meshwork, rather than to true elasticity of its components. The muscle substance itself is a sticky elastic gel with fibrils embedded in it. The fibrils can be pulled out with some success by micro-manipulation. The fibers of the Z-line Y form the boundaries of the sarcomeres, are attached to the sarcolemma and differ in composition from the myofibrils in most muscles, particularly insect muscles. These Z-lines are clearly distinguished, and they serve to keep the myofibrils aligned with one another during shortening, so that the cross striations of the fiber as a whole remain undisturbed. In the middle of the sarcomere, between the Z-lines, is the anisotropic or A or Q disc, which usually appears darker than the isotropic disc adjoining the Z-lines. The A discs are definitely anisotropic or birefringent and are often regarded, on rather meager EV idents, as the contractile part of the muscle. This belief depends chiefly on the fact that in contraction the Q discs shorten while the discs lengthen slightly. The contrary finding of franking has been attributed to artifacts due to fixation of the muscle. but Studnitz has found that the I disc does eventually shorten if the shortening of the A disc exceeds 50%. In the arthropod muscles with which he worked, Astacus fluvialia, a shortening of the whole fiber to 30% of its resting length may occur. It seems possible, therefore, that there is some power of contractility in the D discs, and this is confirmed by Schmidt WI disc is slightly birefringent. This is consistent with the idea that the myofibrils pass continuously from one Z-line to the next. Indeed, the diagrams of the fine structure of muscles offered regular orientation of the myosin chains, which make up the fibrils in the isotropic regions. Without some such continuity, it is difficult to see how the force of contraction of the A discs could be applied to the shortening of the fiber as a whole. It may be concluded, therefore, that the A discs are possibly stronger than the discs, but that both are contractile. According to Buchtel, Knappes, and Lindhard, the A discs are more easily stretched in the resting muscle than the D discs, while the reverse is true in the stimulated muscle. We have as yet no real understanding of the significance of the cross-triadic NS, except that they seem to be important for rapid contractions. One theory is that the discs are specialized for the process of recovery, while the A discs are predominantly contraction sites. The only experimental confirmation for such a theory comes from histochemical analyses. In the resting fiber, glycogen is found predominantly in the A discs, chiefly between the fibrils and sarcoplasm. During recovery, especially when the total glycogen is small in amount, there is more in the D discs, where it is supposedly being rebuilt from lactic acid diffusing out of the A discs. It is perhaps difficult to know just how literally to interpret these measurements of the dimensions of the A and J discs and the separation of their functions. According to Jordan, there is in contraction an actual reversal of striations in wasp muscle, the I discs becoming dark, and vice versa. This could be accomplished by a movement of granules from one disc to another. Many writers have preferred to regard the striations as dynamic rather than static structures. Carey, for example, regards them as standing waves of some sort. More generalizable is the intriguing theory of Bernal that they owe their origin to the reverse spiral arrangement of the parallel packed protein chains. According to him, substances with long molecules tend to show this structure when they arc aloud to set in long loops. The more highly birefringent bands then represent places where the spirals reverse, so that the nilocules lie parallel to the long axis of the fiber. At intermediate regions, the molecules are diagonal, some to the right and some to the left. It must be admitted, however, that the fibrillar structure of microscoance dimensions, which has been described for muscle, always appears to be parallel to the longitudinal axis, rather than diagonal. On the whole, this theory seems to be based upon very little evidence. One thing Willich seems certain is that the protein chains or the contractile units must extend from end to end of the fiber. And this being so, there is grave danger of placing too much emphasis upon the A discs as the contractile units. There is considerable evidence to show that during embryonic development, the fibers become capable of contraction and show double refraction before they become cross-striated. Atish has studied muscles in the dark field. Using light directed either parallel or perpendicular to the long axis, parallel illumination brings out the cross striations, while perpendicular illumination extinguishes the cross striations, but brings out the fibrillar structure. Some fibers are found to have lost their cross striations, but they still show uniform double refraction throughout their length. Similarly, Liang has found that muscles put into rigor with caffeine, quinine, or chloroform lose their cross striations but retain their fibrillae, which become uniformly birefringent. Conversely, treatment with ammonia destroys the fibrillar appearance, but preserves cross striation. The conclusion seems to be that the longitudinal and cross striations are due to different structure. The cross striations are believed to be due to periodic accumulation of granular material outside the fibrillar and the sarcoplasm. The true contractile unit is the birefringent fibril, which is continuous and uniformly birefringent from one sarcomere to the next. From this point of view, smooth and striated muscles differ only in secondary details, the fundamental structure being the same in both. This view confirms, therefore, the theory of Basler-Jay already discussed. It also may be said to support in some measure the ideas of Carey, who regards the sarcomere as a morphological myth, because according to him, the number of sarcomeres in a given muscle can be increased from 97 to 1124 by 10 seconds treatment of the fiber at 45 steps. It has, apparently, never been possible for anyone else to confirm this finding as yet, Spaetel and Jordan. With this exception, all the evidence indicates that the sarcomere is a fixed structure. Not so long ago, particularly under the leadership of Batazzi, it was fashionable to consider that the sarcoplasm was contractile, as well as the myofibrils. The situation in muscle was considered analogous to that described by Engelmannicke in Stentody, where he saw longitudinal, somewhat striated fibrils which were responsible for the quick shortenings of the body. The animal was also capable, however, of slow turnings of the body which left these fibrils folded and slack. These slower movements were attributed to some microscopic fibrils in the protoplasm. From this point of view, a muscle might be described as a striated muscle embedded in a smooth muscle. And this idea was advanced in explanation of all the slow movements which muscles can make under proper conditions. Biermann has also championed the idea of contraction without visible fibrillage and presented the evidence at some length. It seems unnecessary to suppose that all the myosin chains are packed together into bundles or fibrils. Some of them may well be scattered around more or less by themselves in the viscous sarcoplasm, where shortening would be more difficult and hence slower. The fibrillar arrangement of such chains would facilitate rapid contractions, just as the movement of large numbers of persons is facilitated by some military order in their ranks, thus preventing one individual from interfering with CC. Spaetel and Tukiranana ILAC 82 470 1942. The movements of his neighbor, most investigators at the present time, discount this assumption of sarcoplasmic contractility, for example, as unnecessary, but in our desire to make only the simplest necessary assumptions, we may sometimes forget that it is not difficult for nature to do things in ways which seem unduly complicated to physiologists. In many ways, this would seem to be a very natural arrangement, even though a complex one. Among others, Basler would discard completely the idea of contractile sarcoplasm, and he explains many of the phenomena of slow and fast contractions occurring in the same muscle, tetanus fibrils in the same fiber. The former are smaller and less darkly staining, and are arranged usually in the interior of the fibers and contract slowly, while the latter are larger, contract more rapidly, and are arranged around the periphery of the fibers. In spite of the validity of this idea, it is perhaps unnecessary to discard altogether the theory of sarcoplasmatic contraction, although the direct evidence for it is very scanty. The best evidence available comes from the work of de Reny and Hodgon, the behavior of cardiac muscle fibers in tissue cultures. Some of the cells contained visible fibrillus, and some did not, but both kinds contracted equally well. Moreover, some cells were figured which contained fibrillar on one side, but not on the other. The non-fibrillar parts of such cells were found to be more quickly injured by toxic substances, such as water. In such a case, the cell contracted on the fibrillar side only, and became concave on that side, whereas previous to this injury, the contraction was symmetrical on both sides. The authors conclude that both the sarcoplasm and the myofibrille are forms of contractile substance, which, in the embryonic state and under normal conditions, act synchronously. 3 visible diffraction spectra. It has been known since 1874, Ranvier, that a muscle, when traversed at right angles to its long axis on a screen placed beyond the muscle, ascribes a spectra of orders I secundicere, etc., extending indefinitely in the longitudinal axis of the muscle. From the positions of the series of spectra which are so produced, it is possible to calculate the grading constant, or the distance between the bands. Recent measurements, Sandal, Buchthal, and Knapez, give values which agree closely with the distance between the anisotropic bands of the same muscles. measured microscopically. When the bands move farther apart, as when the muscle is stretched, the first-order spectrum moves closer to the direct image. The percentage increase in the calculated width of the bands is the same as the percentage increase in the length of the muscle, at least after the initial stages, when the slack in the muscle was being taken up. Sandation. When the muscle shortens in contraction, the bands also become closer together. At the same time, the intensity of the spectra of the first, second, and third orders increases. This is interpreted as indicating a decrease in the width of the dark bands and a relative increase in the width of the more transparent D bands, Lichtful and Snappies. Some indication has also been found, one a phalar diffraction pattern at right angles to the main one, and caused by longitudinal surfaces in the fiber. Since these W shear obtained in single isolated fibers, they were attributed to bundles of fibrils or Cohnheim bundles. Pleasy diffraction patterns are surprisingly sharp, even when taken from a W muscle, and this LSS possibly some alignment of the anisotropic bands of one muscle fiber with those of the neighboring ones, so that the whole muscle is obliged to act like a single grating. Of special interest is the finding of Nicolai, who photographed the positions of the first-order spectrum during a tetanus of the muscle and reported fluctuations in its position, and therefore fluctuations in the width of the individual sarcomeres which were coincident with the frequency of the individual shocks in the stimulating current, thus even at frequencies of 60 per second. When the tetanus appears externally to be complete, it is possible to show that internally it is really still incomplete. Elhe's finding is of importance in the interpretation of some of the data on heat production during a tetanus. The diffraction spectra so far considered are those due to the microscopically visible cross striations or the first-order grading. Evidence of visible diffraction spectra from a still finer or second-order grading has been presented by Langellon, who has J. Repar. A model of the finer structure of the anisotropic discs based on dark field observations with the Shearer lens. The model consists of two systems of fine wires at an angle of 60 degrees with each other. These wires themselves form a third-order grating, but when viewed at different angles, the overlage of successive layers produces the appearance of a coarser or second-order grating, the diffraction spectra of which are just visible. All the structure observed in this way is confined, according to Langellon, to the anisotropic disc, the isotropic disc being congelately dark in the dark field of the microscopic. Other evidences of the submicroscopic structure of muscles are, however, more convincing and must now be considered for submicroscopic structure. However important it may be to know the microscopic structure of muscle, none of the histological details offers any very reliable clue as to the nature of the contraction process. The wealth of detail, which can be seen in some muscle fibers, especially those of the insects, has sometimes proved too tempting, and some writers have been over-venturesome in applying their imagination to the visual images which they gain from the microscope. The newer knowledge of the subject ascribes the process of contraction rather to the submicroscopic structure, and the unfolding of this field has provided an effective restraint for the previously unbridled imagination. An excellent recent review of this subject has been given by Fischer or Birefringent. Two tethods have been in use for the elucidation of the molecular structure, the measurement of birefringence, and the X-ray diffraction pattern. From the study of the birefringence of muscle, support has been obtained for the idea, originally suggested by Bricke, that within the iniofibrils pli-J routine chains are not uniformly distributed, but are arranged in micelles, or small bundles. The existence of double refraction requires some orderly arrangement of the molecules like that found in a crystal. It can also result from an orderly arrangement of larger units or giant molecules, such as those found in a solution of myosin. It is believed, therefore, that the birefringence, dr of muscle, is due to 1. The orientation of molecules within mixtles, intrinsic dr, and 2. To the orientation, micelles, in the long axis of the myofibrils. Form dr. The form dr disappears if the refractive index of the micelle is the same as that of the surrounding solution, and increases in proportion to the difference between the refractive indices of these two phases. It depends also to some extent on the relative volumes of micelles and solution. In order to measure the form double refraction, it is necessary to vary the refractive index of the solution around the micelles without modifying the volume of the micelles or the arrangement of the molecules within them. Strange as it may seem, the results appear to indicate that this is possible. The muscle is fixed in formaldehyde and is then immersed in solutions of varying refractive index. The fixation process itself has little or no effect on the double refraction, i.e., in spite of the use of different substances for the variation of the refractive index of the solution. The double refraction of the muscle falls along a smooth curve in the shape of a parabola, indicating that the refractive index of the solution is the only variable. This is shown in Figure 45, where Fisher's curve for smooth muscle and Weber's curves for striated muscle and myosin threads are represented. The curves are all very similar and pass through a minimum at the point where the refractive index of the solution equals that of the micelles. The dr remaining at this minimum is the intrinsic dr due to the molecular orientation inside the micelles, while the increase on either side of the minimum is due to form dr. This experiment must, therefore, be regarded as strong evidence that there are micelles in muscle. The fact that artificial myosin threads give similar results is evidence that the micelles in muscle are also composed of myosin. The volume of the micelle has been estimated by Weber from its molecular weight as determined in the ultracentrifuge as 1100 prom. The Wiener theory for form double refraction, the fraction of the muscle volume occupied by micelles, can be calculated. One can guess, further, that the rods are long compared to their diameter because they show double refraction of flow, and only rod-shaped particles would be oriented by the shearing stresses set up in the solution during flow. Since, however, the rods are anisotropic themselves, they must contain enough polypeptide chains in parallel to show this intrinsic double refraction. Thus, it is possible to estimate that the micelles may be 5 inches in diameter and 55 mm long. Wongber calculates that a micelle of this size would contain about 20 myosin chains. The distance of one micelle from its neighbor being 11.5 m-lieutenant, whether the micelles are arranged end-to-end or with a space between, is only a matter of guesswork. The most uncertain part of this whole estimate of the size of the micelle seems to lie in the figure for the volume. It is based upon a molecular weight of myosin of 100, which is a very large value and one which is likely to vary widely with changes of pH and electrolyte environment. It is known, for example, that urea lowers the molecular weight to 100. How constant in size the micelles are is unknown, but since the calculated size is based upon the only figure available for the molecular weight of myosin, the micelles might equally well be termed giant myosin molecules. Decrease in an isotonic contraction due to a diminished orientation of the myosin chains in the micelles act likewise. In an isometric contraction, there is usually a decrease, although at large extensions there may be no change. Or even an increase. Such an increase may be attributed to the effect of the increased tension per se the photoelastic effect, rather than to a better orientation of micelles. In smooth muscle, there is an increase of birefringence in stretching, but no change in frog-striated. Our muscle, the effect of stretch on a whole muscle is certainly a complex process. The rate metabolism increases, for example, and an increase in lactic acid, which may also occur, leads to a decrease of birefringence. In addition, the A bands stretch more than the B bands, and the effect of this is difficult to estimate. Correction is, of course, made for any decrease of the thickness of the muscle due to stretch. Stretching per se should cause a better orientation of micelles and myosin chains, and hence an increase in double refraction. From the birefringence studies, therefore, we learn that there are micelles in the muscle, and furthermore, we learn that they may lose, to some extent, their regularity or the completeness of their orientation during contraction. In addition, the micelles themselves may lose some of their intrinsic birefringence. The behavior of the micelles as a whole seems to leave us still unsatisfied in our search for the fundamental nature of contraction. B. X-ray diffraction. Further information must be sought in the change of the ultrastructure of the micelles. Evidently, the molecules of which they are composed are less regularly arranged when the micelle shortens. Further information concerning this change is derived from the diffraction patterns produced by X-rays. Here, the wavelengths used are so much shorter than they are in visible light that the dimensions of the grating which produces the pattern are of molecular rather than microscopic size. The molecular diffraction patterns tell us more concerning the probable nature of contraction than do the A-band diffraction patterns previously considered. When a muscle is dried, it is perhaps surprising to find that the X-ray diffraction pattern does not change. Evidently, the removal of the water has not changed the intermolecular distances. The water must therefore have been contained between micelles, and this may be regarded as an additional confirmation of the micellar structure. Furthermore, the side chain spacings of the protein molecules, as given by X-ray analysis, are so small that they would be inconsistent with the high water content found in muscles if this same close packing were continuous throughout the muscle substance. The micellar theory reconciles these two observations. Many investigators have presented data on the ultrastructure of muscle as it is shown by X-rays. It is, however, to the extensive papers of Asbury on this subject that we owe the most at the present time. Since his work will be discussed later in this chapter in connection with various other manifestations of contraction, it will be sufficient here to give only a very brief outline. The principles of the X-ray method have already been when a beam of X-rays is passed through a muscle perpendicular to its long axis. A diffraction pattern is formed on the screen, which shows a strike in resemblance to that caused by other fibrous proteins, such as the keratin of hair. In this particular form, it is known as a keratin and is composed of long polypeptide chains in a partially folded form. E5 water at ordinary temperatures, the keratin can be stretched out to about twice its original length, until the chains are unfolded and the pattern changes to the form indicative of 3 keratin. In this form, it is similar in structure to fibroin, the protein of natural silk. With further treatment in water at elevated temperatures in steam or in caustic soda at ordinary temperatures, the hair may be made to shorten to about 70% of its original length. This treatment apparently removes certain of the side chain linkages between adjacent polypeptide chains, and it is the nature and firmness of these linkages and the length of the side chains which determine the mechanical properties of the muscle or hair. With these linkages and hair broken down, the protein chains are able to fold still further, and eventually the hair passes into a supracontracted state. Thus, both the hair and the muscle show both contractility or ability to shorten and extensibility. It is apparently not a simple matter to stretch a muscle into the T3 keratin form, but in Asbury's latest paper, it is stated that this can be done and that the details of this procedure will be presented in a later paper. It has likewise proved impossible hitherto to cause a hair to shorten like a muscle to 30% of its initial length. If, however, Asbury is right in proposing that a globular protein is simply a fibrous protein folded up into globules, then still further folding of the myosin chains might be expected, continuing at last under proper conditions until they were reduced to the shape of a sphere. A sartorius muscle shortened until spherical would have a length not far from 20 to 25% of its initial length. The sphere length of a single fiber of the sartorius would be a still smaller percentage of the initial value. To realize a shortening of this magnitude in hair would presumably require further breakdown of side linkages. The nature of these side linkages has been discussed. See also Huggins. Speakman showed in keratin that the development of the supracontracted state depended upon the breaking of the cysteine linkages. This suggests, therefore, that hair is a sort of vulcanized fibrous protein or a vulcanized form of myosin, which thus has its extensibility much reduced. In conformity with this idea, hair has a far higher sulfur content than myosin. There seems to be little doubt that this picture of the fundamental machinery of contractility is correct, so far as it goes, but it leaves much of the story untold. When one tries to explain what forces actually cause the folding, how these forces are released by stimulation, and how the changes in myosin fit into the simultaneous transfers of energy, the uncertainties of our present position become increasingly clear. The chief disadvantage inherent in the X-ray diffraction method is the prolonged exposure which is necessary to obtain a readable photographic image. Even with the latest improvements described by Spyataliadov, Henny, and Ashkenaz, an exposure of 3 to 6 minutes is needed. With this technique, however, it was possible to show a change of patterns on contraction indicating a disorientation of the myosin chains. A similar change is caused by shortening in rigor mortis or contractures due to chloroform or caffeine. 5. The physicochemical structure of the muscle, myosin, in a striated muscle, 18 pounds 0% of the weight is protein. And about 60% of this is the globulin myosin of these proteins. The myosin is peculiar in that it shows double refraction of flow, as was shown by von Rudolf and Edsel, which become oriented muscle and has made myosin threads by forcing the solution out of a capillary tube into distilled water. These threads can be dried until they contain the same amount of water as skeletal muscle. Such threads show double refraction like the original muscle, and when stretched, they develop as much tension per unit of cross-section area as an intact resting muscle. Asbury and Dickinson have also made artificial muscles by drawing thin films of myosin on glass. Such myosin films give X-ray patterns similar to those of intact muscle, and when stretched, they change from the A to the pattern like other fibrous proteins and like muscle. It is generally considered, therefore, that the myosin represents the contractile machinery proper and is responsible for most of the double refraction of the intact fiber. Some double refraction remains even after all possible. Myosin has been removed by KCl. When a muscle is dried, it shrinks in the lateral dimensions, but not in length. The water appears to find space for itself largely between longitudinal fibers or between the myosin polypeptide chains. These chains have side chains which are in general the highly solvated tip. This provides a mobile protein grid rather than one of more rigid, rough ERDs, and gives a watery medium suitable for the maintenance of a high metabolic rate. When water is removed from between the fibers, the myosin molecules unite with one another rather than with the water, and the mobility is lost. The myosin is more soluble in potassium salts than in an equal concentration of sodium salts. Myosin solutions tend to lose, with time, their birefringence of flow, this being taken as a sign of denaturation of the protein. As judged by this test, the solutions are much more stable in potassium solutions than they are in NiQ or NaCl. There seems to be, therefore, for the muscle cell, at least, some reason for the high concentration of potassium, which is found in the interior. In spite of this high concentration of potassium inside the muscle fiber, it appears that the myosin under natural conditions inside the fiber is readily changed from sol to gel. Other factors contributing to its instability are the low GIVOL and its own high concentration. Batesmith has studied the solubility of myosin and states that at pH 6.5, it will begin to dissolve if the KCl concentration is 0.25 m. While at pH 7, a concentration of 0.7 m will suffice. HDPA, assuming a pH inside the fiber of 6.9, it is evident that little or no myosin would be in solution. Since the potassium concentration inside the fibers is at most 0.17 ml furthermore. If all of the myosin were uniformly distributed inside the fibers, its concentration would be 8% or even higher. If the myosin is confined to the fibrils, and Mirsky has shown that at high concentrations of the order of 10%, myosin tends to become denatured. Inside the fibers, therefore, the myosin may be regarded as on the verge of denaturation or gelation, relatively little being in true solution. Proteins of cells are of both the globular and the fibrous types. The former offer advantages of greater mobility, as in the plasma proteins, while the latter serve better for structural purposes. Myosin is the particular fibrous protein responsible for the structure and contractility of the muscle cell. There is evidence that fibrous proteins play a role either for contractility or conduction or in less highly differentiated cells. Merely structure, but little is known at present of their nature. Recently, however, Banga and Sen-Oryorji have isolated a fibrous or structure protein from kidney, which they have called renosin. This protein shows double refraction of flow and forms viscous solutions and thixotropic gels. All these prop cruda 5s indicate that the molecules are long and closely tangled with their neighbors, or connected to them by side chains or hydrogen bonds. Renosin resembles myosin, therefore, in many respects. A protein similar to renosin was found in liver, brain, lung, and other tissues. It is extracted after extracting with water. The globular proteins are removed, but the microscopic appearance of the tissue has not been seriously altered because the fibrous proteins remained. A subsequent extraction with a urea salt solution removes the renosin, but still leaves the microscopic structure largely intact. The remainder of the structural protein is removed by Urcanal solution. Renosin differs from myosin in showing negative rather than positive double refraction of flow. It also contains non-extractable phosphorus and is a nucleoprotein, although not derived exclusively from the nucleus. Likewise, Mirsky has extracted a protein from sea urchin eggs, which closely resembled myosin, and Benzel Chui has given the name plastinosin to a structural nucleose protein extracted from liver by 10% NACL, which has somewhat similar functions and properties. It may be mentioned also that myosin can be extracted from smooth muscles, as well as from striated muscles von Merle and Edsel-Bond. In any event, it is evident that myosin or myosin-like proteins are of rather widespread occurrence in cells. Soon after the death of an animal, the myosin of the muscles becomes insoluble or denatured, so that less of it is extracted by likewise contraction and fatigue of the muscle are related fundamentally to a regulation of the protein. According to Mirsky, the type of coagulation that occurs in rigor mortis is similar to that caused by dehydration, and it differs from other types of denaturation, such as those caused by heat, acid, and alkali, in that it is not accompanied by any increase in the number of detectable H groups. For some reason, the myosin behaves differently in this respect when it is in situ in thigh muscle than after it has been extracted. There is considerable evidence, therefore, that the act of contraction is intimately concerned with changes in the condition of the myosin. Just what these changes are and how they are brought about is still very uncertain, but a suggestive clue is furnished by the recent indication that the myosin itself is concerned somehow with the phosphate changes in muscle I hanga and A. Sent, Georgia and Zamologia 9, 111, 1942. This might be, therefore, the method by which the physicochemical changes of myosin are coupled with the energy-yielding chemical reactions, which are known to occur during contraction. When Dutikman noted that there was a decrease in the solubility of muscle protein during rigor, he observed also that there was a simultaneous decrease in the power of the muscle to esterify hexose with phosphate. More recently, it has been shown by Engelhardt and Ljubimova and confirmed by St. Georgi and Banga that the myosin fraction of muscle is able to split phosphate from adenosine-triphosphate. If the effect is not due to the myosin itself, it is due to some other substance or enzyme with which it is in intimate association. That myosin itself is the active agent is suggested by the experiments of Needham, and who reported a decrease in the birefringence of flow of myosin solutions after the addition of adenosine-triphosphate. These results suggest that the phosphate may be split off from the adenosine-triphosphate because of its tendency to combine with the myosin. According to the theory of contraction proposed by Kalkar, stimulation involves the removal of phosphate from combination with side chains of the myosin, thus permitting a further folding of the polypeptide chains. This may be the method by which chemical potential energy is transformed into mechanical energy in the myosin. Other points concerning the physicochemical structure of the muscle fiber might be mentioned. although there is no evident reason for connecting them specifically with the contractile mechanism. The interior of the cell contains what is practically an isotonic solution of potassium, most of which at least must be free enough so that it can exert an osmotic pressure. If this solution were brought into contact with the outside of the muscle fiber, the result would be the immediate death of the fiber on the inside. However, the potassium appears to serve a useful purpose in keeping the myosin in solution. On stimulation, the fiber loses some fraction of its potassium in exchange for sodium, while the reverse occurs in recovery. It is not known whether this is related to a change in state of the myosin, which then combines with less of the potassium, or whether the metabolism of organic phosphate fractions is involved. There is some reason to believe that both processes are concerned. In the bloodstream, potassium and carbohydrate frequently rise or fall together, suggesting that carbohydrate metabolism is somehow connected with the movements of potassium. The adrenal cortex seems to be concerned with both potassium and carbohydrate metabolism. In yeast cells, Pulver and Verzar have shown that the AV St. Georgii Banga Science 93 158 1941 R. Pulver and F. for Helvichem Acta 23 1087 1940. Also Nature 145 823 addition of sugar to starved yeast, suspended in a dilute solution of hydrotashuin, causes a sudden intake of large amounts of potassium. Senchigitergi has Marsh all tool the evidence in favor of the view that muscle contraction is caused by the slift of water resulting from the sudden liberation of ionized potassium from myosin at the moment of stimulation, potassium, as well as adenosine does apparently cause a decrease in the double refraction of flow of myosin. But what J-arc prevents this from occurring before the stimulation is not clear. In any event, it seems not unlikely that the secret of the contraction of muscles may lie in the reactions between myosin, potassium, and adenosine nitrophosphate. 6. Mechanics of muscle. Since judgments of the activity of muscles depend in such large measure on the tension developed, or the amount of shortening and be reduced, it is important to examine the fundamental basis of these observations. A. Resting muscle. Why in a muscle is stretched without stimulation, the tension increases, not in accordance with Hooke's law, but in such a way that equal increments of length cause progressively greater increments of tension. Over a considerable range of lengths, this resting tension is an exponential function of the length. This suggests that as the stretch increases, new structures within the muscle are called upon to offer resistance. This resting tension is probably due entirely to the connective tissue of the muscle, the endomysium and perimysium, and the sarcolemma surrounding each individual muscle fiber. Banas and Zcetylin have succeeded in removing the sheath from the frog gastrocnemius muscle and have found that the sheath alone gives the same tension on stretch as the whole muscle. It seems probable, however, that the muscle fibers themselves must contribute something to this tension, for after dissection, free from the connective tissue of the muscle, they show the same exponential type of stretch curve as the whole muscle. Azarnus and Ramsey and Streetot obtained a similar result with single muscle fibers, but they showed that this tension is almost entirely due to the sarcolemma, because the continuity of the muscle substance itself could be broken by pinching the fiber without altering the resting tension. And the sarcolemma alone gave the same percentage stretch for a given tension as the intact fiber. From histological study, the sarcolemma has been likened to a knitted stocking composed of a network of fibers, and as such it seems well suited to its task of resisting injurious this idea that the contractile mechanism itself, the myosin chains, exerts no resting tension on stretching is somewhat at variance with the finding of Höber, who made artificial myosin threads containing, like muscle, 20% protein, and found that they resisted stretch with the same tension as would be expected from whole muscle of the same cross-section. Presumably, the molecular configuration of the myosin chains differed in these two cases, those in the muscle fibers being perhaps more folded. Below a certain critical length, the artificial myosin threads showed jolt-lastic rather than clastic centage. If these myosyncleanes are really capable of shortening spontaneously with release of considerable chain previously stored energy, as soon as some stabilizing influence is removed by the stimulus, this easy extensibility seems remarkable. If the protein grid is too stable to shorten, it might well be too stable to stretch without the application of an appreciable force. If, however, contraction involves the development of new shortening linkages, the high extensibility of the resting myosin is readily understood. When a resting muscle is stretched, other changes occur in addition to the mechanical increase in tension. Most important of these are the increase in threshold of stimulation, a slight 10% increase in the velocity of conduction of the excitation wave, Wilska zero, and increased alkalinity, Margaria, and an increase in the rate of resting metabolism, Fang Alpine. Apparently, the stretch of the muscle has some sort of stimuli-Wang effect, and this stimulation is diminished in amount by previous electrical stimulation of the muscle, even if the muscle twitch is not diminished in magnitude. Stimulated muscles. A plot of muscle tension against muscle length constitutes a length tension diagram upon which can be represented all the various types of mechanical behavior of the muscle. The nature of the length tension diagram for resting muscle has already been described. The type of curve obtained for stimulated muscle varies with the nature of the stimulation. Many authors have confined their attention to the tension developed with single twitches. Thus, Banas and Zetlin reached the conclusion, that the tension of the contractile mechanism increases linearly with the length of the muscle until a maximum is reached, after which the tension increment causal obtained with single twitches of mammalian muscles stimulated in situ. This, however, does not measure the maximum tension which the contractile mechanism is really capable of developing at each length. It measures rather the rate of development of tension at each length, or the amount developed in the brief time of a single shock. To obtain information concerning the nature of the contractile mechanism, it is necessary to use the maximum tension developed in a tetanus of indefinite duration and optimal frequency. Only in this way can the muscle be kept more or less continuously in a state of contraction. The tension will increase to a maximum with increase of both duration and frequency, and it is this maximum which may BCVS be regarded as an index of the fundamental tensile capacity of the contractile mechanism. After deducting the resting tension due to the connective tissue, it is found that the increment of tension due to the tetanus increases linearly with length until ply unloaded resting length is reached, after which it decreases more or less linearly until the length is about twice the resting length. See figure 46, ETG 4. Single isolated muscles fibers. Ordinates tension developed, total minus resting, in percentage of 10. Maximum developed, abscissa, length in percentage of the resting length. Each symbol represents a separate fiber. Measurements of R.W. Ramsey and Sybil E. Street. At length less than 0%. The shortening is irreversible, and the fiber is in the delta slate. This property of the contractile mechanism is one of great importance as a clue to the nature of contractility. It is easily confirmed in human muscle, so far as the lengths below the resting length are concerned. Over this range, the longer the muscle, the greater the tension. Greater lengths cannot be investigated in man, since the resting length and the maximum extended lengths in the body are approximately coincident. The length tension diagram of the stimulated muscle indicates an increase of tension with increase of length, at least over the range of lengths which are physiologically useful. This is by no means an obvious relationship, and when pushed to its logical conclusion, becomes a point of some importance in any attempt to formulate a theory of muscular contraction. If, for example, one imagines that a muscle consists of n cylinders of radius r arranged in parallel, which are caused to shorten as a result of a sudden increase in surface tension AR, then it is evident that the pull will be equal to E turn rate. The radius will have to be nearly molecular in dimensions and n correspondingly large in order to explain the force. Now, if the volume of the fibers remains constant as the length decreases, the value of R will increase, and therefore the total pull will increase so long as the change in surface tension remains the same. Hence, a length tension diagram would be obtained in which greater forces were exerted at shorter lengths, or conversely, if a muscle could lift a load at all. It would lift it as much as if the load weighed little or nothing. This theory, therefore, does not work very well. According to another theory, the force exerted by a muscle is due to an attraction between opposite electric charges. Oppositely charged metal plates attract each other with a force which varies inversely as the distance between the plates. According to this theory also, the force should increase as the muscle grows shorter, and we are confronted by the same type of difficulty as before. However, both of these theories, so far as this objection is concerned, can be salvaged by a supplementary hypothesis. As the muscle shortens, the connective tissue is pulled out of shape. Just as it resists an undue amount of displacement by stretching, so also it offers elastic resistance to increasing shortening, which stretches progressively the transverse connective tissue elements. In the absence of a load, the tension of the contractile mechanism comes into equilibrium with the transverse elasticity of the connective tissue. Whole unloaded, isolated muscles, when stimulated, do not shorten to more than 50 or 60% of their resting length. In the body, the antagonistic muscles, as well as the anatomical limitations of the joints, offer still other barriers to further shortening. The individual muscle fibers may shorten more in percentage than the muscle as a whole. But even so, they do not shorten in the body to more than 50% of their length when individual fibers are isolated from frog muscles. They do not readily shorten to less than 66% of their resting length, but with continued stimulation, they do proceed to shorten more slowly down as far as 20%, Ramsey and Streets. After shortening over this unaccustomed range, the fiber is no longer capable of relaxation and passes into what the authors call a delta state. See, in this new state, the fiber can be re-extended as before, and it will give the same resting tension as the normal muscle. This means that the longitudinal fibers in the sarcolemma have not been changed in their elastic properties. When stimulated, however, the delta state fiber develops less tension than before, and at all lengths below the resting length, it fails to relax. The diminution in the contractile force may indicate that some of the myosin chains folded up so much that they remained folded, while the rest of the myosin chains were re-extended. There is, however, no evidence of this in the microscopic appearance of the fiber, which shows all the usual cross striations after it has been stretched out again to the resting length. Before such stretching, it shows typical contracture bands. The failure to relax might be due to excess sive bulging of the sarcolemma in the highly shortened muscle, so that the circular fibers were stretched beyond the elastic limit and broke. The elastic return would then fail to occur. If the sliepe of the length tension diagram is to be attributed to the elasticity of the sarcolemma, then this same elasticity may be responsible for the active relaxation of the muscle. It is evident, however, that the sarcolemma alone is not strong enough to resist undue shortening when it is deprived of the added support offered by the connective tissue of the WI muscle. When stimulation is continued, the sarcolemma gradually bulges. Its resistance, however, makes it difficult for the myosin chains to find room enough in which to fold up, and the shortening process is therefore much retarded. In the process of folding, some of the myosin, diins, or the micelles may break loose from their terminal attachments. This may contribute to the delay of relaxation, and it may also eliminate a certain number of contractile uni is from future gearticipation in the development of external tension. The phenomenon of the delta state offers some support to the theory that shortening of the stimulated fiber, and therefore the shape of the length tension diagram, is limited by the elastic resistance of sarcolemma and the connective tissue to bulging of the sarcolemma over the range of lengths between 65% and 160% of resting length. The fiber acts in a reversible way. Beyond these limits, stretching or shortening becomes an irreversible process. It is reasonable to suppose that this irreversibility is due in part, at least, to rupture or tearing of the longitudinal or the transverse components of the sarcolemma. Sarcolemmas may also be involved. In SJJ of these arguments, it is quite possible that the role of the sarcolemma in the length tension diagram has been exaggerated. It is necessary, therefore, to consider another explanation for the decrease in shortening with increasing loads. This explanation, which is perhaps more in accord with recent thinking on this subject, attributes contraction to the folding of myosin chains or to the closing of successive links in these chains. If, when the muscle shortens, all links do not close with the same tension, then, when the muscle subsequently lengthens again, some links will pull out with a small load and others will resist extension until a large load is applied. Thus, a statistical distribution of various elements in the shortening mechanism will explain the type of curve obtained. Both of these explanations of the length tension diagram appear to be reasonable, and both may contribute to the actual behavior. Consideration of the second explanation would be much facilitated if an experimental length tension diagram of pure myosin threads in the supercontracted state were available anywhere in the literature, but nothing of the sort appears to have been published. How uniform the various links of these chains may be in their mechanical properties remains a matter of conjecture. From the work of Asbury, it is well established that the contractile mechanism consists of myosin polypeptide chains, which in the resting muscle resemble the partially folded keratin. The myosin differs from the QUJ keratin in its side chains, which are less firmly united, so that it is capable of a wider range of length changes. By heating in steam or immersion in caustic soda, the side linkages, SS groups, JDRHAPs, are broken down so that the keratin can be readily extended or unfolded into the 3-keratin form. One ditch has twice the natural length or can be made to shorten into the supercontracted form, which has about 70% natural length. Thus, when the keratin is devulcanized, it comes to resemble myosin. Asbury was properly impressed with this similarity between muscle and myosin, which was brought up most strikingly by the similarity between the X-ray diffraction patterns given by the two structures. It leads to the idea that the resting muscle, which has the same configuration as myosin, is lealed in that position and prevented from shortening by side chain linkages similar to those in a keratin. On stimulation, side chain linkages may be loosened, thus permitting shortening to occur. On the assumption that the tension of the resting muscle is due to connective tissue, the increment of tension due to stimulation at different lengths, see figure 46, must be due to stimulated myosin, and, in accordance with Asbury's picture, must be similar to the length tension diagram or force elongation curve of supercontracted or devulcanized alpha keratin. It is hardly likely, however, that the force of an elastic myosin thread would decrease when stretched beyond a certain point, as seems to be the case in muscle. The interpretation of the muscle curve at extensions greater than the resting length is therefore difficult. Possibly it means that at a certain degree of extensiology, the joints of mutual attraction on the polypeptide chains become too far apart for further interaction, or the force exerted might represent merely the number of links still existing in the alpha form. See time relation and latent period. It is not proposed to discuss the time relation of muscle contractions in any detail. Both the development of tension and the rate of relaxation are diminished by lowering the temperature and by fatigue. In the twitch, the isometric tension reaches its maximum before the isotonic shortening. This is natural, since shortening can continue so long as the muscle is capable of developing a tension in excess of the load. The duration of the latent period between the moment of stimulation and the beginning of the mechanogram is also diminished by an increase in temperature. It is suggested that this represents some chemical reaction which supplies energy to the myosin. There are those who believe that the true latency is measurably short or less than 0.4 milliseconds. while others find an interval of about 3.3 milliseconds in the frog, however, to doubt whether any amount of increase in mechanical sensitivity can detect the first curling of the myosin chains. The mechanogram begins asymptotically, so that the true point of onset appears to depend upon the sensitivity of the apparatus. Recently, however, Sandow, using a piezoelectric crystal and a cathode ray oscillograph for recording, has reported an initial relaxation of the muscle before the first detectable increase in tension. The same observation has been reported previously extensibility of the myosin chain just before actual shortening begins, as if certain stabilizing side chain linkages were broken before new shortening linkages could be established. During this brief interval, the weight is able to stretch the muscle slightly before the contraction begins. In the light of these results, the question of the reality of the latent period loses some of its interest. Indeed, it becomes of more importance to learn what evidence of stoichiometric changes in the myosin may be detected during the apparent latent period. Types of contraction. Contractions are conveniently classified into two groups: 1, those accompanied by shortening, and 2, those accompanied by lengthening. An alternative classification is that proposed by the distinguished pioneer in this field, Adolf Fick, to whom we owe so many important contributions to the mechanics and energetics of muscle contraction. He it was who coined the familiar terms isotonic, constant load, and isometric, constant length. Each of these, however, is oidy and abstraction and seldom occurs in practice without very special precautions. On the other hand, all contractions observed in practice are accompanied by more or less shortening or lengthening. Moreover, the differences between these two types of contraction, particularly the differences in rates of energy supply, are of fundamental importance for an understanding of the nature of the contractile process and the progressive change in the contraction, which occurs as the rate of shortening diminishes and the rate of lengthening increases, provides a quantitative relationship of great importance as a clue to the nature of contractility. Theoretically, between these two types of contraction, there stands a third type, the isometric. With this addition, the following scheme will be helpful. 1 Epic, Mech, Arbiet, und Weiderdrehtiermenweg 1. Bei der Muskelzähligkeit Leipzig 1882-112, wherever tension is steadily maintained without movement, we may conveniently speak of an isometric contraction, although the length certainly changes during the onset of tension, and even while external tension is apparently constant. The internal tension within the individual sarcomeres or inside the muscle fibers themselves is undoubtedly fluctuating with the arrival of each successive nerve impulse, Nicolai. Some internal shortening, therefore, is undoubtedly involved. In the rest. 72. Effect on tension of increasing velocity rate of energy supply. Some F-points illustrated in this scheme will require amplification later. It is only necessary to add here that a contraction accompanied by lengthening is in no sense an artifact of the laboratory, but a phenomenon which occurs wherever muscles operate in antagonistic pairs. In such a case, the agonist accelerates a limb at the beginning of its stroke, and the antagonist decelerates it at the end of the stroke. During the latter process, the muscle lengthens during contraction. Equation of Nyovment. A part of the schema given above may be expressed more generally and more completely in the form of an equation xi low and low is the length at which t equals 0. A is a coefficient of elasticity and is constant only over a range of lengths where Hooke's law applies. This states that the total tension T of which the muscle is capable at the observed length and under continuous optimal stimulation will be utilized either to balance a load IC or elastic force stacks, or to overcome viscosity energy will be in the process of conversion into geotential energy, clastic or gravitational, or heat, viscosity, or kinetic energy, acceleration. Such an equation applies, of course, to all moving objects. Since wave motion can be analyzed in terms of moving objects, it applies also in its essential to the transmission of sound waves and mechanical waves. And to electric circuits, of course, T is potential, A is capacitance, H is resistance, and C is impedance. A great variety of experiments on muscle have been analyzed with the help of this equation, and with the assumption that T is constant for a given muscle at a given length. The object in all cases was the accurate description of the mechanical properties of the muscle in both the resting and the stimulated condition. Thus, a small hammer is allowed to strike against a muscle, and the extent and rate of the rebound are measured, Steinhausen, Richter, or a suspended muscle is set into a to and fro rotation around its longitudinal axis, Weber, Lindhard, and Molleron, or alternately stretched and released by attachment to a vibrating spring. And the period and damping of this oscillating movement were recorded, Iosser and Hill. Hogben, the results of these and similar investigations are not in good agreement. So far as the elasticity is concerned, some have reported a decrease in elastic ida due to contraction, DQI an increase, and some no change. This confusion is due chiefly to difficulties in interpretation, which may be illustrated by one of the experiments of Gasser. And they found that for a given sudden stretch of the muscle, the rise of tension was much greater if the stretch was applied immediately after stimulation than it was if applied at later or earlier periods. The experimental result is very clear and is confirmed. Tension L length, this result may be said to indicate an increase in elasticity. This maximum elasticity was obtained, however, at a time when the tension, at least the internal tension, if not that manifested externally, was increasing and way, just because the muscle had been stimulated, and not because of the stretch. Two factors contribute, therefore, separately to increase the T at this moment. The interpretation is accordingly complicated, and it might well be argued that no change in the fundamental mechanical properties was indicated. Similar difficulties exist in the attempts to measure changes in viscosity of muscles during contraction. Gasser and Hilp have reported an increase, but Hogben and Pinhey, with essentially the same method, found no certain change. Petit found also an increase, but Winton in smooth muscles found an increase with direct current stimulation and a decrease during actual contraction with alternating current stimulation. Followed that what has been called viscosity in muscles is largely a delayed shortening due to limitations in the rate with which energy for shortening can be mobilized. And Hill has more recently accepted essentially this view. The story about muscle viscosity, therefore, has become outmoded at just about the time when it succeeded in making its first appearance in the textbooks of physiology. Weher in Wagner's Handwater book, Die Physik 3, Part 2, 110, Braunschweig 7 chemical changes associated with contraction. Aerobic, the rate of oxygen consumption is enormously too increased as a result of contraction. Itzerig, a thin frog sartorius muscle in a respirometer, this increased oxidation persists for some 30 minutes. The increased oxygen intake apparently begins before the increased excretion of carbon dioxide. This could be due, in small part, to the smaller diffusion constant of the carbon dioxide, or possibly L efficiency in carbonic anhydrase. But it is chiefly due to the increased alkalinity or binding capacity of the muscle resulting from phosphocreatine breakdown. Millikan has shown by spectroscopic observations of the muscle hemoglobin bands during stimulation that oxygen is used almost simultaneously with the contraction. And it has been argued, that oxidative energy is primarily available for direct use during contraction, and that it is not purely a phenomenon of recovery. According to this theory, it is only in the absence of free oxygen that lactic acid is formed, which must be removed in recovery. It has been shown, however, by D.K. Hill using a thin muscle in a respirometer at low temperatures that even when free oxygen is continuously available, it is not used until the contraction is over. At low temperatures, the rates of red ion are so much slowed down that the rates of diffusion have very little effect upon the rate of oxygen consumption, and correction can be made for this delay. The analysis shows that the usage of oxygen coincides in time with the appearance of the recovery heat, even under these SLI initial conditions. Since recovery heat always follows the contraction, it can be concluded that the use of oxygen also is a recovery process, even at higher temperatures. The studitives of oxygen consumption show, therefore, that the contractile machinery is not coupled directly, but only indirectly, with an oxidative reaction. Measurements of the respiratory quotient indicate that it is carbohydrate which is used directly for the I-ditamate superior of energy for recovery. When the RQ indicates a burning of fat or protein, these substances are used only indirectly after conversion to carbohydrate, JCMLP. Even in isolated frog muscles, the excess respiration resulting from activity there is evidence that the resting respiration of muscle differs in nature from that initiated by contraction. The latter can be inhibited by a concentration of sodium azide, which leaves the resting metabolism unaffected. Standard 0, it is proposed that the latter is not mediated by the cytochrome oxidase enzyme system. Energy measurements made simultaneously by direct and indirect calorimetry have shown that within the limits of error of the metaliide used, all the heat developed in an isolated muscle during contraction can be accounted for in terms of the oxygen used. Fennerdu. This is, of course, to be expected. The disposition of this total excess oxygen, which is used in recovery from contraction, is shown in Figure 47. In the diagram, it is assumed that, after contraction, the metabolic products, chiefly lactic acid, requiring oxidative removal are equivalent to 1 gram of glycogen. The complete combustion of this would require 3,800 calories, but Meyerhof has shown that only do this need be burned for complete recovery. The total oxidative energy is therefore 700 calories. But if oxygen is admitted to the muscle after an anaerobic contraction, only about 380 calories will appear as heat. The remainder, described as latent heat, serves to reverse the initial heat or the anaerobic processes, which, altogether liberated during contraction about 380 calories per gram of lactic acid formed, the exact figure depending on the amount of phosphocreatine breakdown, the fatigue of the muscle, and other conditions of the experiments. This 380 calories of initial heat is equal to the latent heat of recovery, and is the algebraic sum of the heats of all the anaerobic processes of contraction, including the formation and neutralization of the lactic acid, phosphocreatine breakdown, etc. Only about half of the energy which appears as initial heat can be used for work, the remainder being wasted as maintenance heat, or as unexpended mechanical potential energy, that is, relaxation heat. The maximum amount of energy which can appear as work under optimum conditions will be equal to the free energy of the reaction, which is indirectly coupled energetically with the shortening of the myosin chains. Since myosin has been identified as the enzyme essential for dephosphorylating adenyl pyrophos, HATE, it seems most probable that it is the free energy of this reaction, which appropriates as work. The work amounts to only about half of the initial heat, or UFI% of the total oxidative energy. The efficiency of isolate muscles is less than these figures from human XBC primates, but Hilchen has recorded an initial efficiency of 40%, or 20% for the whole cycle. KIG 48. Diagram representing various energy reservoirs in muscle. Energy is derived from oxidation, glycolysis, lactic, phosphocreatine breakdown, or adenyl pyrophos B anaerobic changes. There is so moily complexity to the anaerobic changes which occur in muscle contraction that no attempt will be made to give a thorough review of the subject. It will be sufficient to present the theory of Lomians and Meyerhof, which illustrates the probable relation between the three most important of the chemical reactions concerned. This can best be done, perhaps, by the use of a simple model illustrated in glycolysis and PC resynthesis, occurs again through a phosphate transfer from 1,3-diphosphoglyceric and phosphopyruvic acids. The schema indicates that all these reservoirs could be returned to their resting levels at the expense of oxidative energy alone. And there is good evidence for this, at least in the case of lactic acid and phosphocreatine. The flow of oxidative energy can be blocked by KCN or sodium azidenans, and the energy from glycolysis can be stopped by iodoacetic acid, LLA, which inhibits the conversion of triocephosphate to phosphoglyceric acid. This being the first step of great energy yielding the process of lactic acid formation. When this block is applied, the phosphocreatine is readily lowered by stimulation to the level at which rigor mortis sets in. In the absence of lactic acid formation, this is an alkaline rather than an acid rigor. The breakdown of 2 moles of phosphocreatine yields just enough energy to cause the resynthesis of 1 mole of adenyl pyrophosphate from adenyllic acid. The matter is still further complicated, however, by the fact that adenyl pyrophosphate reacts also with glycogen to give hexose diphosphate, and eventually the adenylic acid form can also be phosphorylated once more by reaction with subsequent intermediary products, such as triocephosphate and phosphopyruvic acid. The more recent evidence, moreover, indicates that myosin also combines with phosphate at some point in the cycle, removing it from adenylic acetyl. See pages 485 and 521. Thus, it may be said that J-phosphoric acid is passed from adenylic acid through myosin to hexose, which forms lactic acid and gives it to creatine, which finally returns it to adenylic acid. The reactivity of all these compounds is, therefore, much facilitated by the phosphoric acid. Recent discussions of the subject are given by Needham and Meyerhof. The evidence that adenyl pyrophosphate is the first source of energy to be called upon has been incomplete, because of the difficulty in showing that the amount of this substance present in muscle is appreciably diminished after activity. In the light of this scheme, it would have to be assumed that it is rebuilt about as rapidly as it is broken down. Studies with radioactive phosphate show evidence of the expected turnover in aphosphate if due allowance is made for extracellular phosphate. Furchka and Shore. Indirect evidence of the correctness of this view, as illustrated in PG 48, has been supplied by measurements of the changes in the volume, OI-acity, and of the muscle. C. Volume changes. A decrease in volume on contraction was first reported by Ernst and was extensively studied by Meyerhof and Hartmann. G. Meyerhof and Hartmann have measured the volume change which occurs with each of the chemical reactions pictured in Figure 48. Lactic acid formation causes an increase of 24 cc per mole, while the dephosphorylation of phosphocreatine and adenyl pyrophosphate causes decreases in volume of 14 and 21 cc per mole, respectively, alternately. Using these figures, it was possible to account quantitatively for the observed decrease in volume on the basis of chemical analyses for lactic acid, L-14, and PCO. This was true, however, only when muscle was prevented from engaging in spurious reaction with the surrounding linger solution by immersing it in paraffin oil. With this refinement, the method provides a valuable means of determining the time relations of the chemical reactions involved in the contraction of muscle. In later studies, however, by Meyerhof and Einvial, in which the muscle volumes were followed over longer periods of time, the agreement was less good, and in general, some further factor causing a decrease in volume had to be postulated. This discrepancy might possibly be due to increase in intracellular water, decreased solubility of the myosin, or liberation of the potassium. An interesting experiment of Hartmann's using this method is illustrated in Figure 49, which shows the tension, upper curve, and the volume changes, lower curve, in a series of 2-second tetani in a gastrocnemius muscle, and Marseille in the dilatometer under paraffin oil. In each tetanus, as the tension rises, the volume decreases rapidly, indicated by a rise in the curve, and more slowly as the tension is maintained at its plateau level. In relaxation, the formation of lactic acid predominates and the volume begins to increase. At first, the volume remains smaller and the curve higher after each successive relaxation than it was originally, but as lactic acid accumulates, the volume begins to increase more and more, and this continues until the point of etadomalm fatigue. The last tetani of the series are illustrated via altered calibration values in the lower two curves. A slight decrease of volume with each tetanus still remains, suggesting that no tension can be developed unless some volume decreasing reaction, like dephosphorylation of AP or BC, is available. Since the first change in volume which occurs during stimulation is a decrease, this supports the idea that PC or API breakdown occurs nearly simultaneously with the actual contraction. Thus, it appears that the volume changes of a muscle can be used to some extent as an instantaneous index of the chemical reactions occurring inside the muscle fibers. The method must be used, however, with caution, for Fischer has observed an increase in volume of the sartorius muscle when stimulated at high initial tensions. This matter deserves further investigation, for it suggests that all the factors have not been successfully elucidated. Deopacify the moment of stimulation. A muscle becomes more transparent due to pliosphoconitin breakdown, and this effect is more slowly reversed in relaxation. Von Bayer and Moralt lactic acid formation causes an increase in the opacity if the muscle is in Ringer's solution. Willier swelling and an increase in the volume of the colloidal particles can occur. Thus, in heat rigor, lactic acid forms, and the muscle becomes opaque, while in iodoacylate poisoned muscle, the lactic acid no longer forms, and rigor causes a decrease in opacity. This method confirms, therefore, the conclusions obtained by other means. It should be pointed out, however, that the opacity of the muscle will vary greatly with the wavelength of the liglet that is used. The result described was obtained with white light and may depend upon a change in the amount of light scattered by the muscle proteins. A more specific effect is obtained if a wavelength is used, which corresponds to the absorption band of a special chemical constituent of muscle, such as those of myoglobin, the yellow ferment, or cytochrome. In this way, it can be shown that within 50 milliseconds after stimulation, the yellow ferment and cytochrome are reduced, as is indicated by increases in the opacity of the muscle to the particular wavelengths used. The reduction of the myoglobin begins 0.22 seconds after contraction. pH, many investigations indicate an initial increase of pH, which is followed later by a decrease in a respirometer. The alkalinity can be shown by the absorption of CO2 and the acidity by an elimination of CO2, Lippmann and Meyerhoff with a glass electrode. Similar changes have been demonstrated in human arm muscles, Maison Orth and Lemergier, with a somewhat modified glass electrode technique. Dubuisson has all attained confirmatory results in isolated muscles. In general, a muscle shows an increased alkalinity early in contraction, which gives way to an acidity from lactic acid formation in recovery. The time relations are more accurately analyzed in a slow, smooth muscle, frog stomach, from which the data for figure 50 were obtained. Similar results were obtained, however, in gastrocnemius muscles. He dotted line shows the tension as a function of time in seconds. The other curves show heat production in cal x10 per gram of muscle, as calculated from the observed pH changes. The earliest curve, 5, represents the breakdown and resynthesis of adenyl pyrophosphate. Curve C, representing the breakdown and resynthesis of phosphocreatine, begins to rise as B4. Finally, curve D represents the heat produced by the formation of lactic acid, which is the only metabolic product remaining after 30 seconds. In a later publication, Dubuisson discusses a still earlier phase of increased acidity, which is observed in short isometric or in isotonic contractions of the sartorius muscle. When a muscle is stretched, he finds, in agreement with Margaria, that it becomes alkaline. Hence, when it shortens isotonically, it should become acid, as Dubuisson found. This change is attributed to a change in the isoelectric point of the myosin as a result of the stretch. Myosin would therefore be expected to liberate base when it is elongated. This curious reaction may play some role in the electrolyte changes of muscle, and because of it, the initial pH change varies in different muscles according to the load and length of the muscle. It is not certain just where in this phosphate cycle the contractile machinery comes in, or in what particular form the energy has to be presented in order to cause contraction. An interesting suggestion made by Kalikar is that myosin itself combines with phosphate, which might serve as one of the side chain linkages in the myosin grid. If this phosphate stabilizer were removed on stimulation, it could then start the energy cycle going by reaction with one of the other components in the phosphate cycle, and the protein chain, freed of the stabilizing influence of the phosphate, would be able to release its previously stored potential energy. Finally, after the end of stimulation, the phosphate could recombine with myosin, thus pulling the protein chains back to their previous resting length and storing up potential energy for another contraction. This postulate has at least the advantage that it illustrates for the first time a possible method of energetic coupling between the anaerobic reactions and the contractile mechanism proper. The most recent development in muscle chemistry is the discovery that myosin itself acts as a dephage horylating enzyme, and therefore participates directly in the phosphate cycle. The importance of this for the theory of muscle contraction has already been mentioned under myosin, and will be discussed further at the end of this chapter. Impedance. For the measurement of the impedance during contraction, the muscle is placed between electrodes and balanced electrically in a Wheatstone bridge circuit, using a high-frequency alternating current. In strictly isometric contraction, such as can be obtained in a sartorius muscle, there is an increase of impedance, Bosler. A shortening of the fibers, however, is accompanied by a decrease of impedance. The greater the shortening, the greater the change of impedance. In the isometric contraction, the increase in impedance is analyzed into a quick component, which reaches its maximum before the tension maximum, and a slower urasis, which is approximately coincident with relaxation. The interpretation of these two processes is difficult, but they correspond respectively fairly well to the myosin adenyl pyrophosphate reaction and the phosphocreatine breakdown, and have been so interpreted by Dubuisson. In time relations, they also correspond to the spike and the after potential of the electrical change of muscle. It is usual to think of action potentiles as reflecting the potential difference across the surface membranes of the cells. A similar explanation could also apply to the high-frequency impedance. It is not impossible, however, that the effect is an expression of a change in the state of aggregation of myosin, which would not be an exclusively surface process. This idea is confirmed by the parallelism between the decrease in double refraction, which likewise occurs in two peaks, von Marolt, Fischer, and has indicated theoretical reasons why a change in double refraction of myosin should result in a change in impedance. 8. The thermoelastic effects. The mechanical behavior of muscle, as well as its ultrastructure, has already been considered. We must now deal with another method of attack, which is concerned with the interpretation of the mechanical behavior of muscle in terms of its ultrastructure. It represents an approach to the problem through applied principles of thermodynamics. Unfortunately, the method is really applied only to the resting muscle, although some attempts to extend the application to the stimulated muscle have also been made. The general principle of the method may be illustrated by a concrete example. It is known that a muscle over certain ranges of length gives off heat when it is stretched and cools when it is raised. All these was originally shown by Heidenhain and was confirmed by Alarchy and Hilfgen. Conversely, the same reaction should be reversive, so that the addition of heat to the muscle or an increase in its aerature should cause it to shorten or to increase its tension. This deduction from the irreducibles of thermodynamics was worked out by Lord Kelvin, and the relationship, as expressed by Hill, is represented by the Isueshu increase of tension, mean length, where Q is the heat in calories. A VDIO OVF thermal expansion T the absolute temperature AMI, where tension is expressible in gins, weight, and the length in centimeter. In this respect, muscle BIs like rubber and unlike most other clastic bodies. If now we can tell from thermodynamic principles what kinds of ultramicroscopic structures behave like muscle and what kinds become cooler when they are stretched, then we shall learn something about the ultrastructures in muscle, which are responsible for the manifestation of tension. The theoretical treatment of this subject has been presented by Meyer and Meyer and Picken. Already an abbreviated presentation will be attempted here. Two fundamentally different mechanisms for the development of tension on stretching may be distinguished. These may be illustrated by a steel spring and B gas put under pressure by the stretch, figure 51. In the first case, the molecules of the steel are pulled apart and displaced from their positions of minimum energy. As work is done on the SEI ring, and the accipial amount of geotensile energy is stored in it, and there is no exchange of heat. In the case of a stretch which is resisted by the compression of a gas, as in by the work which is done against the gas in compressing it, PV as heat in the gas, and is not stored as potential energy. Conversely, double Newey pressure is released. The work the gas does in expanding back to its original J position is derived from heat energy, and a PP as a cooling of the gas. Idle resisting force is due to the bombardment of the molecules against the piston. The movement is merely the statistical result of the larger concentrations of molecules which are present below the piston. A third TJE of structure is really a mollification of type B. It consists of long molecules fastened together in chains. The movements of these molecules are not quite random because they are somewhat restrained by the chain structure. When the structure is stretched, ply restraint is increased, just as the molecules of a gas are more restrained when the piston in B is pulled down and the gas is compressed. It is the tendency of the molecules to revert to a more random Josephson or to increase the entropy of the system, which is responsible for the restoring force in both cases. The classical example of type C is rubber. A useful lecture table demonstration can be set up with a steel spring and a rubber band, each supporting a weight, and so attached to a light lever that changes in length are greatly magnified. If each of these structures is warmed slightly with a match, the steel spring lengthens while the rubber band shortens, unless the load is too small. Likewise, if the rubber is suddenly stretched, it will feel perceptibly warm if placed against the upper lip. Similarly, the gas in type B warms up when it is compressed. Rubber is composed of long primary valence chains which can fold and unfold to give long-range elasticity, and which by their regular orientation when stretched cause birefringence. It is possible to account quantitatively for the elasticity of rubber by the entropy changes, since muscle shows the same type of thermoelastic behavior as rubber. It is natural to transfer to muscle the theoretical concepts which have been developed so thoroughly for rubber, and to explain the elasticity of muscle by the thermal agitation of the myosin molecules, which tend to resist the forced longitudinal orientation. Some samples of rubber show a short Hooke's law region in the beginning of a stretch, and over this range they belong to the A system Guth. Meyer and Picken found the same thing for resting muscle, but WDA Lish and Rank do not find this when most of the connective tissue is eliminated. All observers agree that both rubber and muscle behave like the Yi type at large extensions, where molecules are pulled apart out of their positions of minimum potential energy. In the muscle, this is attributed to the connective tissue elements, which begin to resist further stretch at this point, like Ys at such lengths. Muscles begin to lengthen instead of to shorten with increase of temperature. They become cooler instead of warmer on stretching. This interpretation of the reversal of the thermoelastic behavior at large extensions is confirmed by the behavior of the connective tissue itself, which definitely belongs to the A type of elasticity, which always cools on stretching. This analysis of the resting muscle, according to the thermoclastic theory, appears to show that the tension of the muscle during extension is at first due to the decrease of entropy of the myosin chains, and at greater extensions, to the clastic potential energy of the connective tissue. Some attempts have also been made to extend Tly theory of elasticity of rubber to cover the active contraction of muscle. This is the thermokinetic theory of WISA. Test of this theory might be found if it could be shown that the force exerted by the contractile machinery increases in J-rho or portion to the absolute temperature. According to Bernstein, the force in a muscle twitch decreases with rise of temperature and has therefore a negative temperature coefficient, like surface tension. This, however, can easily be shown to LA due to the fact that the heat production is also increased by a slowing of the reactions which accompanies the fall in temperature. The amount of tension produced per unit of energy liberated is independent of the temperature. The constancy of the TH, tension to heat, ratio in twitches at different temperatures would seem to indicate that temperature had little effect upon the contractile machinery itself. This is not in conflict with the thermokinetic theory, which demands only an increase of about 10% in tension for a rise of 80 degrees Celsius in temperature. It would seem that more reliable evidence could be obtained from the effect of temperature on the asymmetric tetanus tension. It would be necessary to choose a maximal stimulus, and a frequency W-inch is sufficiently high to produce maximal economy, bronch, even at the highest temperatures. It might be argued then that the tension so produced at each temperature would represent the maximum of which the Miocene machinery was capable. On this basis, it appears that in striated muscle, temperature increases the tension, produced tension decreases under similar conditions. It is doubtful whether this can be taken as a fundamental difference between these two types of muscle. It seems more likely that the interpretation is incorrect, and that there is no certainty that the supply of energy can be made optimal at all temperatures. In striated muscle, the energy liberated at different temperatures increases more rapidly with temperature than the tension developed, and the increased tension may reasonably be attributed to the increased amounts of energy available. The experiment of Winton with smooth muscle may then be attributed to the fact that the heat did not increase with temperature sufficiently to compensate for T-like more rapid dissipation of the tension. These difficulties of interpretation make it impossible to obtain a satisfactory test of the thermokinetic theory by temperature effects. It should be emphasized at this point that it is the resting muscle which resembles rubber in its thermoelastic effect. In the stimulated muscle, exactly the opposite behavior is neonatal shortening liberates more heat, while stretching decreases the H at G reduction. Taken at its face value, this does not support the idea that the active contraction of a muscle is merely a matter of forcefully oriented molecules seeking a more probable position by random heat movements. To apply this theory, some excuse must be found for the heat. Nevertheless, Kerrer has supposed that the long-chain molecules arc held taut in the resting muscle by some stabilizing influence, which is suddenly RCN, oved at the moment of stimulation by the excitation process. The resulting contraction should be accompanied by cooling, so the observed heat must be attributed to some other purpose. The analogy between rubber and muscle is also illustrated by the fact that an increase of external pressure causes a shortening in both. As was first shown by Höber and Eisenbrink, both structures have a high compressibility and a low extensibility in the direction of the fibers, and vice versa in the direction across the fibers. In other words, they show elastic anisotropy. From this point of view, the shortening is to be regarded as a purely physical effect rather than a physiological response. It leads W.D. Yalish-Dye to suppose that at the moment of contraction, some change occurs in the muscle, which is equivalent to an increase in the external pressure and causes a shortening. Höber has a somewhat similar idea in proposing that the contraction of muscle is a heat contraction, since heat also causes both rubber and muscle to shorten. In the muscle, the heat is supposed to act in some ill-defined way, not by doing the work itself as in a beat engine, but by liberating previously stored energy. Again, the theory seems to work well enough for a resting muscle, but the stimulated muscle does not lend itself readily to this simple interpretation. The contraction of a muscle produced by high pressure has been thought to be related to the Ernst effect, according to which the muscle decreases in volume when it contracts. Thus, a highly external pressure weakly would cause it to decrease in volume should also cause shortening. According to Hartmann, the decrease in volume is due merely to the breakdown of phosphocreatine, since that reaction in vitro gives a similar decrease in volume of the right order of magnitude. In that case, the compression shortening of Abeka could be due to a breakdown of L-phosphocreatine caused by the pressure rather than to an effect of pressure directly upon the myosin fibers. This interpretation seems to agree better with the view of Cattell, who regards it as similar in every way to any chemical contracture, CP 511, there being the usual energy change, etc., associated with it. Moreover, if the pressure is maintained, the contracture undergoes relaxation after a short time, and the amount of tension developed is not directly proportional to the pressure, but is instead an S/I ed function pressure. Finally, it has been stated by Hassan-Bring that there is a latent period of 0.03 seconds betwixt the onset of pressure and the beginning of the shortening. This is taken to indicate that ply compression shortened is a physiological response rather than a true physical effect, taking all Tly's matters into consideration. It is difficult to believe this thermokinetic theory of W.W. Klein offers much help in explaining the mechanism of active contraction, although it appears to explain certain phases of the behavior of resting muscle fairly well. The effects of pressure. In this connection, it is appropriate to mention other effects of increased pressure upon muscle. A thorough discussion of the general problem has been given by Chittle-Idle Essential Facts, which seem to bear upon the problem of contractility, are as follows. Amoeboid movement. Protoplasmic streaming and ciliary activity after initial acceleration are inhibited or stopped by pressures of the order of magnitude of 300 atmospheres. This is regarded as due to oscillation of the protoplasmic gel, the contraction, of which is believed to be responsible when a pressure of 100 to 200 atmospheres is applied during the early part of a contraction, at which time the muscle is still in the process of being activated. It causes an increase in the tension developed as well as an increase 207 DA Marsland, P127. In the structure of protoplasms, by W.A. Seifriz, AM's entire amount of activation as judged by the measurements of the heat production. In other words, the tension is increased because more energy is liberated by the same stimulus when the pressure is Higlite. When still higher pressures of 800 to 400 atmospheres are used, the muscle may be activated without TBE application of any electrical stimulus, thus causing the conjuration shortening of Abeka, while each Lias already been discussed. 4. The direct effect of PSRQ-UG on the contractile machinery is to cause a diminution of the tension developed, just as it causes a diminution in the contraction of amoeba, etc. Thus, pressures applied after the initial stages of the contraction cause a decrease in the tension developed, and a sudden release of pressure causes a sudden increase in the tension as this inhibiting influence is removed. In fatigued muscles and muscles at very low temperatures, the increased activation due to the high pressures appears to be absent, and the inhibiting effect only remains. The latter effect may be purely exhibitical, while the former involves more physiological processes, such as possibly the breakdown of G-hospleocreatine. The alpha process, the effects of J-Drescher have been utilized by L. Aranen to study the time course of what he calls the alpha process, and what Gasser and Attilas have called the fundamental process of muscle contraction. During the early stages of contraction, the AA application of high J-Dreschers, as already mentioned, results in an increase in the tension developed. This effect soon decays to zero, and in its time relations it coincides roughly with the action geotensile spike, the rate of development of tension, and the rate of initial heat production. If the pressure is applied after the first 0.01 seconds of the contraction period, it causes no augmentation of tension, because after that time the amount of energy liberated by the stimulus is settled and is no longer subject to modification. Gasser and Hill found that a sudden stretch of the muscle causing a sudden increment in the tension was much more effective in that resect act WHM applied immediately after stimulation than later in the course of the muscular resonance, and they interpreted this as indicative of an increase in muscle viscosity at that time. Double Lyell, these two effects are similar in their time relations. They appear to differ in one Ein F. Morton articular time. The application of pressure results in an increase in the energy liberated, Kettle and Edwards. W. Dual, the sudden stretch, tends to decrease the energy liberated, deducting, of course. The work done on the muscle by the stretch itself, zero effectiveness of 20 when the mechanisms of these two effects need not be identical. In the amoeba, it has been attributed to a decrease in viscosity, 20 Weyl, and in the muscle to an increase in viscosity. 20. The sudden stretch depends upon the fact that it takes up I high internal slack in the submicroscopic I contractile elements, thus accelerating the transfer of the energy into tension. The A application of pressure, on the other hand, appears to increase the amount of energy available for transfer into tension. Both effects are additive to the normal rate of tension development and therefore have the same time relations, small differences being of no general significance. The time relations of these effects can be expressed as a curve which has a peak about 0.01 seconds after the stimulus and reaches zero, at about the time when the twitch tension is at a maximum. It does not seem necessary to signalize this by calling it an alpha process as if it were some new and different reaction. It does not detract from the importance of these observations to consider that it indicates merely the time when the rate of the initial breakdown, or the rate of the development of tension, is at a maximum. It does show, however, that Ti internal tension is developed much more rapidly than it is manifested externally in the record of the ordinary isometric twitch. 9. Heat production. Lee, first measurements of the heel production in muscles were made by Helmholtz, who wished merely to demonstrate the principle of the conservation of energy. After him, Caillou, many others, the most outstanding being Eli de Haen, Fick, and Hill, Avexny with the slow thermopiles which were available in the early days, the most tunai. Orton of the facts known today were soon outlined. Both Heidenhain and Pick, for example, discovered that the amount of energy liberated dpp ended upon the mechanical conditions of the contraction, and they recognized the general principle that the greater the work performed, the greater the energy liberated. The imperfections of the technique were such, however, that it was impossible to make this into a clear and quantitative story. The entrance of A.V. Hill into the field was marked by improvements in the technique, which continued with few interruptions until the outbreak of the present war Dauer. In his first work, attention was focused particularly upon the isometric contraction, which was technically the simplest, because it did not involve the complications due to the movement of the muscle against the thermopile. If the part of the muscle beyond the thermopile is, for example, slightly warmer than the part touching the thermopile, then a contraction will bring warmer material against the junctions of the dissimilar metals and will cause spurious effects. It was in the early study of the isometric contraction that the classical work on heat production was completed, and the time relations of the heat production, its various phases, and the variations in the magnitude of total heat under different conditions are Heidenhain, Mechanische Leistung, Warnentwicklung und Stoffresatz bei der Muskeltätigkeit. Breitkopf Hotel, Leipzig, 1864a. Schick, Mechanische Arbeit und der Entwicklung bei der Muskeltätigkeit. Brock of temperature, duration of stimulation, and length were all described in terms of the isometric twitch. Until recently, it has been supposed that isometric contraction was essentially simpler than any other type, but now it has become evident that no contraction is ever really isometric as far as the internal mechanism is concerned. Even though the two animals of the muscle are absolutely fixed in their relative positions, the result is that complications of internal short-ending enter into the interpretation of the isometric contractions as well as the isotonic. And they are the more dangerous because they are not realized, and if they are realized, they are not measurable. The isotonic era, in which proper attention was paid to the heat G reduction during shortening, was ushered in with the measurement of the isotonic contraction in Heer's laboratory in JN. That work, the finding of Heidenhain, was rediscovered. When a muscle shortens Dunlow's work, it liberates an extra amount of heat, which is Cp annotatively equal to the work performed. It is for this reason that the subject of the heat to reduction of muscles can now be better presented from the point of view of the isotonic contraction than from that of the isometric contraction. The former enables one to deal with shortening, while the latter manifests internal shortening in appreciable amount without enriching one to deal with it in a quantitative way. The heat production may be discussed under the following headings. A. Initial heat. The contraction heat and relaxation heat together make up the initial H DAT. In a single twitch, this fraction is given off during the twitch itself, which lasts only about LFO a second in the frog sartorius muscle. The initial heat is followed by the recovery heat of nearly equal magnitude. Since the latter lasts perhaps 600 times as long, 10 minutes, the average rate of recovery heat production is 600 times smaller than the average rate of initial heat production. Consequently, the initial heat appears explosively and determines the magnitude of the initial deflection of the galvanometer, which is recording the potential difference developed in the thermopile. In 1923, A.V. shared with Meyerhoff the Nobel Prize in Medicine, chiefly for his success in working out the time relations of these various phases of the heat production. The recovery heat is largely due to oxidation, and most of it disappears if the muscle is in an atmosphere of nitrogen. The small fraction which persists in nitrogen, amounting to AV. Hill, Muscular Activity, William and Wilkins, Baltimore, 1926. The contraction is over, which provides energy for the synthesis of phosphocreatine. The amount of this anaerobic delayed heat varies considerably with different conditions. It is complete in 20 sec at 17 degrees Celsius. It occurs in oxygen and an equal amount, but is then merged with the true relaxation heat, appears coincidentally with the mechanical relaxation, and is due to the degradation to heat of the elastic potential energy stored in the tensed muscle during the contraction phase. If the muscle lifted a load during contraction and lowered it during relaxation, then the work done in raising the load is added quantitatively to the relaxation heat. In a supposed isometric twitch, the relaxation heat amounts to 0.4 of the total initial heat. In a truly isometric contraction, it is possible that there would be no relaxation heat, because there would be no internal shortening of the anisotropic discs or folding of myosin chains, and no possibility of stretching any other internal structure, such as the isotropic disc. Hence, there would be no elastic potential energy stored during contraction to be degraded to heat in relaxation. H. Contraction heat, shortening heat. Likewise, if there were no internal shortening, that is, if a true isometric contraction could be realized, it is probable that there would be no shortening heat, but only main finite heat. This is shown articularly by the galvanometer deflections, or are recorded in Figure 52 from A.V. Hill-Yang. The insulation on Tly thermopile was so thin and its heat capacity so low that with a rapidly resonating galvanometer he could record muscle temperature directly without serious error. The graphs show, therefore, heat plotted against time from the beginning of a tetanic stimulus. In Figure 52a, curve represents the heat production in an isometric tetanus. It shows a quick rise at the beginning, which was formerly called an outburst of heat, accompanying the development of tension in the muscle. The slower, more or less constant subsequent rise is attributed to the energy for maintaining tension. The isometric maintenance heat rate could, therefore, be measured from the slope of this line at different temperatures and muscle lengths. If now the muscle is not held at fixed length, but is allowed to shorten freely for increasing distances under a fixed load, the galvanometer gives deflections B, C, and D for distances 3.4, 6.5, and 9.6 millimeters, respectively. From these records, it can be said that the initial upswing of the galvanometer could have been caused by an initial internal shortening corresponding in its heat equivalent to about 3 millimeters of external shortening. Here, then, is the evidence that all the true isometric heat is really maintenance heat. This also is the evidence for the shortening heat, a fraction which is proportional to the amount of shortening and independent of the load. The relationship to the load is shown in Figure F2b, in which E is isometric, while F, G, G, and J represent isotonic contractions for a fixed distance under progressively decreasing loads. With all these loads, the same TR tall heat is liberated, provided the weight is removed from the muscle after shortening is complete. With light loads, the shortening galvanometer, in AI, BK, and AK, the initial is isoelectric. When the muscle is released and begins to shorten, the galvanometer gives a sudden deflection upward, as in the other curves. In A, the muscle after release is allowed to shorten under the same load to increasing distances represented by VCE and 1. In B, it is allowed to shorten the same distance under increasing loads represented by J11G and FF. In C, the muscle is released at different times during the isometric tetanus, shortening each time under the same load and to the same distance. To IG, heat is given off quickly in proportion to the high velocity of shortening, and with large loads it is given off slowly, but the total amount is the same. The total energy, of course, is not the same because varying amounts of energy have been taken out of the system in the form of work. This work fraction may be as large as or larger than the shortening heat. The curves of figure 52 are not continued long enough to show the relaxation heat, but if the muscle has to lower the weights in relaxation, then the energy which goes into the weights in contraction reappears quantitatively in the muscle as heat in relaxation. If the loads are not lowered in relaxation, then the relaxation heat will be the same in F and G as in dual, and X shorting to allele. The shortening heat is not due to frictional resistance because an equal amount of heat does not appear as heat during the reverse movement in relaxation. There seems to be some doubt whether in relaxation there is any heat either given out or absorbed, which is equivalent to the shortening heat in contraction. If there is an accipial absorption of heat in relaxation, then the shortening lead may be regarded as a sort of thermoelastic effect for TLI-stimulated muscle. If so, it is opposite in direction to the thermoelastic effect of the resting muscle. The fact that the shortening heat is proportional to the amount of shortening, but independent of load and speed, certainly suggests this interpretation. The answer to this problem will come from a more careful study of relaxation heat. The relation between the shortening and the maintenance heat can also be shown in mathematical form. The heat of an isometric contraction was described by Illertry and Tuuppila. The equation was the heat expended in setting Uji the tension in the muscle, and BT was the maintenance heat, and T the duration of the stimulation. In this accipiation, the moiety includes the relaxation heat, because the 2 refers to the total initial heat of the contraction. If, however, the relaxation heat be subtracted from both sides, the same equation still applies. In this form, the if refers only to the heat liberated during contraction, and the A has become X, the shortening heat, X being the amount of shortening, and being a constant. If the external shortening is 0, isometric, then X represents entirely internal shortening. Energy stored as potential energy in the myosin chains, which will appear as heat in relaxation, is not included. In ideal case, never realized in practice, it may be supposed that X is 0, in which case only the maintenance heat remains. Since the heat varies with TLI size of the muscle and its ability to develop tension, it is usual to write the equation here again 11 refers to the total initial heat, and the absolute values of A and B are correspondingly changed. The ratio 2TL is equivalent to the ratio heat per GM of muscle, tension per centimeter cross-section, since length is approximately weight divided by cross-section. Where T is very short, as in single twitches, TLI maintenance heat lit approaches 0, and the value of 2TL is more constant. It is found to be independent of temperature and has a minimum value at resting length, increasing at both longer and shorter muscle lengths. 225 value also varies with the arrangement of the fibers in the muscle. In the long parallel fibered sartorius muscle, its value is 1/6/8, while in the short diagonal fibered gastrocnemius muscle, its value is 1/13/8. The difference would be expected merely from the arrangement of the fibers if the individual fibers in the two muscles were similar in nature, because the total shortening per cm of length is less in the gastrocnemius muscle, .22s, The T in the equation is unduly large for the gastrocnemius muscle because of the diagonal arrangement of the fibers in the large physiological cross-section of the muscle. Because the ratio 2T is constant, it is usual to suppose that each unit of energy liberated provides for 1 unit of tension. This idea has been extended, is stretched, its oxygen consumption, Meyerhof, Gemernil, and B. Annotato 22.2, in its resting Liat production, Feng 2, are increased along with the increase in tension. According to the figures given by Reichel, the ratio HT of the stretched resting muscle is the same as that for the stimulated muscle. The inference seems to be that a given amount of tension, however it is produced, leads to the liberation of a corresponding amount of heat. In the resting muscle, however, the tension is in the connective tissue, while in the stimulated muscle, it is in the myosin. Moreover, the ratio 2T in both resting, stretched, and stimulated muscle increases with increasing duration of the tension, so there is something arbitrary in the comparison. In addition, the Liat measurements were made at different muscle lengths, without separate calibration of the muscle. The interesting relation brought out by Reichel must be regarded, therefore, as probably fortuitous. If a muscle stimulated at resting length can develop an isometric tension tie, or can shorten without load to a length 3, then the area of this length tension diagram is approximately 1/2 Tx through y equals TLQ, and in the sartorius muscle, this is approximately equal to the observed heat, which was found to be UQ equals R6.8. Thus, it was supposed that the heat represented the degraded potential energy of the length tension diagram. If this had been strictly true, all the heat should have appeared in relaxation when this mechanical potential energy disappeared. According to more recent findings, it must be supposed that this equality between the heat of a single isometric twitch and the area of the length tension diagram of the muscle was largely accidental. Not over 0.4 of this heat is actually due to stored potentile energy, and this appears as heat only in relaxation. The remainder is partly due to maintenance heat and partly to the internal shortening heat, which is produced when the internal potential energy is stored. See maintenance heat. With stimuli of longer duration, the maintenance heat becomes increasingly important and is in a sense the most characteristic quantity which can be measured in an isometric contraction, because it is free of complications due to internal shortening. This maintenance heat is supposed to represent a constant rate of energy production, which maintains the muscle in an excited or contracted state. It is independent of the frequency of stimulation, provided the frequency is sufficient to maintain maximum tension. Theoretically, it begins with the beginning of contraction and continues until shortly after the end of the stimulus. It is difficult to measure it until after the tension has reached its maximum, because otherwise the galvanometer deflection is complicated by the shortened end heat, which is being liberated at the same time due to internal shortened engine. The maintenance heat can, therefore, be measured most simply by measuring the H at production during a short and a long tetanus. The difference between these two quantities of heat, divided by the difference in time, represents the rate of production of the maintenance heat. The shorter tetanus should, of course, be long enough to permit maximum tension to be developed. In Hilf's measurements, the maintenance heaton was found to be increased at high temperatures with a TMJ coefficient between 0 and 15 degrees, GT of 2.8. Bosler suggested that the ratio between the rate of heat production per gram of muscle and the tension maintained per unit cross-section area be called the economy of the contraction. Thus, the economy decreases with rise of temperature and with fatigue. It is greater in slow, smooth muscles than in the raw-id striated muscles, in tonus muscles. It is so great that it has been argued that such muscles possess a catch mechanism which enables them to maintain tension without any expenditure of energy. Actually, in such muscles, the maintenance energy is probably too small to detect readily in the presence of the resting energy production. But there seems to be no necessity for postulating any SBCT mechanism. It is reasonable to consider that the tension in a muscle is spontaneously disappearing at a certain rate, and that this must be replenished at the same rate in order to keep the tension constant. On this basis, any condition which accelerates the rate of disappearance of tension, such as a high temperature, would be expected to increase also the maintenance heat. Whether the tension is maintained by electrical stimulation or by chemical contracture, the maintenance heat and economy seem to be of the same order of magnitude. From a theoretical point of view, the variation of maintenance heat with length of the muscle is a matter of special importance when isotonic and isometric contractions are to be compared. The effective length has been tested by measuring the heat production during isometric tetanic contractions. At different lengths, using this method, Fenn and Latchford found that the maintenance heat, like the heat to develop tension or the heat in a single twitch, passes through a maximum at a length equal to about 90% of the length at which the maximum tension is developed. Similar results were also obtained by Fischer-Dohn in turtle muscle. In these experiments, the heat was elevated for a constant length of the muscle. When the muscle short-schneel, a correspondingly larger fraction of the total muscle weight was to T found between the electrodes. If the heat is calculated for ecipial weights of muscle or for the whole muscle, then the heat falls off even more rapidly as the shortening increases, but less rapidly on the other side of the maximum, where the length increases. With this method of calculation, the heat maximum and the tension maximum half occur at the resting length. Of course, the cross-section of thigh muscle also increases as it shortens. Angle this tends to increase the tension. If the heat is to be compared with the tension which it maintains, then the heat per gram of tissue should be compared with the cohesion per unit cross-section area, or the heat per unit of length with the tension uncorrected for cross-section area. As in the original report, the maintenance heat can also be measured by a sufficiently quick thermopile from records similar to those in figure here. The slope of the curve represents the maintenance heat. Using this method, Ilil-Jang has found values increasing at first with decreasing lengths, and then decreasing. This would seem to be a confirmation of results described above and obtained definitions of resting length. There is a possibility that the shortening heat should be regarded as a fraction of the maintenance heat, for Brown has shown that the sura of these two moieties is remarkably constant in a series of contractions, with increasing shortening as under decreasing loads. As the shortening heat increased, the remainder left for maintenance heat decreased. If this is the case, then the observed decrease in maintenance heat at a sufficiently diminished muscle length may be attributed to the increase in the shortening heat liberated in shortening to that length. With this possible exception, the maintenance heat is regarded as independent of work done and of shortening, but varying W-Itli length and total tension. Required to lower the load in relaxation, i.e., if the extra energy which was liberated for work was left as potential energy in the load, and was not restored to the muscle as heat. At the end of the stimulation, regardless of load, the galvanometer reaches the same point of deflection. This is a confirmation of the experiment first performed by Fenner's degrees when he permitted a muscle to lift a series of increasing loads a fixed distance and measured the heat production. The loads were caught by an electromagnet after they had been lifted the required distance so that the muscle relaxed under a constant small load just large enough to J-U-L back precisely to its original lengths, without any elaborate calibration of the muscle to permit the heat to be expressed accurately in work units. This experiment proved that the extra energy liberated was just equal to the extra work done. The result has since been duplicated in the writer's laboratory by J.B. Hirsch, Ph.D. thesis, with some indication of a slight decrease of heat at the larger loads. Hartree also repeated the experiment in the same way, but added an analysis of the heat production to show the distribution with time. Lai compared an isotonic and an isometric contraction, both of which gave the same amount of total heat minus work. The two differed, however, in the fact that the isometric gave less heat during contraction and correspondingly more during relaxation. The excess heat in relaxation of the isometric muscle represented the greater elastic potential energy of the tensed muscle, while the excess in the isotonic muscle during the contraction represented shortening heat. This experiment, as WTL as the more recent ones of A.V. Hill illustrated in Figure 52, shows that the extra energy for contraction is liberated during the contraction rather than during the relaxation. This liberation of extra energy for work is all the more remarkable when it is recalled that the extra energy is exactly equal in amount to the work which is performed. Indeed, a muscle may be likened to a taxi cab, which charges 1 so much per hour for the rent of the machine, maintenance, heat, 2 so much per mile, shortening heat proportional to distance, 8, so much for each unit of actual work done, force x distance, and 4, so much for overhead, recovery heat. It will be convenient at this point to consider some of the theories which have been proposed to account for this peculiar phenomenon. According to the classical elastic body theory, the muscle is transformed same length and has a shorter natural length. This is similar to the theory of active relaxation, in that both picture a certain amount of mechanical potential energy thus made available for muscular work, and predict that this amount of potential energy can be determined from the area of the length tension diagram. Certainly, the work which the muscle does can be represented on the length tension diagram, and the work bears some relativity onto the heat produced, but this does not mean that there is any direct relation between area of the length tension diagram and the heat. This theory was discredited by the discovery of the importance of work in the muscle economy. The energy liberated seemed to depend not so much upon the amount of the length tension diagram covered by the shorten, as on the amount actually used for work, and those areas of the length tension diagram which were used for work represented energy which otherwise would never have been developed at all. Energy for shortening was liberated or transformed from chemical potential energy at the moment of shortening, not before. A perfectly reasonable attempt was made by Wyman to salvage the elastic body theory by the suggestion that the energy corresponding to these unused portions of the length tension diagram failed to appear as heat, not because it had never been developed, but because it reverted to chemical geotensile energy at the end of the contraction, in case it had not been utilized as work. The end result, however, was the same in either case. The muscle had taken what energy it wanted for work and left the R-uniller for unlicker time, and the heat production could not be predicted from the length tension diagram. Some writers have been unwilling to accept the idea that the muscle does discriminate in some way between the performance of work and imply mere development of tension. Design has argued that the extra energy for work is nothing but internal friction, as if it could for that reason be neglected. Likewise, Lindhard and Bullera have argued that it is nothing but deformation heat, and derives from a change in the shape of the muscle fiber, which loses more potential energy at the big end, where it shortens, than it gains at the small end where it is stretched. This argument loses its point, however, when it is recalled that the reverse process must occur in relaxation. Moreover, all the energy ultimately comes from chemical breakdown in the muscle, whatever it may have been used for. Flout. This is clearly shown by the chemical studies which have been made. Fischer showed that a muscle uses more oxygen when it does work than when it contracts asymmetrically. Both Rothschild and Nagayaji, who measured lactic acid formation, found variations of lactic acid with work in the gastrocnemius muscle, which might have been predicted quite well from the heat production figures already available. However, on May 248 in reading papers on the gastrocnemius muscle, it is important to remember that the arrangement of the fibers is such that the internal work, due to change of shape and asymmetric wish to explain it, the extra energy for work remains a reality which does not fit with the elastic body theory. A point of particular importance in connection with the work heat is the fact that stretching the muscle during the period of contraction has the opposite effect. That is, it causes a diminution in the amount of heat energy liberated after deducting the heat due to the work done on the muscle thermodynamic basis. And from this point of view, it is very similar to a thermoelastic effect. It is, however, opposite in direction to the thermoelastic effect found in resting muscle. As an analogy, one may think of an electric motor which uses more current when it is loaded than when running idly, and which will reverse the process and deliver current if it is turned backward. In addition, It has been found that it is only during the contraction phase that a stretch causes a decrease and a shortening and increase in the heat production. During relaxation, the reverse is true. Thus found that the lengthening associated with ordinary relaxation caused, usually, an additional heat production, and conversely, if the muscle was released and allowed to shorten during relaxation, the heat production was usually diminished. These results were also confirmed by Azuma, but they should be repeated with modern improved methods. 10 rail of heat production, viscosity, and the speed of shortening. It seems very natural that a muscle should lift a light load higher than a heavier load, and likewise lift it more rapidly. The more one tries to explain these simple facts, however, the less obvious do they seem to be. The explanation of the first has already been discussed in connection with the length 10 SI on diagram. It remains now to explain, if possible, the greater speed with which lighter loads are lifted. The original observation on muscle viscosity was derived from experiments of A.V. Hilp with human subjects pulling against an inertia device. He observed that the work performed decreased linearly with increase in the speed of the movement. This suggested that at high speeds, the subjects were really working against a viscous resistance, which increased in proportion to the speed. In other words, the second term of the equation of movement was of predominant importance under these conditions. The importance of viscosity was shown, however, to be overestimated in this theory by the measurement of the work done in sprint running. Using the work measured in this way, the efficiency turn contractions is much larger than it is in the sartorius muscle. Hence, the isometric heat is larger than the isotonic heat, except for certain intermediate loads where the external work is at a maximum. For a discussion of this point and further references, CWO. Then called out to be just as great as in slower movements. If much of the work had been expended against internal viscosity, the externally measured efficiency would have been correspondingly low. Finally, measurements were made of the speed with which various loads were lifted by isolated muscles, or in other words, the force exerted at different speeds of shortening. The results of these measurements over a wide range of forces and speeds showed that the force did not decrease linearly with increase in speed as the theory demanded. This is shown speed of shortening. The upper graph shows that this curve can be represented by an exponential equation after making a small correction for a hypothetical amount of viscosity. This equation leads to no particular interpretation of the data, but the authors expressed the opinion that it was not viscosity, but a chemical delay in the liberation of the extra energy for shortening, which was of chief importance in limiting the speed of movement. It is not necessary, of course, that the viscosity of a muscle should remain constant. Myosin probably belongs to the category of thixotropic gels, which give a lower viscosity the more rapidly they are stirred. Thus, in rapid movements, the muscle may have less viscosity than in the slower movements. This by itself, however, will not explain why smaller loads are lifted more rapidly, because the viscosity is just as high at the beginning of the contraction, whether the load is small or large. If for some other reason a smaller load can be made to move more rapidly than a larger one, then it may be able to continue with still greater rapidity because of the diminishing viscosity of the rapidly stirred myosin gel. Likert and Delru have made a model muscle with a concentric cylinder viscosimeter with a thixotropic gel between the two rotating cylinders. With this model, they have been able to duplicate very nicely the curves of lengthening and shortening of a C and M muscle. It seems likely at present, however, that viscosity does not play so large a role in striated muscle as it does in smooth muscle. From the work described, it was concluded, therefore, that large weights are lifted by muscles more slowly than smaller ones because they demand more excess energy liberation in the process, and this requires more time. This conclusion, however, was reached largely by intuition, and quantitative support for the theory was lacking. Stevens and Metcalf had tried to show that a muscle contracted with constant power, or constant rate of performance of work, or constant PV, force px velocity. This quantity was constant, however, only over a very narrow range of loads. the quantity Y at P plus, where W as frictional resistance gave a better constancy, Fenn and Marsh 0, this being the sum of the rates of internal work against viscosity and external work. But even this did not fit at the larger loads where V appreciates 0. More recently, the requisite mathematical formulation of this theory has been submitted by A.V. Hillingen, who proposed that the rate of expenditure of energy for work in shortening is not constant, but is proportional to P, or to the difference between the isometric tension P and the tension of the load P. This idea was expressed in his equation of muscular motion. In this equation, frictional force is replaced by the constant A. This constant has the dimensions of force and is found to be a rather constant fraction of the isometric tension, so that AP is constant and has an average value of 0.257, while AV is the rate of expenditure of shortening heat. The constant B has the dimensions of velocity and an average value at 0 degrees Celsius of ls per second, wherever I is the length of the muscle. The value of B increases rapidly with temperature, the temperature coefficient for 10 degrees Celsius. The equation states that the rate of expenditure of energy in excess of the maintenance heat is proportional to P. The constant A is not frictional resistance because an equal amount of heat apparently does not appear during the reverse change of shape in relaxation. Instead, it is a sort of thermoelastic effect, presumably accompanied by an equal absorption of heat in relaxation, although there seems to be no certain evidence of such a reversal. Hill's equation is tested as follows. In the first place, the value of A is determined from heat production measurements, similar to those illustrated in Figure 52. Here, the added heat due to shortening ex sini sui, the value of I, is calculated. With this constant determined, the muscle is allowed to shorten while lifting various loads, P, and the corresponding velocities of shortening, V, are determined. Thus, P plus AV can be calculated and plotted against P. The graph, according to equation 1, should be a straight line of negative slope equals B. Hill gives a number of such plots in which it was determined by heat measurements on the same muscle used for the measurements of P and Vi. And it is evident that satisfactory straight lines were obtained. While this is, to be sure, a very nice theoretical confirmation, its real value as a test of the theory is to some extent limited, because it cannot be determined with great accuracy from the heat measurements, and unfortunately, even a 50% variation in it does not affect the linearity of the graph very seriously. Since this equation is of the form ZI equals constant, it may be concluded that the force velocity or PPE curve is a section of a rectangular hyperbola with asymptotes at 5 and 8. Thus, both B and M may be determined by fitting force velocity data to equation 2. B has the dimensions of velocity and A is a force. The values are often not very precisely determined in this way because a rather wide variation of values will give a good fit. The value of B cannot be determined from heat measurements alone, and the critical test of the equation depends upon a comparison of the values of A obtained thermally with those obtained on the same muscle by velocity measurements. No survey of just this sort is available, but it seems unlikely that any discrepancy in excess of the experimental errors will be found. On the other hand, the experimental error in the measurement of a while some further testing is therefore desirable. This theory is nevertheless good enough to justify further consideration of its implications. In the first place, it should be emphasized that this does not represent the total rate of energy liberation, for the equation says nothing about the maintenance heat, which proceeds at a rate determined, presumably, by the length of the muscle, and apparently serves the sole purpose of keeping the muscle in a state of continuous excitation. So far as the performance of work is concerned, the maintenance heat is pure loss. Hill's equation also neglects relaxation and recovery heat, and applies only to the shortening liet and the work heat or the excess energy fraction. In theory, the equation should apply to muscles undergoing stretch as well as to muscles which are shortening. The only difference is that the values of the velocity would be negative instead of positive. The evidence for a negative extra heat production lengthening muscles has already been discussed, p. 502. This is in accord with the theory because P po so that HPOP, which is equal to the rate of liberation of excess energy, would also be negative. In trying to verify the relation between P and V in the stretch, difficulties are encountered due to the danger of injuring the muscle by too rapid a stretch, and the tendency of the muscle to give or slip while it is being stretched. No precise application of the equation to lengthening muscles is therefore possible at this time. Cat's D0, the essential feature of Hill's theory, is that the rate of liberation of this excess energy is proportional to Pope. As an explanation of this peculiar relation, Hill suggests that during shortening under a load, P, a certain fraction P po of the total number of active points or myosin linkod, are occupied in maintaining tension. Only the remainder, Po P, being available to serve as centers for the liberation of energy for shortening and work. It is peculiar, however, that these shortening linkages must then be supposed to liberate energy at a more or less constant rate, so that the longer the time taken in shortening, the greater the amount of energy liberated. This conclusion results from the assumptions which were made, for the total rate of extra energy liberation is BPPP, and the number of linkages engaged in liberating this extra energy is Pope-Enberg linkages. Then the rate of extra energy turnover in each linkage is equal to the ratio of these two quantities, or 6PON, which is constant. But the total extra energy for a constant amount of shortening X is P plus AX. That is, it increases with increase in the load P. The only way in which a smaller number of linkages, each working at a constant rate, can liberate more energy is to spend more time doing it. Thus, the extra energy liberated by each linkage depends upon the time during which it is kept at the business of shortening, rather than being directly a function of the load. It is also, of course, proportional to the amount of shortening, but this is because it takes Loeffler to shorten a greater distance under the same load. Each shortening linkage must be regarded, therefore, as a center for the continuous liberation of extra energy. If this may reasonable and plausible hypothesis, it would provide an explanation for the fact that a muscle liberates extra energy for work. This concept is, however, a difficult one to entertain, and it is made no easier when one realizes, in addition, that the linkages or active points of the muscle which are supposedly engaged in maintaining tension P, that is, the fraction PPO, are also acting as centers of continuous energy turnover, the rate of turnover being independent of the tension maintained. This is evidently true, because the rate of liberation of maintenance heat does not appear to change when an isometrically stimulated muscle is suddenly released and allowed to shorten, either under a large load or a small one. We evidently know too little about the submicroscopic contractile mechanism to make speculation of this type profitable. But it seems that Gell's theory does not provide an adequate explanation for the excess energy. If, however, one assumes the necessity for the liberation of extra energy for shortening and work, then the theory provides a good explanation for the force velocity data. Hill's theory has also supplied a good explanation for the shape of the isometric tetanus. The muscle is regarded as a contractile clement, governed by the characteristic equation P plus A V plus B equals constant, and an undamped non-contractile elastic element composed of tendon, sarcolemma, etc. The contractile state is assumed to be set up instantaneously, and as shortening begins, the elastic component is stretched so that the tension P rises. At first, however, P is low and the velocity of shortening is high. As the elastic element stretches, P becomes larger and the velocity of shortening becomes less, finally reaching 0 when P equals P. According to Hill, the values of A and 6 obtained from force velocity data and heat measurements explain satisfactorily the shape of the isometric tetanus of the same muscle at the same temperature. The explanation introduces one new quantity, which dns UHMN, the elasticity of the free elastic component of the muscle, but methods for determining this are proposed. The theory applies racked well to the frog sartorius muscle, but not so well the tortoise muscle. Various other equations can be suggested, which fit the experimental isonuclear curves, as well as, or in some cases, perhaps better than, the deviation of Hill, but the latter must be credited with offering, in addition, a rational basis for the equation. In any event, this work of Hill's must be regarded as a great step in advance. Henceforth, it is not enough to describe the chiasm of isotonic and isometric curves. Now it is necessary to explain why they have these particular shapes, and the constants of the mechanism must be related to the constants of the thermogram. Muscle physiology is advancing, therefore, to the status of a science, before leaving the problem of the explanation for the velocity of shortening. One further theory may be mentioned which has been advanced by Dr. Kamsey, personal communication. Consider a muscle at resting length OLO, which can develop an isometric tension IPO, figure 54. This muscle is allowed to shorten against various loads, PP isotonically, until it strikes a stop at length. The velocity is measured over this short range of lengths between L and long for the smaller loads. At least the velocity at any load is reasonably constant over this whole range. For loads greater than Lottie, the velocity will become 0 before the length O is reached. 260 maintenance beat does vary to some extent as a function of and of length, as already mentioned. Without the stop at O, the muscle would shorten to the line depot, and it approaches this line with a velocity which is more or less proportional to the distance which it still has to go. Further shortening is impossible beyond BPO because of the internal elasticity of the muscle CXN connective tissue, and it may be supposed somewhat arbitrarily that if this extraneous factor did not enter in, the myosin chains themselves would be able to shorten further, perhaps to the line of Poe. With continued stimulation, Ramsey and Street have actually observed a further shortening of single muscle fibers to a rather similar line. See figure 46. At lengths less than OB, the muscle is in the condition described as a delta STIE. The initial velocity of shortening may be assumed to be proportional to the distance the muscle must go before it reaches the line oppo. If this line is properly drawn, it will of course provide an explanation of the velocity of shortening at different loads. For the larger loads, the line 0 EPO may nearly coincide with the line RPO. But at lower loads, the experimental basis is somewhat less adequate. Actual force velocity curves can be fitted to such a curve very satisfactorily. In this case, distances to the left from low represent velocities and vertical distances represent force. Although this theory is rather arbitrary, it does emphasize the fact that the velocities which have been measured at different tensions are all initial velocities, and Hill's equation has been verified only over this narrow initial range between L and O. The rules which govern the heat production and the velocities as the muscle approaches equilibrium positions along the line RPO have not been worked out. Summary: Hill proposed that rapid movements of muscles were limited pretation by showing that the force did not fall off linearly with increase in the speed. He proposed instead that the muscle contracted more slowly under large loads because it needed more time to liberate the extra amount of shortening energy and work energy required by these large loads. Hill then accepted this idea, but gave it quantitative expression in his equation of muscular motion. While this theory is very ingenious and successful, it requires further and more precise verification. Some of its logical conclusions as applied to the myosin chains are unfortunate. Another simpler but less quantitative theory is proposed from Ramsey, 11, is relaxation active or passive? The answer to this question is fundamental to any theory of contractility. On the one hand, the resting muscle may be regarded as a loaded gun containing stored potential energy ready for delivery on stimulation. Contraction is then purely passive in the sense that it is analogous to the release of a previously tense spring, the mechanical performance of which is fixed in advance. During relaxation, the spring must be stretched back to its original position, and this is then the active haste where work is done at the expense of chemical potential energy. If, on the other hand, contraction is an active process, then chemical potential energy is transformed directly into work during shortening. And during relaxation, the muscle returns to its original resting condition by its own internal elasticity. Simply because the linkages which cause shortening are no longer energized, The supply of chemical energy having been turned off, there can be no question about the fact that a muscle will tend to return to its resting length after contraction, even in the absence of any external force, such as that provided by a weight. An unloaded muscle, or one floating on mercury, will do this fairly well. This active relaxation was more convincingly demonstrated to me by Dr. Ramsay with one of his single muscle fibers, skillfully dissected by Mrs. Ramsay, and stimulated while suspended freely by one end in Ringer's solution. See Figure 55. The weight of this tiny contractile thread in water was so small that the fiber only slowly fell to a vertical position under its point of support. The relaxation process, however, was too fast for this fall, and the much shortened fiber seemed to be so violently extended by some internal force at the moment when the stimulus ceased that the fiber was thrown sideways into loops and then slowly floated back into its original position. In this sense, therefore, relaxation is undoubtedly active. The origin of this relaxing force, however, is uncertain. Two possible forces causing relaxation May 261 personal communication. In writing this chapter, the author has received much help and stimulus from frequent discussions with Dr. Ramsay, to whom grateful acknowledgment is made. His own views differ somewhat from those here reported, are included in the article published in the Handbook of Medical Physics, p. 704, 1943, edited by Otto Glaser. The yearbook publishers, Chicago, Ill Pernin of Ramsay and Street, on single isolated muscle fibers, suspended from one end in Ringer's solution and tetanized. The fiber relaxes more rapidly than it can fall to the vertical position. He mentioned 1. The elastic bulging of the sarcolemma of the shortened muscle, or some similar elastic resistance to shortening. Some chemical reaction yielding free energy, which is used to re-establish side chain linkages between myosin chains, thus forcibly pulling the polypeptide grid back into its resting configuration. The elasticity of the sarcolemma has already been considered. Some force of this sort certainly exists, but it is a question whether by itself it is sufficient to explain the amount of activity which has been observed in relaxation. As to the second suggestion, it is sufficient to point out that it is just as easy to imagine linkages between neighboring myosin chains, which when established will pull the chains out into more or less straight lines, as to imagine linkages between different parts of the same chain, which will cause shortening or kinking of the chain. Thus, we could postulate relaxation linkages as well as contraction linkages. Until we know exactly what the chemistry of the folding process may be, either of Tlia's postulates has equal a priori validity. The combination of phospholipid with myosin, as proposed by Caulker, behaved as a relaxation linkage, but it might equally well have been suggested as a contraction linkage without, apparently, violating any known facts. In most results, there is little to choose between these two theories. The active relaxation theory has the advantage to lie at it. Fits betwixt with the all or none law, particularly as it AI applies to single muscle fibers. These studies, Ramsay and Street, suggest that there is no fatigue of the contractile mechanism proper. If an individual fiber responds at all, it develops the same tension or shortening, whether it is fatigued or not. There are some exceptions or aptogenic exceptions to this rule, which need not be considered here. After a certain number of stimulations, the fiber may suddenly fail to respond, and it is this progressive dropping out of the individual fibers in the multifibered muscle which accounts for the phenomenon of fatigue, as it is ordinarily encountered. In terms of the active relaxation theory, this means that the myosin machinery does not fatigue, but only the ability to respond or the excitability. On the other hand, there is no evidence that the relaxation, assuming it to be active, ever fatigues either, except after the fiber has been allowed to shorten excessively when it goes into the delta state and thus fails to relax, or under conditions which induce rigor. Rigor itself may well be regarded as a sign of failure of an active relaxation process. When a single fiber fails to respond, however, it remains inactive in a state of relaxation, not in contraction. It has recovered from its last contraction And energy has been, this theory was very nicely stated in 1871 by Radcliffe, whose ideas on the subject developed from seeing a rabbit in the death spasms from strychnine poisoning. Is it possible, I ask myself, that life may show itself not in causing contraction, but in keeping the muscle relaxed, and that the doctrine of muscular motion may need reforming in accordance with this idea? Dynamics of Nerve and Muscle. Preface. Macmillan, London, 1871, by C.B. Radcliffe, restored to the myosin chains, which are ready to contract again. But contraction has become temporarily impossible because the fiber can no longer be stimulated. For this theory, therefore, it is necessary to suppose that the process of excitation is something quite apart from the process of contraction, which apparently does not fatigue. On the other hand, the active contraction theory is favored by the heat production data. In the first place, Hilfag has found no heat in relaxation other than that derived either from the lowering of the weight in relaxation or from the potential energy of the tensed muscle. With his improved technique, he did not confirm a moiety of the relaxation heat described by Fenge, which agent apparently came from some active relaxation process. More important than this evidence, however, is the fact, already mentioned, that extra energy for work is liberated during contraction, rather than relaxating. If contraction were merely passive, the energy, heat plus work, liberated during contraction should depend upon the amount of shortening, or upon the area of the length tension diagram covered by the shortening, but should be independent of the load or the tension under which the shortening took place, since, in the last analysis, the total initial heat is a measure of the total chemical breakdown. And since the variation in the LUAT plus work due to load occurs in contraction, it is reasonable to conclude that the variations in the amount of chemical breakdown also occur during contraction. The number of myosin linkages which are activated, therefore, by a given stimulus, or the amount of energy which each linkage liberates, must be determined by the load or other mechanical conditions after the stimulus is over. Contraction, in other words, is not merely the removal of some stop which prevents shortening of a previously stretched spring, but involves some other process which discriminates between different mechanical conditions and liberates more energy in proportion to the work done, in addition to the energy which might be calculated from the length tension diagram and the maintenance heat. 12. Contractures. Thus far in our consideration of the mechanical activity of muscles, we have dealt only with the typical all or none twitch, and the tetanus built up by a series of twitches. This presents, however, a very limited view of the actual situation, for there is a great variety of muscles, not only in the different parts of the vertebrate body, but also in the various species of animals, and the nature of their activities differs widely. Even in one and the same muscle, there may be different kinds of fibers with different characteristics, and, finally, the same fiber under suitable conditions may exhibit mechanical activities which differ markedly from the all or none contractions usually used for student demonstrations. These abnormal responses are conveniently classified as contractures, meaning that they lack some of the usual features of a muscle contraction. Generally, contractures are not conducted from one end of the muscle to the other, and are diminished in tempo. They differ from tetani in lacking the repetitive action potential, which is the inevitable sign of the all or none mechanism. The most careful and critical analysis of the nature of contractures is to be found in the classical review by Gasser, which is as good today as when it was written. Contractures are usually elicited by exposure to chemical agents, which are supposed to act directly upon the contractile mechanism, without involving the conducting action potential mechanism. The same effect, however, can be produced by electrical stimulation. Gerard and Gelfand arranged to stimulate, in this way, the isolated single muscle fibers in the retrolingual membrane of the frog, and were able to observe local responses in the vicinity of the electrodes which were not conducted, and which were graded in intensity in proportion to the intensity of the stimulus. When the stimulus intensity reached a certain critical point, however, the all or none mechanism was excited, and the whole fiber gave the familiar twitch. R.C. Chachtanz. G.C.L. Fan and Bishop, 1936, showed that their local contractures were not accompanied by any action potential, but represented the response of the contractile mechanism alone. Pricking a muscle fiber with a fine pinpoint may initiate either showers of normal impulses accompanied by action potentials, or on occasions local contractures lacking altogether in any electrical sign. It is even possible to initiate, in this way, conducted contractures without action potentials, which are difficult to distinguish from normal twitches in other respects. Thus, the contractible mechanism is by itself conductile. Once the myosin threads begin to shorten in one spot, adjacent chains may also become involved until the whole fiber is contracted. The fact that this does not always occur and that localized contractures are often observed indicates that the all or none action potentile mechanism is to be regarded as something superimposed upon the contractile machinery, which makes its responses more certain and predictable. The nature of conduction in the contractile machinery is not known, and the process is even more complicated by the finding of Stemon and Pratt that non-conductive contractures may occur at some distance from the point of electrical stimulation without any detectable mechanical activity in the intervening tissue. Nevertheless, something has been conducted which can be blocked by mechanical compression between the point stimulated and the point of response. Some of these curious facts are difficult to reconcile with any current theories. They do serve to show, however, that the contractile mechanism can be separated from the action potential. There is usually, to be sure, a close correlation between the magnitudes of the mechanical and the electrical responses of muscles, but this is usually attributable to variations in the number of fibers involved, and does not prove a parallelism within a single fiber. Even with indirect stimulation through the nerve, localized contractures can be produced, as is shown by the contracture which was observed by Feng in the neighborhood of the end plate at certain frequencies of stimulation, and coincident with the appearance of an inhibition of myoneural transmission. If the hormonal theory of myoneural transmission is valid, it must be supposed that acetylcholine or similar agents liberated in an end islate can stimulate both the muscle action potential and the contractile machinery, and probably, under some conditions, may I reduce only contractures similar to those caused by immersing an isolated muscle in acetylcholine. To what extent such local non-propagated contractures occur in vivo must remain for future investigation to elucidate. But it is not improbable that further instances of this sort will be encountered. In any event, the mere existence of contractures focuses our attention upon the obscure processes which intervene between the initiation of the action potential and the onset of the mechanical response itself. From the point of view of an inquiry into the fundamental mechanism of muscle activity, ply contractures offer as good a starting point for observations as the more typical contractions. Qualitatively, at least the two types of activity are identical, the same physical, chemical, and biochemical changes being involved. In some cases, these are better studied in contractures, which are more amenable to analysis because of their slowness. Since, however, muscles in contracture readily pass into irreversible rigor, the responses cannot be so readily repeated by our further observations. On account of this similarity between contractures and ordinary twitches, no special consideration of the mechanism of the former is warranted. 13. Theories of muscle contraction. Throughout the time of recorded history, man has been inventing machines for transforming energy into useful mechanical work. In many of these machines, the source of energy is in the form of chemical potential energy. To transform this into work, it is first transformed into heat, or the original material is burned to gaseous products which occupy an expanding volume. Various of these man-made machines have served as models for explaining the muscle machine. Thus, the muscle has been supposed to be a heat machine, Engelmann, and the shortening has even been attributed to an increase in volume due to accumulating CO2 in the form of a gas, Wackert. Others have attributed the contraction to electrical forces, as in an electric motor. No one, apparently, has reversed this process and used the muscle as a model of a machine for industrial use, although such a machine, shorn of its powerful afferent impulses, would be more efficient and hence more economical than any machine now in use. Even if the muscle could be imitated in the laboratory, however, its practical usefulness would probably not be very great. Because of the delicateness of the parts, its susceptibility to bacterial invasion and to temperature changes, and the fact that it could not utilize chemical energy in any natural form, such as crude oil, without first elaborating it into very special chemical compounds. The contraction of muscles is so commonplace a phenomenon to all of us that it seldom arouses our conscious curiosity. Yet nothing could be more dramatic than to witness a muscle being galvanized into activity by a modern galvanic cell. A mechanical force suddenly leaps forth from a viscid, transparent, inert jelly. Whence comes this miraculous transformation? We have a conviction that the fundamental idea is no more complicated than any gas engine, and the essential moving parts are probably far simpler. So we find ourselves faced with one of the most tantalizing of biological problems. It is tantalizing just because it looks so absurdly simple and easy. It is nevertheless a problem which has challenged man's scientific ingenuity for centuries, and in every age the most recent advances have deluded investigators into thinking that ultimate success was almost within reach. So it is today. The necessary methods of study seem to i.e. ready for use, and much of the necessary information is at hand to complete the theory. It has now become apparent that TENA contractile machinery of muscles has developed from the routine structure of the undifferentiated cells. The fibrous rather than the globular proteins of the cell have become mobilized in the vertebrate muscle into a close order array of contractile units. There is probably nothing essentially new in the mechanism which is not already present in the amoeba. Muscle physiology, like the amoeba, has advanced through the ages by a series of lunges. The pressure of new discoveries gradually builds up in the endoplasm of the research laboratories until it suddenly bursts through the gelated ectoplasm of preconceived notions, letting loose a flood of new ideas and new theories, which advance like a pseudopod into new fields of knowledge, a historical development. The first noteworthy advance of this type was initiated by the great discovery of animal electricity by Galvani, which laid the foundation for the electrical theories of contraction. Even today, one hears suggestions that a striated muscle is a series of plates with alternating positive and negative charges. A second great advance came from the introduction of the kymograph into physiology by Ludwig Dahl. This let loose a host of papers in which the length and tension of the muscles were recorded under every imaginable condition and situation, and with every imaginable device: inertia levers, tension levers, isotonic and oxytonic levers, pre-shortened headings and after-loaded shortenings, and muscles held tight and released with a snap. All these helped to provide precise observations, but provided no important suggestion as to the mechanism of contraction. As rapidly as the science of thermodynamics developed, it found fertile field for its application in muscle. It was Helmholtz, working on the problem of the conservation of energy, who first sought to find out where the energy for a contraction came from. He measured the rise in temperature of a contracting muscle and satisfied himself that chemical reactions of substantial energy value were responsible for the dramatic mechanical effects observed. Later, Fichte went farther in the same direction and actually established an early value for the mechanical equivalent of heat by comparing the heat generated in a muscle with the energy lost simultaneously by the fall of a weight which stretched it. Fichte was a teacher of both physics and physiology, and he was shocked to see that the mechanical efficiency of a muscle was too high for any heat engine, which was limited for its highest temperature to the relatively low value recorded in stimulated muscle, except for the fact that it was contrary to the newly announced second law of thermodynamics, Engelmann's piece of catgut or violin string. Heated with a coil of wire made an excellent model of a muscle contraction. Later, the same model was made to contract by the application of acid, and served a better purpose as experimental support for a colloid chemical theory, Streitmann and Fischer. The impetus derived from heat production measurements has not yet SEO itself, and the results have formed a substantial guiding framework into which all contraction theories must be made to fit. But the results seem to be far removed from the actual mechanism of the contraction. Another of the great advances dates from the work of Fletcher and Hopkins, the Chegg who did not discover lactic acid in muscle, but who learned to control it. They chilled the muscle before grinding it up for analysis, thus eliminating the traumatic formation of the substance. And so they revealed clearly for the first time the fact that lactic acid is formed in contraction and anoxia and disappears in recovery in the presence of oxygen. Thereafter, up-to-date teachers were no longer satisfied to demand of their students technically perfect chemograph records after loaded muscle A-fit Mac RB. Bei der Muskeltätigkeit, Leipzig, 1882. Twitches, but they began instead to devise experiments on muscle metabolism. Thus began the lactic acid era, which was continued by the classical heat production measurements of A.V. Hill-Uscht and the chemical experiments of Meyerhof, which culminated in the Hill and Meyerhof theory. The contributions of these two investigators, working independently, left the whole matter so well worked out in 1904 that one knew instinctively that it could not last. Lyle Hill and Meyerhof did not commit themselves to any particular theory of the actual mechanism of shortening. The prestige of their chemical and thermal analyses of the muscle contraction readily provided a basis for innumerable theories which all regarded lactic acid as the causative agent. The general fault in all these theories was that no provision was made for the transfer of energy derived from the formation of lactic acid into the work to be done. The lactic acid was formed, and the resulting energy was degraded into heat, after which the lactic acid might migrate to, or find itself in, a contraction spot where it could perhaps raise the osmotic pressure, or ionize some protein, solidify a liquid crystal, Clark, or form a solid surface film on the fibrils garner, and so cause contraction. The energy for such a contraction, however, would have to come from the lactic acid protein reaction, the energy of which would be quite inadequate to explain the high efficiencies observed, and the osmotic pressure produced was also inadequate to explain the work. Even if all the lactic acid appeared in one spot in the form of a 90% solution. Meyerhof the problem of an energetic coupling between the products of glycolysis and the products of contraction, in other words, was not properly taken into account. The lactic acid theories WX are also deficient in a far simpler WSEI because it soon transpired that the lactic acid did not appear in the muscle anyway, until after the contraction was all over. So there came another revolution, which ushered in the phosphate era. Now the first detectable change was a breakdown of the newly discovered phosphocreatine, and later the loss of one phosphate from adenyl pyrophosphate. Little by little, the biochemists began to take over the teaching of muscle chemistry, while the physiologists seemed to be turning their attention elsewhere. More and more chemical reactions, more enzymes and coenzymes appeared on the muscle scene, and they all appeared to revolve about the phosphate molecule. If a molecule of adenyl pyrophosphate lost a phosphate on stimulation of the muscle, it collected another from a phosphocreatine neighbor, and the creatine in turn picked up one from phosphopyruvic acid, which, as hexose, had received it originally from adenyl pyrophosphate. Stimulation started the phosphate gears grinding, and somewhere in the chain, the energy of EF phosphate bond was thrown out into the contraction machinery. The phosphate era was one of marked progress, not unaccompanied by bewilderment, for it seemed to lead to no clean-cut theory of contraction which an elementary student could comprehend. The theories of contraction were left to the pictorial powers of histologists, and students of physiology were to be found struggling with multitudinous details of muscle chemistry. With the work of Myers and Asbury, aided by many others, the Miocene era dawned. Once again, the physiologist had some machinery to talk about in connection with muscle. The flame has spread into the remotest corners of biology. The newer knowledge of the configuration of protein structure has illuminated innumerable CMs from the structure of protoplasm to nature of viruses. Muscle physiology also has advanced greatly under the impetus of these new ideas. Every aspect of the subject must now be scrutinized again to see how it will fit into the submicroscopical picture. It is from this point of view that this chapter has been written. Now, at last, we seem to have not only the chemical cycle which provides the energy for work, but also the machinery for doing the work. The phosphate cycle provides the energy. The partly folded molecules of myosin cause the contraction by superfolding. And even more recently, the myosin itself seems to have been fitted into the energy cycle to give us an outline of the modern theory, the surface tension theory. To some extent, it is true that the prevalent theories of muscular contraction do not change as the subject progresses. They are merely revised as to details and brought up to date without alteration of the fundamental idea. An example of this is the surface tension theory. When first presented, the main experimental support was found in the fact that the tension developed in a twitch, like surface tension, is decreased by a rise of temperature, Bernstein 2. Further consideration showed, however, that this evidence was of no value. The difficulties encountered in the interpretation of temperature effects of this sort have already been discussed. In general, it may be said that the tension produced under a given set of conditions may be regarded as dependent upon the amount of energy available, and cannot be regarded as a measure of the true force of the contractile machinery. Originally, the surface tension theory was conceived of as applying to cylindrical sarcomeres, which increased in surface tension at the moment of stimulation, thus rounding up and shortening. When it became evident that the surfaces involved were too small to provide the required forces, smaller units were considered. But even if the surfaces of the fibrils are considered, it is necessary to assume a surface tension of 110 dynes per cm. In order to explain the force exerted by muscle, considering that each fibril is made up of micelles and assuming, with Fischer, that each micelle has a diameter of 5 mz, it may be calculated that the distance along the edges of all the micelles, numbering about 3.7 × 10, in a square millimeter cross-section of muscle is 5,800 centimeters. If the muscle force is taken as 22.0 dynes per mm, Hilfen m, then it is necessary to assume a change of surface tension of only 22.0 over 5,800 or 3.8 dynes per cm. 2xg lie the force. The interfacial tension between oil and water is about 20 dynes per centimeters, so that this is a reasonable figure. Surface tensions against air are no higher and cannot properly be used for comparison since no water-air interfaces can be involved in muscle. If, instead of using the micelles, one calculates on the basis of myosin molecules 10 mi in diameter, there would be 20 chains in each micelle, and the total circumference in each mm of muscle would be 23,200 centimeters, so that a change of surface tension of a little less than 1 dyne per cm would suffice to explain the force. A somewhat similar calculation was recently made by Lang, granting that it is permissible to formulate the contraction process in this way. It still remains necessary to explain what change in the myosin molecule has resulted in the calculated change in surface tension. It certainly seems probable that all the myosin molecules must be involved in the contraction, rather than only those which are confined to the surface. In a micelle of 5 mi diameter, however, nearly half of the myosin chains, each 1 milligrams dm, would be on the surface, so that the reaction of myosin with the soluble products in the solution would be rapid. A greater difficulty with the surface tension theory is in explaining the change of length. If a sphere is stretched out into a cylinder without change in volume, it will become unstable and burr into two when the length of the cylinder becomes actual to its circumference, or when the length is about 3.8 times the diameter of the original sphere. Since muscles or contractile tissues in general can shorten to, say, their resting length, and can be stretched reversibly to twice their resting length, an overall change of 8-fold or more is required. In any event, it hardly seems possible or profitable to regard a myosin molecule, or even a stack of 20 such molecules, as a fluid cylinder. When reduced to these dimensions, the surface tension hypothesis has largely lost its usefulness as a guide to investigation, but it has nevertheless attained thereby a certain measure of truth. 6. Colloid chemical theory, another group of theories, may be classed as colloid chemical, Lilly, McDougall, Streitman, and Fischer, or osmotic pressure theories, Rofe, Wacker, Deinberg. Both relate contraction to moveniance of water, resulting either in asymmetric lateral rather than longitudinal swelling of colloidal particles, or in an inflation by osmotic pressure of cylindrical structures, sarcomeres, which become spherical and thus shorten. In the latter form, the theory is certainly untenable. One, because it does not apply to unstriated muscle percent, because the known chemical changes in muscle could not produce enough osmotic energy to do the work of a muscle. And three, because a shortening of only 33% is explained in this way. It seems unfortunate, therefore, that the theory should have been revived in recent years. St. Georgian. In this latest application of the idea, however, the theory has also been extended into submicroscopic dimensions. And in this form, the distinctions between the Kawai-Teachmeal theory, based on the behavior of catgut or gelatin in electrolyte solutions, the myosin theory, based on protein structure, aradiosmotic theory, based on changes in the hydration of myosin molecules, largely disappear. All these theories now appear to have been reduced to a single least common denominator, or the myosin chain. Indeed, the folding of the myosin chain seems to be the inevitable terminus of Dewey theory, once it is granted that a fibrous protein is responsible for the contraction. Then, when the whole muscle shortens, the long molecules of which it is composed must also inevitably roll up or kink themselves into some more compact structure. Speaking very generally, it may be said that the fibrous protein coagulates or aggregates in a reversible manner. Now, coagulation can be explained in a variety of ways. An increase of surface tension correlated with a change in potential may cause an aggregation of individual particles and a resulting decrease of surface area. Or there may be a decrease in hydration, resulting in loss of the solvate layer, closer peroximation of neighboring molecules, and the establishment of new hydrogen bridges. Finally, there may be more specific reactions which permit neighboring points on a chain to combine together, thus drawing the long chain into loops. Thus, in the original theory of Meyer, a change in PLL was supposed to bring the myosin to its isoelectric point and cause a sort of intramolecular Zwitterian formation, whereby adjacent U and NH2 groups became Cu and NHJ, and so combined together, causing the molecular chain to coil upon itself. Other stereochemical suggestions have been proposed by Astbury to account for the various degrees of folding of the fibrous proteins, as illustrated by the extended 3-keratin, the partially folded akeratin, and the supercontracted keratin, which results from the treatment of hair with steam. The work of Mark on the stereochemistry theories of the stretch-in of rubber are of interest in this connection, for they suggest the great range of lengths, which results from molecular rearrangement, cosinal merely VE rotation of a molecule about its free CVC bonds. The problem of coagulation, therefore, has reached the molecular stage, and so have theories of muscular contraction. In some measure, it is true that many of the older theories have in consequence become one. Present discussion is not concerned with a theory of contraction, but rather with the chemistry of the folding of myosin molecules, myosin fold. In the older theories, the folding of the myosin was due to the formation of lactic acid, and later to the alkylatiety resulting from the breakdown of phosphocreatine. According to St. Georgi, Gio is due to the liberation of potassium. In support of this, he can show that artificial myosin threads will shorten when exposed to XD, it is known. Furthermore, that K is liberated from muscles on stimulation, and that muscles will contract when exposed to KD. It is hard to believe, however, that the myosin is not exposed to the same amount of K before stimulation as afterward, and it is quite as likely that the loss of K from stimulated muscles is the result, rather than the cause of contraction. It is asserted unbound or ionized at the moment of stimulation, but this conclusion is nothing more than a description of his experiment in W. Hitch. It was found that more X can be extracted from muscle when stimulated than when resting. The decrease in volume which occurs in contraction is no Euchar roof of this theory, even though it be true that the solution of X in water leads to a decrease in volume. And even though the decrease in volume is approximately what might be expected if the amount of X lost from the muscle were suddenly dissolved, there is still no reason to believe that all the X in muscle is not dissolved or ionized, even before it is stimulated. Indeed, It is impossible to make up the necessary osmotic pressure in the fibers unless nearly all the potassium is in ionized form. Most of the dekinase in volume is quantitatively explained by the breakdown of phosphocreatine, CP 482, so that there would seem to be little volume change remaining, which could be attributed to X, the crucial experiment which elucidates the relation of K to muscle contraction, still awaits discovery. Muscle contraction has also been attributed to the liberation of calcium, but the evidence for this theory is even more fragmentary than the evidence for the K theory. The theory is based largely upon the contraction which results when a muscle is immersed in isotonic solution of Kali. Such a concentration is far beyond physiological limits. Some evidence for a liberation of Qi in contraction has been advanced, but other attempts to demonstrate such a change were unsuccessful. Qi degrees Ka has been found to serve as a coenzyme in adenylpyrohosphatase reactions, but this by itself means little. Mg is also a coenzyme in other reactions of the energy cycle. Further evidence concerning the role of Qi is therefore required, particularly some better proof that the concentration of free or ionized Qi is increased by stimulation, myosin as an enzyme. The most recent theory of myosin shortening originated in the discovery that myosin can act as or is an enzyme for the removal of phosphate. Adenyl pyrophosphate, moreover, when added to a myosin solution, causes a shortening of the molecule's asa, indicated by a decrease in the double refraction of flow, needham. In this respect, the AP is highly specific and produces its effect in a much lower concentration than is required by other substances. Thus, a 50% decrease in birefringence is caused by 1.5 m uracil on m monovalentation, 0.2 m bivalentation, or a PL tension, or a PLL of 10, while AP has the same effect in a concentration of 0.04 m and can cause 15% decrease. In these are striking facts, but it is still not clear just how they explain the combination of AP with myosin WS and the AP with myosin. Immediate cause of the shortening and that relaxation occurred at the expense of free energy derived from the splitting off of 1P, 4 from AP. It may be, however, that this energy is released during contraction and is the immediate source of the muscular work which is performed. Furthermore, the actual shortening may result from a phosphorylation of the myosin by the PO4 donated by AP, as suggested by Kalker, CP 485 above. Mention has already been made also of the theory that the shortening is due to the effect of K-abdodh, also causes a decrease in birefringence. Daly has supposed that the stimulus to the muscle liberated Qi, which causes the contraction by assisting in the transfer of PO4 from AP to myosin. Possibly there is truth in all these theories, and contraction should be regarded as intimately dependent upon K as well as AP, PO4, and myosin. In any event, the discovery of Enclart and Ljubimova seems to be the starting point of a new era in muscle physiology. In this new era, the coupling between myosin and the energy cycle will be clarified. This is now easy, for myosin itself has become a component in the phosphate cycle. At last, the contractile machinery has become more than a structure which is passively acted upon by lactic acid or some outlet metabolic product. It is as if steam had at last been admitted to the previously empty cylinders of the muscle machine. The manuscript for Tully's Sectioid was received for publication in it.